office wear as wrap dresses. We have the casual wear and rich auntie as well.
We have kids dress. We have necklaces, fans, and materials.
Okay, my name is Francisca. I deal with all kinds of African wear and African crafts in handmade. Example like this. This is a talking drum. It's made with animal skin with a symbol at the front and the back. And I have the clock made with African print. This is African print. And there's a Ghana map inside.
We have magic bag. This is called magic bag. This is how it is. And when you finish shopping, you just fold everything back. This.
Good morning. So, it's last day. I hope everybody is excited to end on a high and have some drinks this evening. So today we are keeping with the spirit of linking with our brothers in the diaspora. This morning, I'm very pleased to introduce one of my friends and collaborators who has been engaging with us over the last several years and who has, on many occasions, promised and not showed up. You knew I would call you out, right? This year he said he has to come. So even though his schedule was, tight as usual, he made the effort to, break the jinx because he knew that after this year that would have been it. I would just write him off and then, our friendship will be over. So I'm very happy to introduce Musa that many of you have seen over the last few days since he arrived. Musa, I met Musa first in University of Cape Town through some of my collaborations I have there as part of the Creek Africa Network. And since then we've been good friends. He's bought me some good wines and some steaks. And we've always been in touch. He then also turned around and gave me some work to do for him on the Human Cell Atlas. I'm sure he'll talk a little bit about it here. He's one of the leaders of the equity initiative of the Human Cell Atlas. And when they wanted to enter the continent, he asked for my support and assistance, and I think we've made some progress in that regard. We've had a number of workshops, including one here, to engage the young people on single-cell transcriptomics. Anyway, so like I said, Musa is in the diaspora. He did his education in France and the U.S. He did his PhD in the New York University School of Medicine and the Rockefeller University. After that, he did a postdoc at Institut Pasteur on a U.S. National Science Foundation fellowship. After about how many years? After several years, he moved back to the continent to University of Cape Town, where he spent several years there working at UCT. He's now back in Europe, in the Radboud University in Nijmegen, in the Netherlands. He has his own group there, and he's the chair of the Epigenomics and Single Cell Biophysics group. So, ladies and gentlemen, I present Musa Flanga.
This was the last time. This is the very last time. It's not allowed to come back. Thank you so much for the really kind introduction. I knew that there was going to be some pain involved, so, I felt like a little kid getting an injection, I was just waiting for the needle. So, I'm over that. So thank you so much for inviting me. I'm really excited about basically being able to interact with your institute and your colleagues here. And I also am very, very impressed by what you've done here over a decade. And I think you're really, really shining black star. So I'm very impressed, and anything I can do to help, please let me know. So today I'm going to talk a little bit about some work we are doing in innate immune memory. And I just want to say ahead of time, I'm not an immunologist. Actually, our lab works predominantly on transcriptional regulation, gene regulation. And just jumping straight to that, really, we study epigenetic regulation in these contexts, from chronic inflammation. Here, I'll use this pointer. From chronic inflammation to immunosenescence to oncology and vaccination. And we're very interested in how the 3D architecture of the genome, which I'll describe in a moment, and epigenetic modifications to this, along with non-coding RNA, are able to regulate transcription. So we really study this in these contexts because if you look at genetic variation, 90% of the variants that we see are all occurring in the non-coding space. And these non-coding variants are influential to non-coding RNAs that are transcribed from this. And there was a talk a couple of days ago about non-coding RNAs, so that was nice to introduce the subject. And that allows for variable regulation of multiple types of genes, including immune genes. So 9 out of 10 of the variants that we see between all of us in this room are emerging from this non-coding space. And for this reason, I think it's become extremely important to be able to link the non-coding variation that we see, which is quite different from coding gene variation, to how that is manifested in transcriptional regulation of coding genes and ultimately in phenotypes. And so we can also use this framework to try to understand something way more important, which is how transcriptional regulation influences particular systems, like the immune system. And so as Gordon said, I think one of the big reasons that we started to work on the immune system was not because we were interested in the immune system, but because this type of variation and the study of epigenetic transcriptional regulation was very easy to do in a very easy primary cell accessible type, which is the immune system. So we chose this system to be able to understand fundamental concepts of regulation. And it was when I was at the University of Cape Town, and our lab was, I think, like we were 1 or 2 years away from actually moving back to Boston, to the United States. And then I met this guy, Mihai Natea, and he was giving a talk on immunology. And we had just discovered a particular regulatory mechanism, and he was describing an observation that he made about 5 years before. And I love to go to talks that I am not working on at all. So another part of our lab works in optical physics, because my background is in biophysics. And so we build microscopes and things like that. And it's because I try to have as much scientific range as possible. So I went to this immunology talk just because I was super interested in it. And he ended up really changing the course of what a lot of our lab would do. And the way to tell the scientific story about my relationship with Mihai is like this. So the first vaccine was invented in 1798, and it was invented against smallpox. And I think many of you know the story of how Jenner sort of looks at the milkmaids, and they have these pussycles, and he makes this really strong intellectual leap between their protection from smallpox and the fact that they have these pussycles. And so he uses that to inoculate people to protect them from smallpox, and that becomes our first vaccine. And we know that lots of really famous people had smallpox or died of smallpox. So that was the first vaccine. But then what happened is we had vaccination become widely used around the world, and the live vaccine of BCG for tuberculosis started to be used in many different parts of the developing world, including Guinea-Bissau and West Africa. And this really curious observation was made. So this is 2 centuries later. And it was that actually it seems that live vaccines seem to enhance resistance towards unrelated infections. And there are a whole bunch of other principles here, but most importantly, it seemed that even in areas where tuberculosis wasn't endemic, when you were able to get a BCG vaccine at birth, you had dramatic protection not only from tuberculosis, but also from multiple other infectious diseases. And in fact, if you went back and looked at the time when BCG was invented at the Pasteur, after it was deployed in parts of Europe and the United States, you also saw dramatic drops in dysentery and cholera and all kinds of, infectious diseases as soon as BCG vaccination was started. So this tended to suggest that there was some sort of heterologous protection that was being conferred by BCG. And this is, I think, where the observation was that perhaps actually BCG was providing some sort of non-adaptive memory to the immune system for protection. And so that basically is where Mihai's observations that he made about 12 years ago entered the picture. And to really understand that. And again, I'm just saying this because I'm not an immunologist. I'm going to simplify this because many of you in this room are way better immunologists than I am. So I'll just simplify the explanation like this. Mihai's observation centered around the differences between adaptive and innate immune memory. Now, when we look at innate immune memory, what we see is that, the ability of innate immune memory to be able to form memory is essentially the ability of innate immune memory to program two cell types. And those memories are permanent. And those two cell types are the T cells with T cell receptors and B cells with antibodies. And there you have the physical basis of that memory. And what's really important about that is the physical basis of this memory means that that memory is contained either in the TCR receptor or in the antibody itself. And if you look at innate immune memory, well, innate immune memory has a different component. Innate immune memory is regulated at the epigenetic level. And innate immune memory, the physical basis of that memory is an epigenetic modification at the promoter of a specific gene. Okay, so I'll say that again. Innate immune memory is the acquisition of a specific histone modification at the promoter of key genes important for the immune response. And that modification is the acquisition of this H3K4ME3 mark, and that seems to be discretely positioned at the promoters of what we call trained immune genes and what Mihai termed trained immunity. And trained immunity has this important hallmark where you're first exposed to an infection, and then upon exposure to a secondary infection, you have an enhanced immune response. An enhanced immune response in this context means that you express a whole suite of chemokines and cytokines at a much higher transcriptional level, and also can be detected at much higher levels at the level of protein. And this enhanced level of expression confers enhanced protection to these individuals. And it would seem that things like BCG are able to serve the role of training the immune system so that these particular marks are acquired. And we have to understand what acquiring an H3K4ME3 mark means. It actually converts transcription from being stochastic. So this is the fundamental basis of transcription, is stochastic. So here, these red squiggly lines are RNA, and they make a gene that turns cells green. And you can see, like, when these cells receive a stimulus and you have stochastic gene expression, only 3 of them are making the high amounts of this RNA. But if you have robust gene expression, you get many of these cells all expressing this high level of RNA. And the way that you convert transcription from being stochastic to robust is you change the epigenetic character of how those genes are going to be expressed. And that's through writing this H3K4ME3 mark on the promoter of these genes prior to their gene expression. So this poses a very important question, which is, how is it that we can position this mark in the immune system only at the genes where we would like to achieve this robust transcription and immune response? And so a key component to this is nuclear architecture. And I'll just try to summarize this because this is many years of work, and our lab and many other labs have contributed to a fundamental understanding of how the 3D architecture of the genome is organized. And the way that one can understand this really, like at a first principles level, is that in each one of us, we have DNA that's over 1.5 meters long in each cell. And as Gordon said, we have 37 trillion cells, and each cell has DNA that's 1.5 meters long plus, but it's gossamer thin, spiderweb thin. So this DNA has to somehow be compacted and folded to fit in a space that's 1 million times smaller than the size of the cell. And the way that's done is by folding and looping, and is essentially a feat of origami. But when you do this looping and folding, what happens is the genome then gets organized into these topologically associating domains. And a key feature of these topologically associating domains, or TADs, is that they regulate transcription, and their interactions are necessary for transcription of genes to occur. And so in those TADs, we can have these non-coding RNAs that I mentioned earlier, and these non-coding RNAs are able to organize the 3D looping and also activate or inhibit transcription or recruit things like RNA polymerase directly to the promoters of these genes. And so when we take the combination of all of these things, we can see that they can also contribute to the existence of active marks on the chromatin or closed marks on the chromatin. So that means that you change from this state of closed to this state of openness. And most of the time, the genome is in this state. And more importantly, there's an intermediate state or a poised state where you can add this mark, but unfortunately, transcription is blocked, and this keeps the transcription in a state of just high readiness for the activation of that gene. And that's how you get robust transcription. So along these lines, you can see that when you have high H3K4ME3, you get a robust transcriptional response. And when you don't have it, you have a methylated state of DNA, and the DNA is closed. And so now, with this understanding, we can see, oh, okay, now I understand why high levels of this would lead to high transcription of these genes. And the question about how can we get that H3K4ME3 mark directed to specific gene promoters becomes more relevant. And so several years ago, our lab showed that actually in the 3D architecture of the genome sits a class of non-coding RNAs that can recruit the enzymatic machinery that can then write that H3K4ME3 mark to the promoters of these immune genes. And we showed this using multiple techniques where we could look at 3D genome and look at the genes being transcribed, etc. But key to this is this one of these non-coding RNAs, for example, amlilo, that regulates these chemokine genes. And so in this rather complex cartoon, you can see that the 3D architecture is brought together by this class of non-coding RNAs, and then this key non-coding RNA is able to write the K4ME3 mark at the promoter of these chemokine genes. And that in turn, that activity occurs during vaccination or during trained immunity. And so, for example, beta-glucan, which can induce trained immunity, can then trigger the expression of these non-coding RNAs. And by triggering the expression of these non-coding RNAs, you get the trained immune response. And it's also important to understand that these activities that are given by vaccines or beta-glucan or BCG are able to write these marks in the bone marrow, because that allows this mark to have permanence. So if you write this mark in progenitor cells, then in these myeloid progenitor cells, they can then acquire this mark, and it can be propagated to the periphery and be existing in monocytes. And so you can have that pathway that I just showed you, which is also coupled by metabolic changes, which is all driven by this set of agents that are able to drive the trained immunity process. And so it's important to understand that this trained immunity process is writing, but is also reversible, right? So you can erase this mark and write this mark continuously. And this is a feature of the innate immune system that doesn't exist in the adaptive immune system, because unless you got rid of every T cell with a particular TCR or every B cell with a particular antibody, you have that memory essentially for the rest of your life. So this is kind of important when we understand trained immunity, especially in terms of BCG vaccination, because as Mihai showed in this study, actually collaborated with in this paper, I'm sorry, is BCG vaccination can induce this long-term functional reprogramming of neutrophils. So this essentially explains to us mechanistically how we can get this heterologous protection from BCG vaccination. And so one of the key things around this is that we know that therapeutic interventions which can modulate trained immunity, like inhibiting it in cardiovascular disease or rheumatoid arthritis or gout or IBD or in metabolic diseases, are beneficial. And we also know that promoting trained immunity can be beneficial through different types of live vaccine and other vaccine approaches, or in reversing sepsis, or in clearing infection. And this range of responses that we see is telling us that these diseases are actually pathogenic forms of trained immunity, and those are contrasted with beneficial forms of trained immunity which occur through vaccination or other interventions that improve the response to beta-glucan and the stimulation of trained immunity. So this is really important because trained immunity seems to have two sides, a pathogenic side and a beneficial side. And a lot of the diseases that we have of chronic inflammation are essentially dysregulation of this trained immunity process that are driven by the innate immune system. So coming back to this fundamental idea is, do different genotypes in the population respond differently to BCG vaccination? Hence, the ability of some people to be well protected by BCG vaccination and others to be less well or more poorly protected. And so this study from Reinhold van Krevel and Mihai in 2018 showed something really interesting, where BCG vaccination could protect some individuals from yellow fever virus. But as you can see in this graph, if these individuals had this SNP at the promoter region, of IL-1 beta, you can see that their IL-6 and TNF responses, if they're AA, are like a third of those of the people who are GG. And so since IL-1 beta has this apex role in the driving of trained immunity, what this essentially meant is that for the people who have the AA version of the SNP at this site, their protection from BCG vaccination was slightly or significantly impaired compared to those with GG. Now, you could be asking me, like, what's the prevalence of GG in the population? And it turns out that that prevalence is quite high. So in this room, 1/3 of us are going to be AA, 1/3 of us are going to be GG, and 1/3 of us are going to be GA. So this is one of the most common SNPs and seems to exist in balance in populations throughout the world. Now, there are demographic differences, and I did some research before I came to Ghana. So Ghana is like the 1/3, 1/3, 1/3 example. But in, for example, certain parts of Europe, many people are GG. And in East Africa, 50% of people are AA. And so this is very illustrative of some kind of selection that's also occurring environmentally that seems to modulate the expression of IL-1 beta. And so because of this SNP, we kind of decided, well, Mihai and I were like, well, what is this SNP actually doing? And so here, as I told you, IL-1 beta is at the apex of this process, and we have beta-glucan and BCG drives IL-1 beta, and you get TNF, and you get lots of lactate, and that's promoting trained immunity. And the other side of trained immunity is tolerization, and basically you get diminished IL-1 beta, you get less lactate, less IL-6, less TNF. And this is essentially showing us that this two-sided process is really showing that inflammation is a biphasic process where you have inflammation, which is coupled to glycolysis, and anti-inflammation, which is driven by oxidative phosphorylation. And so we looked at the SNP in terms of what it does to trained immunity with beta-glucan, because clearly it was doing something with BCG, and we saw that it continued to be maintained, and you can see that here we have BCG and beta-glucan, and you can see here that after adding beta-glucan, you have very high levels of IL-6 and very high levels of TNF. And, oh, my build is not working, but if you segregate this by genotype, you'll see that the people who are GG make less IL-6 and less TNF alpha. And so we looked where this SNP is, and this SNP actually seemed to fall in an uncharacterized transcript that lay just upstream of IL-1 beta. And we knew this was an uncharacterized transcript. I'm sorry, the bottom of my slide is not showing here, but because there are epigenetic marks here associated with active transcription that only occurs in CD14 positive monocytes. And that's really important because it's saying to us that this region is actually a non-coding RNA region. And when you make IL-1 beta, you also seem to make this non-coding transcript that goes in the opposite direction. So we did some specialized types of sequencing called cage sequencing, where we sequence from the 5' direction, and we were able to see that indeed when you give monocytes strep or salmonella or a whole bunch of other stimuli, candida, from multiple donors, you get a transcript that goes in this direction. And we call this transcript Amanzi because what we could see here is that under the conditions of LPS, you get more expression of this particular non-coding RNA, or under the conditions of beta-glucan, you get more expression of this non-coding RNA, which is curious because it's telling us that there's this bidirectional transcription occurring here. So to keep our story short and punchy, we actually depleted Amanzi, and what we were able to see is that when we turned off Amanzi, we got lots of IL-1 beta, suggesting that there was some relationship between Amanzi's transcription being able to somehow regulate IL-1 beta's transcription. And we called it Amanzi because Amanzi means water, and we named this a non-coding RNA that antagonizes inflammation. So we went on to look at this really carefully, and I'll just summarize some of our data at these different points. But the first thing we were able to discover is that Amanzi is regulated by a previously known enhancer RNA, which actually turns on IL-1 beta and Amanzi. But more importantly, it seems that when Amanzi gets silenced, you also silenced another gene down here, which is on the genome about 80 kilobases from Amanzi and IL-1 beta called IL-37. And IL-37 is the opposite of IL-1 beta. It is at the apex of anti-inflammation. It doesn't exist in rodents, but what it does is that it turns off IL-1 beta's activities and it drives oxidative phosphorylation. And so it seemed that Amanzi's role was when you make IL-1 beta, it seemed to be able to turn on IL-37, and if we silenced Amanzi, we lost expression of IL-37. So kind of looking at this more carefully, we then realized that perhaps Amanzi loops over to IL-37, and we did a number of experiments where we were able to show that indeed what happens is that Amanzi loops causes the DNA to loop and loops over to IL-37 and turns on IL-37. And when we silence Amanzi, we lose this long-range contact and we cannot turn on the negative circuit for inflammation. And that's why when we silence Amanzi, we get way more IL-1 beta. And it seems that, and I won't go into that because for lack of time, it seems that this process of looping that Amanzi does over 80 kilobases takes about 5 hours. And in those 5 hours, you establish inflammation. And then after those 5 hours, you can turn off inflammation by turning on IL-37. And the IL-37 negative regulation is quite complex because IL-37 can essentially recruit a transcription factor, and that transcription factor is actually SMAD3, and SMAD3 pairs with IL-37, forms a partnership with IL-37, and switches off IL-1 beta and IL-1 beta eRNA. And this is, IL-1 beta eRNA is really important because it is the TLR4 sensitive activation of IL-1 beta that turns on inflammation in many different contexts. So just to summarize here, IL-37 is able to turn this on. So just in the last couple of minutes, I want to ask the most important question here, which is, well, how do we get to the resolution of this circuit, right? Because remember, we started off by a SNP that's in 1/3 of the population, and we had variable responses to trained immunity. And then we went and we dug a little bit into this SNP, and we find out there's a non-coding RNA, and we dig a little bit more, and we find out, well, the non-coding RNA seems to have this really interesting property where it loops and turns on an anti-inflammatory gene. And lo and behold, after doing all of that, we figure out that, hey, wait a minute, there's this whole circuit that balances IL-1 beta with IL-37, and it's wired by this non-coding RNA. But it still doesn't tell us why this SNP, which is variable in all of us here, would be so influential to this process. And so to do this, we collaborated with an expert at the Radboud in mass spec, Michael Vermeulen. And what we did is we synthesized the RNA and we made a bait where we put it in the nuclear extract of and cell lysate extract of different genes. Then we did mass spec and we looked at all the proteins that bound to that RNA, and we did it for the G form and the A form. And we wanted to see, like, does the G and A form, do they bind to different things that make it that they behave differently in all of us here? And we did that, and the results were, well, I've made the lettering really small and everything really small because there's very little difference between A and G. And this is all published, I should tell you, so you can go and look at it, and if you don't believe me, you can go check. So the key insight, I guess, came from RNA epigenetics. And RNA epigenetics is the fact that in almost every single coding gene, there are modifications to the RNA. And those modifications include methylation at different parts of the RNA. And the one that we know the most about is called the M6A methylation. And that means that if you have an A in an RNA and it corresponds to a particular sequence, and that sequence is called the DRASH sequence or the H-card sequence, so it can be in both directions, so it can be DRASH or H-card, and you have any one of these sequences in your RNA, the A that resides there can be modified so that it's M6A, right? And so what we found is that here is the canonical M6A site in the H-card direction. And Amanzi, if you have the A version of Amanzi, there you go. There you have it right there. This is the H-card direction. This is the DRASH direction. You can see here, it's just the reverse of that, and you have this M6A here. So now, when we methylated our bait, we found all those proteins that are important for the regulation of this M6A site, and we saw that this SNP creates an M6A site, and that changes the methylation status of people who have a MANZI and an A at this position. And so we went on to show that, in fact, I won't take you through all of this, but we went on to show that actually the A form of a MANZI is more stable than the G form. And because the A form is more stable, and in fact we can identify the A form after 12 hours of LPS exposure really easily, and not the G form, and we can do this by lots of techniques, or we can do antibody pull downs. And because it's more stable, what can happen then is that it's able to actually protect the A form of a MANZI. And because a MANZI turns on IL-37, and this is more stable, that allows us to make more IL-37, and that allows us to be more anti-inflammatory when you have high MANZI. And if you have high MANZI, you get high IL-37. If you have low levels of MANZI, less stable, you get low amounts of IL-37, but more IL-1 beta, right? So this is flip-flopping back and forth. And what we could do is we could do clever things with this, is we could block and use different forms of locked nucleic acids and silence a MANZI, and that allowed us to make more IL-1 beta. And conversely, we can see that like this enhances training 7 days after you train. So this is telling us that silencing a MANZI will dramatically boost the levels of IL-1 beta that you make. More importantly, we can manipulate this because, again, it's an RNA. We can use an RNA that targets people with the A form or the G form. And when we do that, over here, we're affecting how much IL-1 beta they make. And we can do this in primary monocytes, where we can actually modulate the response that people have just by delivering a locked nucleic acid with a different version. One base, A or G, will change you from making lots of IL-1 beta to less IL-1 beta. And so this really summarizes a lot of what we were able to discover by this, but also the fact that maybe we could even use this to rejuvenate different vaccines, because in fact it turns out that IL-1 beta is really the key regulator of how we have a robust mRNA vaccine response. And you may have noticed this, but the first vaccines we had, the first vaccines of COVID we had, were really great. And as the vaccines got cleaner and cleaner, we had a decline in the quality of protection from these vaccines because they were making less and less stimulation of IL-1 beta. Okay, so just to finish, it's clear that inflammation is a biphasic process and is highly guided in the innate immune system by these epigenetic actors that act to regulate trained immunity. And in the studies I've shown you, we've identified non-coding RNAs that regulate the mechanisms of trained immunity, and also have negative regulators of trained immunity, such as MANZI, we've also identified, which discretely regulate IL-1 beta, but because they're regulating IL-1 beta at the apex of the trained immunity program, they're able to modulate this program so we don't have like, you know, an overshoot of inflammation. And so the curious thing is that nature has placed the pro and anti-inflammatory genes within the same TAD, which I described earlier. And this TAD has a pro-inflammatory side and an anti-inflammatory side. And each side drives two different programs of metabolism. And here, down here, I hope you can see that at the back, is that these are the things that happen to people in general who have GG, and these are the things that happen to people in general of AA. And more importantly, every single person in this room is affected by the left or right side of this, depending on your genotype. So with that, I want to thank you for your attention. And this was done by a PhD student, Ezio Folk, from South Africa, whose outstanding work has done this. And he was guided by a former staff scientist in the lab, Stephanie Fanucchi, who really like worked very closely with Ezio to oversee this study. And obviously the other people in red from the lab contributed to this tremendously. And we started this company several years ago to drug RNA. And of course, my collaborator and good friend Mihai, Michael Vermeulen from the mass spec lab, Leo Houston, who also works a lot with Mihai. And the part of the study that I didn't talk about was from Patricia Severino, where she was able to show that with our data, that in sepsis, people who are AA are far more susceptible to death from sepsis because they are more tolerized. All right, thank you for your attention.
Great. Questions? I see the malaria people's hands are up because a lot of what Musa has talked about, you can find parallels in malaria. So Lars is here. I'm sure he doesn't like the theory of tolerance, but I think that I think this is really speaking to the tolerance theory. And those of you who were at my inaugural, you remember that we talked a lot about this, and now Peter and Yao and Cassie have published a paper about this tolerance thing. So anyway, yeah, Jean. DJ has the microphone at the back.
I didn't take it to cut in front of anyone. They just handed it to me because I'm in the back. So first of all, beautiful talk. I'm glad this was early as a morning person. This is difficult to unpack. And let me just make some statements, and you can help me and the rest of the folks in the crowd try to understand this a little better. They may be way smarter than me, but when I'm thinking about this, specifically with IL-1 beta, what we know and what we've shown, for example, in children with malarial anemia, there are different haplotypic constructs that come about within the promoter. We have high producers of IL-1 beta, low producers, and then those map to susceptibility across time, malaria, whether it becomes severe. So at the one level, there's this inheritance and innate immunity that we work on. Now there are these master regulators, right, such as the things that you found and how they do that at a distal place. So what I don't understand, and maybe you can help me and others, is everything you said is really convincing. But in the end, walking around in the environment, it's this balance of pro and anti-inflammatory cytokines across time. So I have my germline immunity with this expression of IL-1, okay? And then I have epigenetic regulation. I'm thinking that it must be a really complex model that's a gestalt of the sum of inheritance. So while what you show is important, there are other SNPs that change transcription factor binding sites. And then here's the question. Does it get even more complex in that these long non-coding RNAs, in this particular case, can also affect microRNAs, which are setting at a top level of regulation? So some statements, not so much of a question other than the interaction with microRNAs, but I'm just trying to understand this. It's a complex problem that seems very difficult to have a summation of what actually happens when I get an infection with all this complexity going on. Thank you so much for the beautiful talk.
All right. You want to comment? Sure. Once we get the microphone to Jean.
Sure. That's a really great question. So we have looked for other regulators in terms of the non-coding space that contribute to IL-1 beta transcription. What I didn't go into is probably something that I should have, which is, the IL-1 beta exists in what we call a super TAD. So there's IL-1 beta, IL-37, and it seems like this TAD is separated from the neighboring TAD where you have IL-36, G, F, IL-38. It's all IL-1 family members, IL-1RA. And when you activate IL-37, it seems like you dissolve the boundary in these two TADs, and now the entire IL-1 regulation seems to be highly coordinated. So now enter all the SNPs you've talked about, and we've looked very carefully at these SNPs all the way to IL-1RA. What we've seen is that people who possess certain SNPs along, they sort of move as a haplogroup, and these SNPs are highly connected across the two TADs, and they influence the secondary effects of IL-1 beta. Now, not to go too much into IL-1 beta biology, but IL-1 beta has many negative regulators, many. And because it's such a dangerous cytokine, it's actually produced in very low amounts. And its regulation is even occurring at the protein level by making a proform that then has to undergo very careful maturation. IL-37 is also a member of the IL-1 beta family, by the way, and all of these were discovered by, the great Charles Dinarello. So understanding exactly what you said means that you kind of have to take into account all of these. We've seen that SNPs, especially in IL-1RA, so IL-1RA is the negative regulator of IL-1 beta, are highly influential to IL-1 biology. I think what is really important to take away from this message here is how something that we could not really conceive of in nature, which is that the positive and negative regulation are wired into the time it takes for this looping to occur. So you need to establish inflammation, and it's not like you have a protein-driven process. You have a 3D genome architecture process that is balancing when you're on for inflammation and when you're off for inflammation, right? And that's like some sort of extra elegance that we could not conceive of until now. And I think the variation that exists in the human population tells us that it's really important to have variable responses in this balance for whatever reason, because they're under positive selection. And the last thing I'll say, and this may help malaria researchers, everybody researchers, there are over 200 papers on this SNP until we publish this, so nobody knows how this works. But if you're GG, you're 2.5 to 3 times more susceptible to mental illness or different types of illnesses. So it's really a very significant SNP to study because it's influencing IL-1 beta. And the other IL-1 beta SNPs that we know are not as preponderant in the population. So maybe that'll help you to see why focusing on this could be very important, especially if it's in linkage with another SNP that may be more rare.
My question is much more simple.
Sorry, my answer will be very simple then.
What is the interaction with the adaptive response, particularly the T-cell response? Because say in a malaria infection, which is what I do, you get this very early inflammatory T-cell response. Is that somehow turning this system on in any way?
Yeah, so this is very important. And in fact, for a long time, we didn't understand how the negative regulation worked from IL-37 because we thought that IL-37 is secreted. We didn't focus on the transcriptional regulation where it affects NK cells and it affects T cells, right? Because they start to make IL-10, et cetera, blah, blah. And that whole process in a paracrine way turns down the innate immune response. And then we discovered this cell autonomous process that helped to kind of make it more parsimonious to understand what was happening. So I don't want to kind of talk too much about this, but we just submitted a paper with our collaborators in Nijmegen where we show that you need a T cell and innate combination to establish trained immunity using CD40. And I can't say more than that at the moment. It's not only our work, but yes.
So I just want to say in the mouse model, I know we don't have IL-37, but we do get trained immunity, which is very, very effective against blood stage infection, and it is T-cell regulated.
Yeah, I guess not regulated, but it's required. Yeah,
Required.
Yeah. There's a distinction between regulation and requirement. That's important. Yeah.
Yeah, really interesting talk. Following on a bit from Jean, so do you think, so these SNPs obviously are genotype, so it's sort of at a genomic level. Do you think potentially exposure to chronic inflammation like malaria or other diseases would modify it so that in people, a lot of the work we do here, Gordon's work, what I've done as well, we look at people with high exposure to malaria versus people with lower exposure, and we find differences in immunity, and we don't fully understand the mechanism for why high exposure leads to this somewhat tolerogenic state. So in addition to these SNPs, which are germline encoded, do you think that there could be a way where chronic exposure to the pathogen over time may induce more of this, regulation such that it will be maintained in individuals in a particular environment who are exposed to the, so it's sort of beyond the germline, beyond their genome, you have this trained thing that is linked to exposure to the pathogen. And if that would be different from people who are less exposed to that kind of inflammation.
Yeah, so I think it's unfortunate because I had a slide that showed that, and I don't know why it didn't build correctly. But the best way to encompass your insight, which is a very important insight, is that LPS tolerizes over time, right? And we know that there are other things that can drive a very similar tolerization. And I think the most attractive hypothesis for me is that you're actually reversing the benefits of trained immunity. So if you think of it as like, as I said to you, these adaptive responses have a permanence, right? T cell receptor, that T cell clone can be expanded on demand. B cell, same thing, antibody stimulate, you start making more antibodies. You get the clonal expansion is how those things are maintained. You can't get rid of them. These epigenetic responses, these epigenetic marks that you acquire can be erased. Okay? And when we discovered Mlilo, we showed that you could program the mark and you could erase the mark. And for example, in sepsis, you actually erase the mark. And when you erase the mark, you get this tolerized state. So think of it as like, I think Gordon and I were talking about this for a while, which is that if you have a chronic infection, you essentially impair the ability to create a very stable H3K forming 3 mark. And if you do this, then it's, at least for me, it's the most attractive hypothesis. You actually impair, over time, trainability of the innate immune system. So if you tolerize the system for long enough, it'll be very difficult to train the system. Now, LPS can do something, can be reversed by beta-glucan. So that was shown by Boris Novakovic, where we gave people LPS, he gave people LPS, and he could reverse the tolerance by giving them beta-glucan, right? And that allowed those monocytes to be trained again, reverse the tolerization. The question is it possible that long-term extended tolerization means that you are not retrainable that easily anymore? And remember, training has to occur in the bone marrow. So you could create a state in the bone marrow compartment that could be essentially untrainable or lead to very low grade inflammation constantly, right? So that's what I would say. I'm not saying that's the only way this can happen, but for me, like with the actors I've presented, this is a way that it could happen. Exactly what you're talking about.
Okay, we need to wrap up, but a lot of hands still up. Wellington and then Abdul. Quick question, then we go. We are late.
Okay, thank you, Musa. I'm sure we will continue talking about that. The hypothesis around the genotype AA and inflammatory response. That's interesting. I don't know whether you can just comment on that. Malaria, bacterial infections. I think I've heard that from you.
Did you hear?
Genotype AA.
Oh, malaria and yes, so people,
yeah.
so sepsis is the great example. If people are AA and they get like a very strong infection, their chances of dying from sepsis are augmented. That's already in the literature. That other labs have shown that.
Yes, Abdul, last one. Microphone is coming.
Thank you very much. So it seems that developed countries are more affected by allergy than poor country, right? So is there any link with trend immunity and particularly in the context of like genetic diversity with what you are seeing for like Amanzi? Yes, thank you.
Yeah, no, that's a really great question. I don't really work on, I'm not an immunologist, so I think allergy is super interesting, but I don't even know the proteins involved in allergy.
You're now one.
So I'm really like out of my depth, but I think the hygiene hypothesis is something that definitely should be more tightly linked to the trained immune status of people, and yeah, so I think that's super important. I'm really apologize. I can't be more helpful than that.
Thank you.
All right. So this was outstanding, right?
Thank you so much.
Yeah. So these are the type of talks that can generate a lot of ideas for your research because it's, basic, molecular biology with implications for many, many, many diseases. And with those who work on malaria, we've been having all these discussions. And just yesterday, we were talking about how we can do some experiments to try to, understand the mechanism of tolerance in malaria. So thank you very much, Musa. And thank you all. Over to, is it Peter or Yao? Yao, I hear Yao, today you are the MC. Peter has handed over to you, so.
I thought I was just emceeing the party. I didn't realize I was emceeing.
It's the whole day. The contract is for the whole day.
Oh, okay.
So you need a program.
Okay. Keep the time. All right. Okay. Since I've been conscripted, it's my pleasure to call up the chairs for the next session. So we have Collins and Frederica. So Dr. Collins Msita Muranga and Dr. Frederica Pate, both of whom are alumni of WACCBIP. And Peter Kwashi has asked me to remind you to please keep to time. It's Peter who has asked it, not me.
Good morning. We'll begin the next session with Dominic, who is also an alumnus of WACCBIP, and he would be talking about polymorphisms in a range of host genes among Africans and malaria susceptibility. Would plead with all, Dominic, please come. We'll plead with all our speakers to try and adhere to time so we can make some gains with time. So Dominic, you do have 30 minutes.
No. 20.
15 minutes. I beg your pardon. 15 minutes, starting from now. Yeah.
Hello. Yeah, good day. It's always great to be back at WACCBIP, where it all began. My career really started from here. So I think we've heard a lot about malaria already, so I'm not going to bore you with stuffs, but this is just to show that malaria trends are going down, and still a lot of people are at risk, with about 600,000 people dying. That was in 2022. And for those who are new to malaria, this is just a life cycle, talking about what occurs in the parasite, and these parts occur in the host. And that is what I have been interested in. And I was ushered into this because malaria is a huge challenge, which is a problem that we need to solve. So my journey began with the department over here with Dr. Jimeni and then Karin Duka during my master's. And then I got the opportunity to join WACCBIP and interacted with some of the faculties, especially those from outside. I got wonderful PhD supervisors that actually gave me the path to And then I was fortunate to be examined by Professor Lars Viet. I enjoyed my viva. It made me more committed, eventually graduated. We've met in a couple of wonderful conferences. And I would like to use this opportunity to also thank WACCBIP for the journey I've been with them for, I think, 7 years now. And I've attended almost all the research conferences, presented in all of them except the first one, because I didn't have any data on malaria. Now I have some data thanks to my postdoc supervisor, Prof. Alfred Ngwa. So to start with, a lot of work have been done trying to understand the host pathogen interaction between the malaria parasite and the host. And all these papers are just to show you how much work has been involved in doing this. And so far, we've managed to see human factors on the surface of the red blood cells that the malaria parasite enters. Also, we've also seen some immune signatures, variations in genes that code for immune signatures in different populations, and also other factors such as enzyme deficiencies. And based on this, there was a paper that tried to do a GWAS in Kenya. And what it did was to pick about a couple of cases, severe malaria cases, and they used healthy controls. And in their study, they came across a panel of genes that were significantly linked to malaria susceptibility. And of these are the normal signatures that we know of, that are linked to malaria susceptibility. And these are some of the new ones that they discovered. So for my postdocs, I decided to pick some of these genes to work on. But the challenge, the reason why this is done is because we have a lot of information on these, but we don't know their distribution within the other African populations. And also, if we are able to understand this, we can quickly use that information to modulate or give us more information on the host pathogen interaction that are occurring, and that will enable us get more vaccines, drugs, and the rest. And for that challenge, I was fortunate to be part of the PAMGen project, which sought to look at both the host and then the pathogen within malaria endemic countries in Africa. This came out of Malaria Gen, which is looking at this, it's looking at the genetics of both the host, the vector, and then the parasite, all together with a database. And it was funded by the H3 Africa project. So I focused on the human genetics aspect of it, which will be linked to the parasite and the vector. So as I said earlier on, certain genes were identified. So these are, some of them are linked to erythrocyte characteristics, and some of them are also linked to immune signatures. And then I added 2 extra known genes to serve as a control for this work. So the research questions were that, based on what we know so far, will we have different SNPs within the different malaria endemic population for the genes that I've selected? And also, will the frequencies in this allele, with the allele frequencies in these genes that I've picked, will it correlate with malaria infection outcomes? And then finally, can these SNPs or the allele frequencies be linked to parasite diversity? You saw a talk with Collins where he showed how some of the parasites are different in different geographical locations. So the goal is to look at the human signatures and then marry the two. And so going forward, the first was to determine these distributions across 8 malaria endemic countries, and then also check for the infection outcome. Now, this is made up of 8 sites. The host site is Gambia, so all the samples are collected across the different PAMGen sites. They are brought to MRC unit. It is processed in terms of extracting DNA, getting other, and then some of them is shipped to the Sangha for the parasite sequencing. And then I continue with the human genetic work that I was doing in the Gambia. And this is part of the data that I'm showing. So I extracted DNA, did Tachman assay on them for those selected SNPs, and then correlated with malaria severity or malaria positivity. So going forward, these are how the countries are and the sampling that I did across the various sites. And some of them had mild and severe malaria. Some of them had positive controls. And this is the pipeline of how this particular assay was designed and was used to genotype the SNPs that I was interested in. So typically, this is how an output will look like on the Tuckman assay, where you have the homozygous on the top panel here. We have the heterozygous that are in between, and then the wild types are at the bottom. This one is showing for ATP just a representation of what all the other SNPs have gone through for malaria itself, and mild malaria and severe malaria. And for my result, this is showing the overall result across the different sites. The ATP2B4 allele frequency was around 0.38, and that of DUP4 was around 0.1. And then this was another SNP in the glycophorin that I was interested in because it looked interesting compared to my PhD work. And the first ones are for the erythrocyte surface SNPs that I identified. And then these ones are for the immune genes that I selected. And the allele frequencies that I obtained were almost similar amongst them. But more interestingly, I used these two SNPs as controls to be able to, so that each population, I want to see what is occurring in there, because these are already known SNPs to be linked to malaria susceptibility. Now, going forward, I decided to now check what are the differences between the different malaria, I would say, regions in Africa, because it's usually, you hardly find malaria in southern part of Africa or the northern part. So I decided to compare between east and west. There was a sample site, Cameroon, but because it was just one site, I did not add it to this analysis. And so this is showing the proportions with the allele frequencies for each of the genes. The orange is for West Africa, and then the blue is for Eastern Africa. And the more interesting thing that I observed in my data was that of the Dantu, which was originally thought to be restricted to East Africa, but we identified some within the West African sub-region. Now, this was the first paper that was reporting this Dantu to be linked to malaria susceptibility was done by Leffler and others, and it was known as the DUP4, and the cartoon you see here is showing the copy number variant. And this variation is occurring in a gene cluster that we call the glycophorin EBA gene cluster region on chromosome 4, on the human chromosome 4. And so originally it was thought to be only restricted to East Africa, as you have seen over here. But the assay that I used, I was able to find some of them in Mali, Gambia, and then Ghana. And this is also shown over here about the distribution of the Dantu in West Africa. That's about 1/4 of the total that I observed. And so moving forward, I intend to pick these samples and do a targeted sequencing on the gene to be sure what we are seeing is indeed the Dantu variant. And then the last objective that I worked on was to check how the allele frequency that I've identified, how they are linked with malaria susceptibility. So the blue bars are for those that were screened and they had no malaria. So this was done by RDT. Some of them were community survey, but bulk of the samples were clinical cases. And then the orange is for mild malaria. And so I did a statistical test to see if what I'm observing is different, if there's any significant difference among them. And as you can see over here, only for the immune genes, only the CD40 ligand didn't show any significant association in terms of malaria groups. And then for the erythrocyte, the genes that are linked to the erythrocyte, again, this is the DAN2, and this is the ATP2B4, and a lot of work has been done on this, so we can now consider it as one of the most genes that have been selected for with respect to malaria susceptibility. Again, I use G6PD as is shown over here as my control, and so I can confidently conclude based on the analysis that we did and the data that we've generated that the following genes, AR14, ATP2B4, DUP4, and INPP4B, showed higher allele frequencies in the higher in the negative malaria group. What this means is that if you have these SNPs within you, your chances of either getting severe malaria, you could get an infection, but the challenge of getting a severe malaria is really low. And as has been shown with the DAN2 as well. So I know this is good data that I need to move further on, generate more data, and see which high-impact journal we could send it to. Because that is the culture of WACCBIP, no predatory journals. You look for the top guys and send it there. So in summary, we've determined the allele frequencies of certain selected SNPs that are in both the erythrocytes and immune genes. We've also shown the relationship between these polymorphisms and malaria infection outcomes. It's also the first kind of survey looking at all malaria endemic regions across Africa in one big study. We've also detected DAN2 in West Africa. Interestingly, even though Nigeria is a very big population, I did not find DAN2 in that population. Maybe the sampling site that was used was restrictive. And also, this will help understand the role that is, or the mechanisms that are going on during malaria infection, and this will lead to better vaccines, among others. Going forward, as I said earlier, we intend to validate the DAN2 within the West African population, and also correlate these SNPs with the parasite diversity in the population, and then continue to identify phenotypes that we can use to do functional assays to see how these SNPs are interacting together. At this point, I would like to acknowledge collaborators and those who have supported the study. It was funded by H3 Africa. The host institution was MRC in the Gambia. My boss is sitting at the back. He's Prof. Amamba Ungwa. And I know I've been a bit stubborn, but you know what, no excuses. You just need to do your best and get the work done. And also Dr. Lucas, Lemu, among others, WACCBIP itself, and our international collaborators, Malaria Gen, the Sanger Institute, where the parasites are being sequenced, Oxford, and also the participants who willingly volunteered to be part of this study. Again, I would like to thank Prof. Ungwa for hosting me in the Gambia, and in front of you all, I'll assure him that the papers will be ready within the shortest possible time. Thank you.
Thank you. I think we'll take 2 questions, if there are any. Okay. No, there are questions. But you go first.
Yeah. Dominic, you were talking about association with severity, but you also said that you found a difference with the group that had no parasites. This is not longitudinal, right? This is cross-sectional. So how would you make any conclusions about people's susceptibility just based on the fact that at that snapshot in time, they didn't have parasites? Because it's, you know, as for malaria, you can have parasites today and none tomorrow, and so on and so forth. If you live in an endemic area, it's a very dynamic process in terms of getting infected and cleared or not, you know, over a period of time. So unless you do a longitudinal study, how would you try to associate this with protection?
That's a very important question. And I agree with you, but then also it falls within what you've already said about tolerance. Sometimes you can have an infection, parasites will be within you, but you will not develop full malaria. Now, most of the SNPs that we've worked on is more or less not really protective, but more or less reducing your chances of getting a severe form of the disease. And then secondly, there are a lot of factors that influence malaria susceptibility, not just even the host factors, the environment and all of that. And then thirdly, the survey wasn't a one-time point collection, was collected over a period of time. And so, and I agree with you that it could be a little bit fishy, but the point is if you are collecting samples over a period of a year across different sites, a lot of things will cancel out each other. It's based on those assumptions that we are making these conclusions. Thank you.
Okay, I think, okay, Kashi has the microphone, then I'll come to DJ.
Okay, just really quick. I know you said you're going to verify the Dantu, but is it, were they collected from the northern part of Ghana, or is all over Ghana?
Okay, so the PAMGEN project has over 10,000 samples collected across different places. And so what I tried to do was to make sure different ethnicities are involved in the samples that I use for this work. So I can tell you confidently it wasn't only from northern Ghana, but I tried to make sure almost all ethnic, to the best of my ability, were represented.
I was asking that more because of the proximity to Mali and the Gambia. So if those samples that had the Dantu variant are sort of segregated in a certain location.
It's a good question, but I quite remember when I was doing my PhD in Oxford, I screened for this Dantu. I saw two, but I was leaving, so I didn't really follow it up. That was not the focus of the PhD. So now doing a postdoc and having the opportunity to screen, I thought myself, no, why don't I put it in and see what comes out of it? And so what we are seeing, I don't think is fixed in the northern part or Mali per se, because the Cameroonian samples also are positive. But I didn't show it because it's only from one country and from one site. And so we need to do more screening in Central Africa to be able to confirm what I've observed over there also. Yeah.
Okay, so we'll go to DJ for the last question.
Yeah, really more of a comment for you and for the crowd, right? We've had this conversation before. And so in a cross-sectional study, granted in more individuals, it's better in different countries. But to reinforce what Professor Awandare said, okay? We have to think about definitions, we have to think about severity, and when we go in and parachute and get a sample, it's not a criticism of you or anyone, but we measure what we have. We will not be able to show susceptibility to severe disease with a cross-sectional study, period, full stop. The other challenge is when there is the definition of malaria with the RDT. Again, not a criticism, a reality that I think you, students, everyone in the crowd needs to understand. How long does the RDT remain positive after one has malaria? Roughly. Anyone want to take a guess? Upwards sometimes of 30 days. And it's not a criticism. You didn't design the RDTs, but my point is, if I look at a child and say she or he has malaria, and I use an RDT, it is perhaps a historic marker, but probabilistic mathematics will tell you, unless you have a concomitant parasitemia, that child had malaria, doesn't have malaria. So then when you go into the cross-sectional nature, what's the difference between me having malaria today versus tomorrow in an endemic region? It's whether or not I had the exposure and got it. It's not necessarily based on genetics. So until one looks longitudinally across time in different levels of endemicity, it's challenging to say that this protects, this creates susceptibility. I just want to make that comment. Thank you, sir.
Thank you for the comment, and just to react a bit to it, the study, as I said, we just wanted to first of all, see the distribution. It wasn't necessarily to link it to malaria susceptibility because that has already been done in Kenya. In fact, that is what informed the choice of picking those SNPs. And also, there is no way you may have malaria infection today and tomorrow it will be cleared. And at that point, if you are testing for RDT and antibodies are not in the system, you may not test positive for RDT. It doesn't necessarily mean you are protected, but the work that we did was just to see what is going on in the population, to have a general idea of these SNPs across the various places that we surveyed. And then going forward, I've tried to discuss with my supervisor where we could pick a particular location where these SNPs are located, and then follow the same participant over a period of 1 year during malaria season, the high transmission season, and then the low transmission season, and see if still these people are not getting malaria, although they are malaria-infected people in the population. And that will give us a clearer picture on what we are observing. Thank you.
Thank you, Dominic. Our second speaker is Christine Echamo, who will be talking about synergistic effects of polyphenols on iron modulation in leishmania.
Hello, good morning. My name is Christine Echamo, and I'm presenting part of my PhD thesis titled Synergistic Effect of Phenolic Compounds and Glutathione on Iron Modulation and Immune Responses to Leishmania donovani. I'm being supervised by Prof. Theresa Manfujira, Dr. Calvin Tenguin, and Dr. Love Yamdi. Okay, so leishmaniasis is a parasitic disease that is caused by leishmanian parasites. The disease is ranked second to malaria in terms of the proportion of the population that are afflicted and the number of deaths associated with it. Globally, approximately 350 million individuals are at risk of infection, with 70,000 to 1 million new cases occurring annually. The death toll also ranges between 20,000 to 30,000 deaths. It is common in regions where the sunflower vector is mostly predominant, such as the Southern Europe, the tropics, and then the subtropics. It's also endemic in West Africa, with some reports in Ghana. Currently, there are challenges in obtaining an effective treatment to combat the disease. Clinical manifestation of the disease is caused by the intracellular hemazygote form of the parasite, which resides in the host macrophage. So when the parasite infects local phagocytic cells that are resident in the skin, it results in cutaneous leishmaniasis, which is characterized by lesions on the exposed part of the skin. Lesions that occur in the mucous membrane causes mucocutaneous leishmaniasis, which is disfiguring if left untreated. The last presentation of the disease is the visceral leishmaniasis, and this form of the disease is the most fatal form, associated with weight loss and affects the visceral organs. So based on the fatality and the disfiguring nature of the disease, there's the need for effective treatment to combat it. In the search for effective treatment for leishmaniasis, a study done in my lab has been able to show that phenolic compounds, which are natural products of plants, are able to elicit an elaborate inhibitory effect on Leishmania donovani. And the mechanism of action elucidated was via ion chelation. So from the study, for promastigotes in the culture media, the presence of the phenolic compound represented in green induces an ion-deficient environment within the culture media by chelating the ion. This results in a differential expression of ion acquisition genes like Leishmanian ion transporter 1 and Leishmania heme transporter. However, the mechanism of action by which these phenolic compounds modulate host ion content has not yet been fully elucidated. Okay. IT. Please, can you help me? Okay. Okay. Okay, thank you. So naturally, sorry for that. Naturally, in a host macrophage, the presence of iron is gained by the catabolic activity of heme oxygenase 1 on senescent erythrocytes. Heme oxygenase 1 is an enzyme that converts toxic-free heme to less toxic forms of bilirubin, carbon monoxide, and iron. The gain of this iron is either used up by the cell or stored up by being bound to other proteins to form ferritin, or is exported out via ferroportin, which is currently the only known mammalian iron exporter. So when this happens, the unavailability of iron in the host macrophage causes invading microorganisms not to get access to the iron, and resulting in their probable death. However, for Leishmanian parasites, they are able to survive and differentiate. What the parasite does is that it induces the host cell to produce some inflammatory cytokines, such as these, which trigger the secretion of hepcidin. And here, hepcidin is represented by the green crescent. So when hepcidin is triggered, it associates with ferroportin here and causes the internalization and degradation of ferroportin. When this occurs, it results in an increase in liable iron within the host macrophage and an increase in the iron stored up as ferritin, resulting in high proliferation of the parasites. Thus, the study sought to identify the effect of these phenolic compounds. How do they affect the expression levels of these iron-related proteins like heme oxygenase 1, ferritin, ferroportin, RAMP1, and hepcidin. In addition, recent advancements made in drug discovery also employ combination therapy to be able to combat other infectious diseases like malaria and tuberculosis. The rationale being that there's less chance of the parasite to develop resistance simultaneously to the two drugs that are being used in the combination. So this strategy was adopted to be able to identify treatment regimen that will elicit minimal toxicity, parasite clearance, shorter duration in treatment, and higher potency. So here are some compounds, for phenolic compounds and glutathione, that were selected and screened against the intracellular imastigote forms of the parasite. So preliminary data has shown that these compounds, rosmarinic acid, curcumin, apigenin, and hesperidin, have iron chelating properties because they possess these catechol and ketoenol moieties. And they've also been shown to be effective against the promastigote forms of the parasite. And this is glutathione, which is the first-line anti-Leishmania drug that is being used to treat all clinical presentations of the disease. So these compounds were selected and screened against the intracellular imastigote forms. So with this, the study sought to answer these two questions. How would these phenolic compounds modulate host iron content in the presence of Leishmania infection? Does combination of these compounds with or without the anti-Leishmania drug will it elicit any synergistic effect against imastigote forms of the parasite? So the study began by screening these compounds against the promastigote forms of the parasite and via the Alama blue cell viability assay. IC50 values obtained for the phenolic compounds range between 12 micromolar to 240 micromolar. That for amphotericin B, which was used as a positive control here, was around 0.28 micromolar, and glutathione was 11.55. Next, we sought to identify see the effect of these compounds on the host cell, and we used it to estimate the selectivity index, which is a measure of the ratio of the IC50 of the host cell to the IC50 of the parasite, of the intracellular imastigote. And here, what was observed was that these three compounds, hesperidin, curcumin, and rosmarinic acid, had IC50 values greater than 10, indicating that these compounds are selective for the parasites other than the host cell. And then for amphotericin B, an IC50 less than 1, indicating its toxicity. So now, having determined the IC50 values of the individual compounds, we used a modified fixed ratio isobologram method to be able to identify the combination therapy and see whether there's any interaction, synergistic interaction that exists among the treatments. For combinations between the phenolic compounds and glutathione, this was what was obtained. There was a synergistic interaction. The curcumin and glutathione combination elicited increased potency over the mono treatment against Leishmania donovani. And here, it was observed that the blue curve, dose response curve, represent that for curcumin alone, and then the red one represent that for glutathione alone, and the remaining colors represent that for the mixtures. It was observed that there was a shift to the left for combination 2, 3, and 4, indicating that these compounds at lower doses are able to elicit an enhanced effect against the parasite. The interaction type was synergistic for combination 2, 3, and 4 as they fell below the line of additivity, and the overall mean FIC was estimated at 0.67, indicating that they are synergistic. Similarly, for the phenolic compounds, curcumin and rosmarinic acid had an increase or had some form of synergistic interaction. And for here, it was observed that it was all the combinations, both 2, 3, 4, and 5, had a shift to the left, and then the interaction type was synergistic for the 4 combination. Overall, FIC calculated was 0.49. So for all downstream interaction, I sought to work with combination 4 for all of them. That is the 2:1 ratio. Combination 4 is a 2:1 ratio, indicating that it's 1/4 the IC50 of curcumin to have the IC50 of rosmarinic acid. Similarly for the curcumin and glutathione combination too. The cytotoxic effect of the combination also showed that curcumin and rosmarinic acid showed a superior selectivity against curcumin and rosmarinic acid, as well as curcumin and glutathione. They showed a superior selectivity against the intramacrophagic mastigotes relative to the mono treatment. So next, I proceeded to measure the cytokine levels, aiming at verifying whether the treatment elicits any inflammatory immunomodulatory response. And here, I used a bead assay that consisted of 5 cytokines and a chemokine. And my focus was on interleukin 6, interleukin 10, MCP1, interferon gamma, TNF-alpha, and interleukin 10. And I used it together. I measured the protein levels in the supernatant for my treatment. So it was observed that for LPS, there was an elevation in the inflammatory cytokines as well as the chemokine. And here, LPS, that's lipopolysaccharide, was used as a positive control as it forms the major component of Leishmania surface membrane. So when I infected macrophages with Leishmania, a similar pattern was observed, similar to what was observed for the LPS. However, the treatment induced a different response. It was observed that for curcumin, there was an enhanced elevation of interleukin 6, MCP1, and then TNF-alpha. For rosmarinic acid, MCP1, TNF-alpha, and the combination of them showed a more sustained and heightened production of these 3 cytokines. For glutathione, I had MCP1 and then TNF-alpha. The combination for here, I didn't see any interleukin 6, and this could be attributed to the synergistic interaction that may exist among them. Okay. So overall, it can be said that Okay, so overall, it can be said that there's an increase in TNF alpha, MCP1 for my treatment, and then a decrease in interferon gamma, and interleukin 10, which enhance enhance growth of Leishmanian parasites. So, knowing that these pro-inflammatory cytokines, they create an environment conducive for macrophages to be able to trigger oxidative burst, I sought to investigate whether they are putative inducers of ROS and NOS. And here it was observed that truly there was an increase in reactive oxygen species and nitric oxide species relative to the infected, which is white. Yes, there was an increase for my treatment relative to the infected macrophages. Next, I wanted to find out, are they really inducers? Do they chelate iron? Are they iron chelators? And truly it was observed that I quantified the intracellular iron content using a ferroxene-based assay. Now I observed that there was a decrease in iron content. That means, and the implication was that the presence of the parasite increased the iron content in a host cell, while the presence of the treatment lowered the iron content in the infected macrophage. Lastly, the expression levels of the iron-related proteins was also investigated. And here, RAMP1, which is a divalent metal transporter of iron, was measured using RT-qPCR, and it was observed that there was an upregulation of this gene, indicating that it implies that these treatments enhance the activity of RAMP1, which results in an efflux of more iron from the phagolysosome into the cytosol and causing the death of the parasite. For ferritin, there was a downregulation for the phenolic compounds and its synergistic interaction, and an upregulation for glutathione and its synergistic interaction. The reason being that the death of the parasite truly occurred. However, the presence of the phenolic compounds elicited this iron chelating property, reducing the ferritin level. And since glutathione does not have any iron chelating property, the available iron was converted to ferritin right away. Okay, so in summary, we can say that treatment with the phenolic compounds caused a decrease in the allowable iron with a downregulation in ferritin expression, and then an increase in RAMP1, induces the pro-inflammatory cytokines to be able to create a conducive environment for rosmarinic for reactive oxygen species generation, and its addition of glutathione enhances this effect. So I'd like to acknowledge my supervisors, Prof. Teresa Manfujira, Dr. Calvin Tengwin, Dr. Yamde, and then my funders. Thank you.
We'll take one question if there are any.
Thank you.
Yes.
A very interesting talk. I actually didn't mention one thing really important, is that Leishmania, this cutaneous stage of the disease, turns on IL-37 to suppress pro-inflammatory responses, and it actually turns on that non-coding RNA amanzi that I mentioned. Yeah, so maybe one thing we could try in terms of suppression of this early stage is to suppress the activation of amanzi, which we've seen happens even with Leishmania lysates and monocytes, which seems to be a pathogenic strategy. Just a suggestion.
Okay, thank you.
Thank you very much, Christine. Let's give her a round of applause. Our next presenter is Tosin, who is a PhD student at WACCBIP, and she's going to talk about the stress phenotypic kinetics that promote antimicrobial resistance, tolerance, and virulence in critical priority pathogens.
Good morning, everyone. My name is Tosin Shembadejo, and I'm a PhD fellow here at WACCBIP. I would like to share with you a part of my research work titled Stress Phenotypic Kinetics Promotes Antimicrobial Tolerance and Virulence in Critical Priority Pathogen. Antimicrobial resistance remains a global health crisis, and infections by bacteria are actually responsible for over 700,000 deaths per year. This has been estimated to skyrocket to like 10 million in 2050 if proper measures are not taken. And this necessitated the need for the World Health Organization to categorize these organisms based on their order of importance. The ones that belong to the critical priority are those that are able to cause severe and recurrent infections and also resist last resort antibiotics. And bacteria that belong to this category include the carbapenem-resistant Enterobacteriaceae, such as E. coli, Morganella. Okay, so today the focus will be on Escherichia coli. E. coli are gram-negative mesophilic bacteria that have optimum growth conditions around of about 37 degrees C for temperature and pH of 6.8. They have been implicated in about 80 to 95% urinary tract infections, 27% sepsis, diarrhea, amongst others. You may wonder how they are able to cause these severe infections. This has actually been attributed to the various virulence factors that they encode in their genome. This can either be encoded on chromosome or on plasmid. So these virulence factors are like tools that the bacteria use in gaining entrance into the host cell and then establish an infection. So alongside other virulence factors, including biofilm, these factors are usually expressed upon exposure of these bacteria to various stress conditions. This is because stress triggers regulatory systems that are involved in survival processes of the bacteria. They're able to use this exposure to stress to adapt and become virulent and then resistant to antibiotics. So therefore, we understand that bacteria encounter different environmental stresses in the environment, and over time they have developed some adaptive mechanisms for them to prevent the effect of the stress in order to lower the DNA damage and cell death of the bacteria. Therefore, they usually trigger responses like the general stress response or specific stress responses to each of these stressors that would now result in changes in gene expression, either over upregulation or downregulation of some of these genes, and therefore mediate the hallmarks of virulence, which include antibiotic resistance, that is the ability of the bacteria to become less susceptible to antibiotics, and then biofilm. You can see biofilm as bacteria trying to come together as a community to shield themselves against different kind of stressors. Then persistence. These are bacterial cells that are dormant in an environment, and when without the effect of the antibiotics, they can now continue to proliferate. So understanding of all this would allow us gain insights on the bacterial potentials in host infections. Therefore, the study aimed to investigate the effect of phenotypic stress conditions on antibiotic tolerance and virulence in E. coli strains, and we achieved this by determining first the growth kinetics of the bacterial strains under various stress conditions. We went further to identify how this exposure to stress would mediate their antibiotic tolerance and biofilm. So I used archived E. coli strains, one from environmental sample and 3 from clinical samples. And as I said earlier, to start with, I identified the different antimicrobial resistance level, that is trying to see their susceptibility to various antibiotics that are used in treating E. coli infections. Then I did this using disk diffusion test and microbroth dilution. I went further to use crystal violet staining assay to determine their biofilm levels. So after doing this experiment, I noticed that the E. coli strains are actually fast growers because their generation time is between 17 to 20 minutes, and they also log phase at around 3 hours. This means that their infectivity rate is actually high, and they can contribute to severity of infection. So these strains are also resistant to more than 3 classes of antibiotics. And I went further to see this, that was at the, for the standard concentration of the antibiotics. So I wanted to see if you increase the antibiotic concentration, what would be the minimum inhibitory concentration for the antibiotics. And I also noticed that all the strains actually had minimum inhibitory concentrations that were above the cutoffs, almost 10 to 20-fold above the cutoffs. These strains are also within the range of strong and moderate biofilm formers. So the strains are actually multi-drug resistant and virulent. This is conclusion from these results. So then the next question was, will the strains be able to survive extreme stress conditions? So for this, I used the tempo stress assay, which included exposing the strains to different extreme conditions, like the temperature, pH, oxidative, and osmotic, and the conditions that are optimum for their growth were used as control. So for the temperature, the red is the control for the 4 strains, while the blue are the strains at 45 degrees C, the green at 4 degrees C. If we look at this graph, we notice that the strains actually had delay in growth. They were at lag phase up until around 8 hours, which means that they were trying to adapt to the change in temperature before about 2 or 3 of them peaked, where we started seeing some increase in the survival of the strains. So we went further to see what their growth will look like under pH conditions. And for this as well, majority of the strains, their bacterial activity were actually altered under the acidic condition, while for the alkaline condition, the 3 clinical strains were able to show increase in survival rate. And this could be because these are strains that usually reside in the urinary tract, and the urinary pH is around 4.5 to 8. I went further to see what extreme osmotic and oxidative conditions would do to the strain. And for the oxidative assay, high concentration of hydrogen peroxide actually inhibited the growth of the bacteria, while for the osmotic, they had a longer lag phase as well. However, the environmental strain and one of the clinical strain peaked after 8 hours as well. And this could actually be because of, for the environmental strain, maybe exposure to different kind of osmolarity in the environment. So I went further to see how the stress-recovered variants would tolerate increased antibiotic concentration. And with this, we increased the antibiotic concentrations to the double of the minimum inhibitory concentrations. And we observed that in comparison to the unstressed, this is the one that did not go through the tempo assay, the stress-recovered variants actually performed a bit better in terms of their growth rates and survival. And this observation was actually similar to almost all the antibiotics and all the strains. Then the next thing was to actually see if the various stress exposure would influence their biofilm levels, because initially we checked the biofilm levels, and the observation here is that the strains actually maintained their biofilm level. The ones that were moderate still maintained being moderate. However, few of them actually reduced from moderate biofilm format to weak biofilm format. And then we also, there was also one of the strains that increased from being a moderate biofilm format to strong biofilm format. We went further to then try to characterize the genome of the strains in order to identify all these virulence-conferring genomic factors in their genome. For this, I extracted DNA and we did whole genome sequencing, and then used bioinformatic tools to check the resistome, virulome, and plasmidome. So this is what the resistome of these strains look like. They have several resistance genes that could have conferred resistance to different classes of antibiotics, including tetracycline, fluoroquinolones, aminoglycosides, and others. They also had efflux pump genes, which is also one of the mechanisms of resistance to antibiotics. So the next thing was to identify if there are mutations in the chromosomal encoded antibiotic resistance genes that could have also contributed to some of these phenotypic traits that we found in these strains. And these strains actually add some substitution, amino acid substitution in some genes that are targets of antibiotics. For instance, topoisomerases, which are these gyrase A, gyrase B, para C, and para E. They add several amino acid substitutions that could have influenced the increased resistance to these antibiotics. This has also been shown that simultaneous presence of amino acid substitution in these enzymes, in these genes that code for the enzymes, mediates high fluoroquinolone resistance. So for the common substitution that have been observed, those are the ones I highlighted in red. We also found some other novel, that we believe are novel substitution that could contribute to resistance in these strains. We are still trying to work on this substitution to see if they affect the structure and function of the proteins. So the next thing was to identify if in the genome of these strains there are some genes that could contribute to biofilm formation and also bacterial survival in their host. And the genes actually had over 60 virulence genes, and some of these genes are genes that belong to various categories of virulence, which include adherence, colonization in host, ion acquisition systems that help them to siphon for ion, effector proteins, toxins, and also capsule-associated genes. So the next thing was to see how some of these genes are being shared among the strains, because we noticed that both clinical and environmental strains actually have comparable resistance and virulence genes. So we checked if they encode some plasmids, and majority of the strains, or all the strains, actually have more than 3 plasmids that encode some of these resistance and virulence genes, which we believe could contribute to the tolerance and biofilm formation of the bacteria. In summary, we identified that extreme phenotypic stress reduced bacterial cell growth and induced phenotypic changes on the strains, and the strains are both common and novel mutations that could contribute to resistance and virulence, and that the resistance and virulence that we observed might possibly be propagated via plasmid and chromosomal genes, and also the genome of both the clinical and environmental strains encode comparable resistance and virulence genes. And the implication of this is that bacteria can leverage this phenotypic stress to tolerate antibiotics, and antibiotic tolerance strains can prolong treatment and also contribute to severity of infections. Some of them can also result in relapse of infection, and changes in genome due to mutations can lead to ineffectiveness of antibiotics. I'd like to acknowledge my supervisors, the antimicrobial resistance research lab members, molecular biology research lab, faculty members, WACCBIP for the funding and platform. Thank you all for listening.
Thank you very much, Tosin, for that great presentation. So we will take 2 questions. Yes.
Fluoroquinolones, and she had septicemia. Based on this study, what would you advise me as a medical doctor?
Okay, for instance, some of these antibiotics, because we have noticed that over time, we become resistant, we show resistance to them. Like when people use these antibiotics, they don't feel better. So there is this combination therapy where they combine, for instance, aminoglycosides and fluoroquinolones in order to treat some of these infections. So that way, if the person is if the person is resistant to aminoglycosides and you combine it with fluoroquinolones, maybe the effects, or like at the point where they become resistant to aminoglycosides, because they have different mechanisms of actions. I mean, all these antibiotics, they have different mechanisms of action. So if, okay, how do I put this? If, yeah, I'm trying, because so that means the person is also resistant to third generation.
Okay, maybe we'll discuss later.
Because
Okay. All right. We'll take the next question from. Yes.
Hi. Hello, Tosin. Thank you so much. I have a quick observation, and your extreme oxidative stress is really extreme because 30% of hydrogen peroxide for 24 hours will completely or almost ruin any mammalian culture system or model that you are using. And so by the time that you are interpreting that 30% of hydrogen peroxide after 24 hours is inhibiting bacterial growth, I'm not sure whether we have lost the model itself, or it's just the bacteria that is not growing. Did I miss that?
So these are bacterial strains. So the reason I chose the extreme conditions was just to see their ability to withstand some of these conditions. Actually, I later went on to use lower concentration of the hydrogen peroxide, and I did like a flash oxidative stress to ensure that I'm able to recover some strains for my downstream analysis. Yes, please.
Yeah, thank you. Because of time, you can meet Tosin after the presentation. Thank you very much, Tosin, for that great presentation. We'll take our next presentation from Isaiah Debra, who just defended his PhD. So he's going to talk about insecticide resistance profiles of Anopheles gambiae in the coastal epidemiological zones in Ghana.
Right, thank you very much. We know that we cannot say we are defeating malaria when we leave the mosquito out of the equation. So we believe that once we kill the mosquito or we bring the population to the lowest level, there will be no malaria. But the question is it easy to kill the mosquito? No. So we'll be looking at the insecticide resistant profile of malaria vectors in the various zones in Ghana. But specifically, we'll be looking at the eco-epidemiological zone. Malaria is still a problem in Africa, and is a major challenge, and is still leading the world with 95% of global death. And Ghana population is at risk of malaria. And in the southern, and then in the southern, that's the forest, and then the coastal zones, it peaks twice in the year. And then in the northern zone, it peaks once in a single wet season. That's from June to October. And vector control is the major component and the key preventive strategy for malaria. And the main vector control tools are the long-lasting insecticide-treated net, and then the indoor residual spraying. They are the major vector control tools. But the challenge now is that these mosquitoes have developed resistance to the 4 main classes of the public health insecticides. And the resistance to pyrethroids. Pyrethroids are the main compounds that are being used to impregnate our bed net. Is considered as a major threat because the major functions of the bed net is serve as
repellency property. It has repellency properties such that it repels the mosquitoes when they come in near the net. And then it also serves as a physical barrier between the person sleeping inside and then the mosquito. And then it also has some killing effect. And the killing effect is what we normally look at, because once they die, then we reduce the population. Then we'll not be talking about vaccines, drug with side effects, and what have you. So the major mechanisms that these mosquitoes use to resist, to develop resistance to our insecticide is the target site mutations, where the target site of the insecticide is mutated to something else. Then we also have they thicken their cuticle. And then metabolic resistance, overexpression of metabolic gene like the cytochrome P450s, the monogenesis, the GSTs, and other proteins leads to detoxification of the compounds. And then also studies have shown sequestration of some compounds. And then the microbiome, it was found out that mosquitoes that are resistant to pyrethroids have certain microbes compared to the susceptible. And then also the last one, which is the oxidative stress. So rationale. In Ghana, there is a growing report of insecticide resistance to public health insecticides. And high level of resistance compelled the National Malaria Elimination Program to switch to piperonal pyrethroid dual impregnated bed net. This PBO is a synergy such that it inhibits the overexpression of cytochrome P450 so that it leads to the killing effect of the bed net. And then we have other newer insecticides for indoor residual spraying, Artelic, that's the Pyrimidines, and then the Fedora Fusion. The main aim was to investigate the phenotypic resistance within the vector population, and then the underlying mechanisms of resistance. So first, we determine the intensity of resistance in the vector population, and then we look at the mechanisms of resistance in the malaria vectors in the various zones in Ghana. So we sample from the various habitats in Accra, in the various areas that mosquito breeds. We got the larvae, and then upon getting the larvae, we move on to raise them to F0s, and then we went further to perform WHO insecticide susceptibility test. We also went further to do synergies bioassays, where we pre-expose the mosquitoes that are resistant to the PBU before we introduce them to the insecticide. And then we went ahead to look at to do intensity assay, that's to look at the strength of resistance in the population. And then finally, we did whole genome sequencing to unveil molecular markers of resistance. Then for our studies in Accra, the sites that we sampled, all the mosquitoes are resistant to the pyrethroid compounds. And what we mean by resistance is that according to the WHO criteria for insecticide resistance, when the mortality rate is below 90%, is considered as resistance. And when it is from 98 to 100, then we say the population is susceptible. Now, looking at all these compounds, that the deltametrin, permethrin, inopebia, all of them are resistant, below 10%. Then when we move on, and then we pre-expose them to the PBU, the synergist, we still have high resistance in the population. And the story is the same for Teshi and then Damfa areas. High resistance was observed in both the pyrethroids only and then the PBU plus the pyrethroids. So we went further to look at the strength of resistance in the population by doing by the intensity assays, and the result is the same at times 5 and then times 10. You see high resistance is observed in the population. And the same across all Teshi and then Tema. So we went ahead to
look at some of these mutations that confer them to resistance. And then for Anopheles gambiae, the KDROS is fixed in all the regions in Ghana. But if you look at this same mutation in combination with other mutations, is high. We have observed high frequencies in other regions. And then several other secondary mutations have been observed in the region. Now, for the Anopheles coluzi, one of the major vectors, KDROS appears to be relatively stable in high frequency in southern region. But when you look at in the northern region, that's in the upper east, we have seen declining, but it's replaced by this V402L and then 15270 mutations, combinations. It's being replaced. As it is declining, this mutation combination is taking over. And then other secondary mutations in combination with the KDROS is in high frequency in southern part of the country. Now, for our metabolic resistance, on the chromosome 2 arms, we have found signals of recent selection in both gambiae and then the coluzi. The CYP6P3 gene locus is confers signal for the metabolic resistance, and then the KEAP1 gene also the second signal also confer the same resistance of the same in the same locus of the same gene, suggesting that this is a new gene that is identified in our study to confer resistance to pyrethroid. So in summary, high resistance to pyrethroid was observed in the population. So the take-home message here is that the mosquitoes are not dying, and if they are not dying, they must die so that we are free of malaria. And also, the PBU couldn't restore the susceptibility to the pyrethroids. And you know, PBU bed nets were distributed about 2 months ago across the country by the National Elimination Program. But that is not to say go and throw your bed nets away, or go to your communities and tell them the bed nets are not effective, so they shouldn't use them. The bed nets serve as a physical barrier between the host inside and then the vector, so it's still effective. Let's use them. Then other mechanisms of resistance, apart from the cytochrome P450, like the KEAP1 gene and then the CYP6P3 that we saw above, they might be the major ones playing a role in resistance. So we have identified a novel KEAP1 gene which conferred metabolic resistance to insecticide, first identified in gambiae and coluzi. I am so fast, I don't know. So going forward, we would like to conduct functional analysis of the KEAP1 and then CYP6P3 gene, and then uncover novel metabolic resistant genes and their potential mechanism using RNA-seq, and then also conduct genome-wide association analysis to uncover new markers of insecticide resistance from the whole genome sequencing data of the samples that we phenotype. So these are my references, and then I would like to acknowledge Dr. Lucas, Dr. Niwe, Prof. Gordon for the significant role they play leading to the birth of medical entomology in WACCBIP, and then the medical entomology team, malaria lab, and then our funders, Melinda and Gates Foundation, the FDC, NIHR, World Bank, Wellcome Trust, Ghana Health Service. Thank you very much.
Thank you very much, Isaiah. We have we can take a few questions. Yes, sir.
So, I mean, that's quite interesting. Before you do your functional analysis of KIP1 gene, KIP1 actually binds NRF2 in stress response. And so it's not surprising that by subjecting the pathogen to these, it would make a mutation in KIP1 and sort of try to escape the stress. Yeah, so what I'm saying is that KIP1 is a gene that is involved in stress response. So I'm not surprised that it is sort of involved in this. It's also quite interesting that the mosquito, or would choose that pathway. And so, what do you think could be the reason for choosing to respond to the stress in terms of making mutation to something that helps you to adapt, rather than completely, say, escaping the compound, in its entirety, because you are choosing a tolerance pathway rather than using a pathway that allows the compound not to come in the first place. So there are a lot of efflux activators that prevent these insects from getting into the parasite in the first place. And that's how most of these resistance have been observed. So why do you think it would probably be more advantageous to go the way of tolerating the insecticides rather than trying to get them out of the system in the first place?
The KIP1 gene have been first reported in these house flies, and then in literature it works to kind of repressor mechanism. It kind of represses the cytogroup P450 gene, like the CIP. If you watch the presentation, the CIP, CISP3 locus, they all appear almost on the same distance. So it works as a repressor. So we will go further to study this gene more and know their role, the role they are playing in this pyrethroid resistance. Other studies have reported this gene, its role in pyrethroid resistance and other insecticides. But their major role, the mechanisms, has not been fully elucidated. Going forward, we'll dig more and get more information so that we can confirm or disprove what is in literature. Thank you.
Thank you. Mauli?
Mosquito nets are not being used for the purpose for which they were intended. Do you think this contributes to the development of resistance, and how would you recommend or suggest, or what steps should be taken so that people would use them properly?
Thank you very much. I agree with you. The purpose of distributing the net is to use it, but in most rural communities, they use it for fencing gardens, and some use it to catch small fish in the lakes and what have you. Some use it as fence and all that. That's very bad. But campaigns are ongoing to ensure, and as we are here, we are from different parts of the country, we can take the message home. That's why I was quick to add that don't go and tell them that they are resistant, so we should not use it. It serves as a physical barrier, such that when the host, that the human is sleeping inside or the child is inside, and the Anopheles comes and make noise around it, you are still inside and you are okay. So it's a matter of campaign. Okay, we campaign more that let's use the net, because once we use it, it will reduce it. Bed net and then the IRS, the two major vector control tools, have contributed significantly to reduction in malaria episodes since year 2000, and they are still effective. So all that we need to do is to make sure in our communities they use the bed net. The purpose of giving out the net. Let's use it.
Okay, we'll end our session with a very last question from the back.
Thank you very much. I'd wanted to know if you try to relate the resistant profiles you had for the mosquitoes to the occurrence of malaria in the specific community, so that if not for anything, you could have specified approaches to solving that problem in those communities. And also, I admire the fact that you want to kill the mosquitoes. So I want to know how you selectively kill the mosquitoes so that you don't end up causing more ecological disasters with whatever approach you want to use to kill the mosquitoes.
Oh, okay.
Yes. Thank you.
To look at the impact of resistance on malaria transmission, really studies have been done elsewhere, and they've reported conflicting results. But going forward, studies are ongoing, and we want to see whether resistance mosquitoes really they can transmit the parasite more than the susceptible ones. And then also link it, like you said, to the community, and then see the malaria prevalence and all that. So thank you. Studies are ongoing. And then the other one, with killing of mosquitoes. I don't think if we kill all the mosquitoes, there will be any ecological balance. Sorry if you're a conservationist, don't worry. But mosquitoes are doing more harm than good. And as you may know, mosquitoes are the deadliest animal on earth. It's not a lie. Yes. A mosquito is the deadliest animal on earth. So once they kill millions every year. Once we kill all mosquitoes, there will not be any problem.
Thank you. Thank
you very much.
On that note, we'll hand over to the MCs now. Thank you to the presenters, and thank you too for sitting in.
Okay, so we're going to have a couple messages from sponsors. First, Thermo Fisher will give us a presentation, and then I hear Karamo will come afterwards. And then we'll have a short 15-minute break. We're a little bit behind schedule, so we'll compress the break a bit and then try to get going. So please, Thermo Fisher.
Good afternoon. I'll make it short and as concise as I possibly can. Quite a controversial follow-up based on mosquitoes being the most atrocious animals in the world. My name is Lindy Tabete. I am responsible for the biosciences business for Thermo Fisher Scientific, and I'm really here to highlight some of the solutions we have to support the work you're doing. Just to give you a brief overview in terms of what my colleague provided on Wednesday, which is more of the downstream application, I'm primarily responsible for what I consider the most important part of your research, which is the upstream application, right? How you isolate your DNA, RNA, as well as your protein could significantly impact your results on the downstream end of things. The most important thing that we really want you to recognize is at the center of everything that we do as a company, with LabMart being an extension of what we're doing here in Ghana, is our customers. Without you and the incredible research you're doing, it really has been a pleasure for me to be here for the past 5 days, to see the high level of collaboration, seeing the young individuals that are truly determining solutions in the context of infectious diseases in Africa, and I really want to applaud the amazing work I have seen thus far. And then really in terms of some of the things that we wanted to highlight is that we are here to support you, whether you're research and discovery, all the way to clinical trials, and we've seen a high level of impact in the context of what Ghana is doing, but also in Africa as well, specifically when it comes to vaccines for malaria and the continuous work that everyone is doing here, going all the way down to commercializing as well as production. I will be here for the majority of the day, so please reach out to me, and my colleague Pierre is also here really focusing on phlegm and flow cytometry. Once again, just really wanted to highlight all the different segments of products we provide at Thermo through LabMart. And then most importantly, one of the things I wanted to bring full circle, right? I'm not going to sit here and give you a sales speech. It's really around what can we do to support you when it comes to the research you're already doing. Anything from basic research to diagnostics development, and most importantly, prevention measures, we can support you there. I noticed and heard a lot of work around mostly the I cannot never pronounce that. Thank you. And dengue, as well as Zika. So the purpose of this slide is really to highlight, if you already have specific Thermo Fisher footprint, it's very, very easy for you to work on a variety of different applications. So with this said, you'll be able to have your sample collection, and then you'll be able to isolate your RNA, doing your qPCR, and then doing the reporting. Same thing in terms of some of the other solutions we have for wastewater surveillance. Same instrumentation, different application. We're here to really support and assist you there. And then lastly, I've heard a high level of information being presented really around the HIV-1. We do have some genotyping kits that we'd be able to support you, especially if you need to be able to optimize or do anything. Specifically, once again, in terms of some of the infectious diseases that really are prevalent and a bit of a challenge in the context of Africa. And then last but not least, I really just wanted to highlight some of the solutions we have, specifically in efficient biologics, whether you're doing candidate identification and selection, and then really around the processing and the analyzing of your info. And then really, when it comes to NGS solutions, I've heard a lot of individuals talking about the Illumina platform. We do have a really great library prep kit that you'd be able to leverage and use, which is a color indicator. We know how it is when you're in the lab, you get distracted, you're not sure what's happening, and the next thing you're not quite sure which step in the process you are. We're able to support and assist you there. And then last but not least, I really, really want to thank our partners, because really, when I was reading up a little bit more about Ghana's national plan of 2018 to 2057. They've actually had a lot of national plans, to be honest with you, but hopefully this is the last one. It's really, really important to highlight the fact that what I'm seeing here at WACCBIP is really around the collaboration and really the focus in terms of building succession planning in the context of young researchers that will really drive the future of Africa. So I really wanted to highlight that what I'm seeing here today really is aligning to a lot of these key points within the national plan, as well as the 25 strategic areas that they really are trying to focus on. STEM is definitely on that list, and you guys really are the future. So once again, thank you. My contact details are here. You can find me on LinkedIn, and I hope I've saved you guys some time. Thank you.
So, Karma?
Yes, good morning, everybody. For those of you who don't know me, I'm Alistair Grant from Carrowmore International. We are a buying and logistic company for research in medical and life science, where that activity is happening in a low middle income country. So we started here in 1992. Really, the heart was in Ghana. We were in Ghana and Nigeria and Liberia at that time. This was pre-internet, so pre the World Wide Web, pre-email. For the younger people in this audience, fax was the way that we used to contact people, and you used to push a piece of paper through a machine, and it would go to one person. You would hope that it would arrive, and you wouldn't really know if it arrived. The world has changed, and we have been able to help, I hope, grow the activity that's been going on in Ghana. As well as equipment, consumables, reagents, the key part of what we're doing is the logistics. So we are an IATA cargo agent. We'll deal directly with the airlines, and we deal with all the cargo, prepare all the documents, send those out, and then help with the processes that are needed for permits and licenses, cold chain, dangerous goods. And in countries like Ghana, where we do a lot of activity, we also do the customs clearance process. So it's a delivered price. And we're working with lots of different manufacturers, so including Thermo, but other companies who may be in that field and completely different fields who can be competitors. So you can bring all those supplies in together. And during the pandemic, when we started, we were in West Africa, but over the years we spread into East Africa, Southern Africa. Now we're in over 100 countries, so we're in every single country in Africa, all 54, in the West Indies, and every country in Asia, bar 1 or 2 in China. So even if you have a project which is perhaps funded here in Ghana, but it's in another country, we can work with you. So we have a number of multi-country projects. We work with a lot of the collaborators in the north as well, so if the funding is there and it needs to go to Ghana or to another country, we can do all activity and report on all of that and deal with all those different countries. So I'm hoping that, going forward, we'll be able to continue to work with Ghana. I'm so pleased that the environment, which in 1992 it was really Japan and JICA, and a little bit, well, I don't think Lars is here, and a little bit of Denmark, but the environment has changed so much that the activity and the energy that's gone on here has brought in so much more activity. It's transformed the environment here in Ghana, and I'm very grateful for the ability for us to be able to help with the research that's going on here. Thank you.
All right. In the absence of the MC, I will take the opportunity to release you to go for coffee break, and then we'll be back in 15 minutes to continue. Thank you very much.
I will be doing like if I'm saying something. Oh, the microphone. I'm sorry.
Yeah, I thought I was taking the day off today, but I'm here. So we're running a little bit behind schedule again, but we'll try and catch up in these next sessions. Michael is not, Michael Yakas, who was one of the chairs, has had to travel, so since he's sitting right in front, I'm going to ask Dr. J.J. Harrison to chair in his place, along with Dr. Christelle Chokpa for the next session, which is host pathogen interactions.
Okay, thank you everyone, and welcome to this new session where we will listen to three researchers for three plenary talk. Let me call here one of our PhD alumnus, Dr. Jesley Shiraura, who is going to talk about investigating the role of epigenetic processes in promoting artemisinin drug resistance in Plasmodium falciparum. Dr. Jesley, you have the floor.
Hello. Good morning, everyone. Yeah, so I'm very excited to be starting this marathon, and I'm hoping that you'll find it fun. So, since today is a special day, special for alumnus or alumni, I'm happy to say that I've had both my master's, MPhil, and PhD training here at WACCBIP, and I'm currently a postdoc. So, are you cool to WACCBIP? Yeah. All right. So, this morning I want to share with you some of the work that I've been doing for the past 2 years, which is focused on investigating the role of epigenetic processes in mediating anti-malaria drug resistance. So the keynote speaker this morning, if you never believed in Bible stories, he was John the Baptist for this session. So, and I thank Dr. Kwashi for the arrangement. And so he already laid the foundation for epigenetics. So I'll just talk briefly about that. So by way of introduction, anti-malaria compounds like chloroquine, sulfadoxine, pyrimethamine, diatroartemisinin are known to generate reactive oxygen species as mode of their action. And this is associated with increased gametocyte production. Sexual commitment in Plasmodium parasite is mediated by, is marked by epigenetic activation of the expression of the master regulator known as PFAP2 gene, which is a transcription factor belonging to the API-AP2 gene family. In most cells, this gene is not expressed because it is epigenetically silenced by trimethylation of the lysine 9 residues on the histone 3 tail. And this H3K9 trimethylation is a prominent silencing mark in Plasmodium falciparum parasites, and it's found in the VAR genes, Refin, and ST4 genes, which I'm sure you've heard of during the course of this conference. And these genes are associated with host immune evasion, adaptation to adverse environmental conditions like drug exposure to antimalarials, and then virulence. And so, based on the keynote talk you listened to in the morning, we know that epigenetic processes are control key biological processes in malaria parasites, and so it is important to study them. So what is the link between epigenetic processes and drug resistance? And although there are several studies highlighting this, I'd like to use this to illustrate. So the first one is a study that showed that epigenetic switches in the clactric genes mediate blastocidin resistance in malaria parasite. And in this study, they found that the plasmodial surface anion channel, which is a solute transporter formed by the clactric protein, is epigenetically controlled by A3K9 acetylation and A3K9 trimethylation events. And this results in different solute uptake efficiency. And they also found that the subsets of parasites that were not expressing this protein, or the CLAC 3.1 version of it, were able to withstand blastocidin S. And the second study is the histone acetyltransferase, PFGCN5, which has been shown to regulate stress-responsive and artemisinin-resistant related genes in Plasmodium falciparum parasites. And in this study, we were able to show that this particular protein binds to stress response genes and regulates their expression under stress conditions such as exposure to artemisinin-based compounds. And inhibition of this protein in artemisinin-resistant parasites was shown to increase their sensitivity to artemisinin treatment, thus implicating the role of epigenetic processes in mediating drug resistance. And so based on this and other talks that you listen to, it is clear that targeting epigenetic pathways could be a winning strategy for malaria elimination. So, that was fast. Okay. So at the beginning of this study, we set out to determine if we expose Plasmodium parasites to artemisinin-based compounds, whether this could result in A3K9 methylation-dependent heterochromatin changes, and whether this had a chance to confer resistance to artemisinin-based compounds. So to do this, rather than using laboratory strains as shown in other studies, we opted to use clinical isolates with a Ghanaian background. And here we selected 3 clinical isolates that were able to successfully adapt to in vitro culture conditions. And we tested them against to evaluate their susceptibility to the artemisinin-based compounds. We screened them against dihydroartemisinin and artesunate, and we determined the IC50 values. So following this, we found that the A11 parasite had decreased susceptibility to both DHA and artesunate compared to the A377 isolate and then the K239 isolate. So following this, we figured that it would be easy to commit this parasite into artemisinin resistance compared to these two. So this talk is going to be based on the A11 parasite. So following this, went ahead to select for artemisinin-resistant parasite. And here, in this setup, we expose ring-stage parasites to either 900 nanomolar or 700 nanomolar of DHA for 4 or 6 hours, respectively, and then also 2.1 micromolar of artesunate, which corresponds to about 3 times the IC50 value of the 700 nanomolar. And we did almost about 5 different exposures, and we also kept them in drug-free media to see if whatever we observe, the phenotype that we observe would revert. So we set up in vitro growth inhibition assays to assess the susceptibility. And what we expect is that the development of resistance will mean an increase in the IC50 value, so we'll see a shift in the curve towards the right. And then we also set up ring survival assay to determine the survival ability of these parasites to these drugs. So in the first setup, after the first exposure, we got some of the clones, some of the parasites coming up after they were exposed to these antimalarials. Interestingly, we couldn't get the DHA, the one that was exposed to 900 nanomolar of DHA, but we were fortunate to have the one exposed to 700 nanomolar coming up. But rather than becoming resistant, it became susceptible, and which was very interesting, to both DHA and artesunate. And I had previously observed this with other drugs that I had tried selecting resistant isolates again. So it looks as if this is not just peculiar to artemisinin-based compound, but probably a general mechanism by the parasite. So we are asking the question whether this could it be mediated by genetic or epigenetic events? And so the goal is to actually find out why this is so. So following this, we did, as I mentioned, we did almost about 5 different exposures, and we found that at this time, the DHA 900 parasite came up, and it was almost 2 times less susceptible compared to the control. And when we look at its survival levels, we found that it had increased survival compared to control. And I'm highlighting this because the rest of the work will be based on just these 2. If you compare it to the artesunate, we see almost a similar mechanism here. So what happens when these parasites that seem to have resistant phenotype, what happens if you grow them in drug-free media? So 1 month after growing them in drug-free media, we noticed that the DHA 900 still maintained its resistant phenotype, and it was even more clear in both DHA and artesunates. But after 2 months, we saw decrease in survival to DHA, but not with artesunate, suggesting that probably this increased survival advantage that it had may be temporal or not permanent. So following this, we went ahead to also check for cross-resistance with existing anti-malaria. So we screened this isolates against chloroquine, mefloquine, and modaquine also. And we found that this isolate that was less susceptible to artemisinin-based products was also less susceptible to chloroquine and amodiquine. But the contrary was true for mefloquine. And this is understandable in the sense that both amodiquine and chloroquine belong to the same chemical class, 4-aminoquinolines, whilst mefloquine is a 4-quinoline methanol. So probably they have different targets, and so potentially this could be the reason why we're seeing this difference. So following this, at this time, this brilliant lady, Bridget Adika, who was starting an MPhil work with Dr. Niwe, we decided to give this isolates to her to test. So she went ahead to check to see their survival advantage. And even though she found less survival levels compared to the DMSO control, she went ahead to do a clonal selection. And the reason why we are doing the clonal selection here was that these parasites were exposed to the compound. We believe that not all the parasites would have been exposed to the compound at the same level, and so there was the potential that there could be different clones in them. And here she found that even though she selected for a number of clones, because for the sake of time, she couldn't test all of them. So with the few that she tested, we found some of the clones actually having higher survival advantage compared to the controls, while some actually also have lower survival advantage, or almost equal to that, and which is quite interesting. And it was also interesting to find that all of them had same genotype, or there were no clonal expansion in this parasite. So she also tested this against chloroquine and found that this parasite that seemed to have increased survival to artesunate also had decreased susceptibility to chloroquine. And this parasite, as you can see, the levels are lower than the one that was, this was the one that was more susceptible or had decreased survival advantage, had decreased survival, sorry. You could see the level, the susceptibility levels were lower than that. So following this, we're not sure whether, we know that mutations actually affect the susceptibility of these parasites to antimalaria. So she looked at some of the common drug-resistant genes, especially the calf-13, which mediates artemisinin resistance, and then also PFCR3 and PFMDR, which mediate chloroquine resistance. And as you see, we've observed only wild type for all of these genes. But interestingly, there were mutations in the DHFR and then DHPS genes in this isolate, which was quite interesting. So we don't know whether mutations in other genes could actually be mediating the resistance or susceptibility patterns that we observe. And this is the isolate that was found to be had increased survival, and this had lower survival. And you can see that there were virtually no differences between the two them. But if you compare the DMSO control, which had mutations, the DHA 900 had a wild type. So it looks as if some mutations, there were some mutations in other genes that were mediating this and not in the targets that we're looking. So in summary, I would like to say that we found increased susceptibility in the DHA A11 parasite to DHA nitrosamine after the initial drug exposure, which could be mediated by either genetic epigenetic events. And we were also able to select for parasites within 6 months that with a Ghanaian background that had increased survival to DHA nitrosamine. And these parasites were more susceptible to mefloquine, but less susceptible to chloroquine and amodiaquine. And we also observed that both these parasites we are selected and their clones did not have mutations in the calcitetin, PFCRT, and PFMDR genes, but had mutations in the DHFR and DHPS genes. So we are asking the question of whether this could be genetics, epigenetics, or both, interplay between the both that is mediating either susceptibility or resistance in this parasite. So going forward, we're hoping that going forward, we want to elucidate the mechanisms mediating genetic. We want to elucidate the genetic mechanism that may be mediating resistance in this parasite. And so far, we've been able to obtain extract samples for whole genome sequencing. We also want to look at epitranscriptomic modifications that may mediate resistance in this parasite. So we so far been able to optimize the protocol for ChIP-seq to look at histone modifications, and then later on also look at RNA-seq to examine gene expression in these genes. And so in the end, what we want to do is to see if there's an interplay between genetic and epitranscriptomic mechanisms that could be mediating resistance in this parasite. So on this note, I'd like to acknowledge the team that has been supportive, as Prof. Gordon, which you all know, and then Keck, who's assisting here, very helpful. And then this is Robin, and then his postdoc Manu, and then and everyone here. Thank you.
Thank you, Jesley, for your presentation. Now we give the floor for 3 questions.
Right.
Hi, very nice talk. I'm sorry I didn't see all of it, but I had just a small question.
Yes.
Because you're looking for these epigenetic modifications and epitranscriptomic effects. I was wondering if you had looked at DNA methylation profiles of your different strains and whether or not you could see changes in DNA methylation profiles related to the drug resistance.
Okay, yeah, so that's the future of the work. We haven't, so far, the first step we took with this project is to be able to get isolates that were interesting to study. And so we've looked at all of this, and so we're looking for explanations for why we are seeing these differences. So over the past few years, we've been trying to optimize a protocol for doing ChIP-seq that will allow us to look at histone modifications. Yeah. And so
so here I'm talking about more DNA methylation.
DNA methylation,
yes.
Yes. So we haven't looked at that yet.
Yeah.
So, but it's part of the future plans. Right.
Just to address Musa's question, Plasmodium doesn't methylate its DNA.
Right. Thank you.
No questions? Okay. Okay, thank you, Jesley.
Right. Thank you.
All right. It's my pleasure to invite Oyebola Dolapo to present on machine learning-based hyperglycemia prediction, enhancing risk assessment in a cohort of undiagnosed individuals. Is that going to be online or is it in person?
Okay, he's online.
All right. Kolapo, please take it over.
First, I'd like to appreciate WACCBIP for offering this platform, even though I'm not physically present. They were magnanimous enough to provide time for a virtual presentation, and I really appreciate that. I also must say that the topic of presentation slightly changed. Over the course of drafting my presentation, I included some of the other work that we are doing in our lab. So the title for my presentation reads Leveraging Artificial Intelligence for Infectious Disease Surveillance, COVID-19 and Malaria Case Studies. Thank you. So, it's important, of course, we all know it's important to carry out.
Kindly share your screen.
Okay, I thought I was sharing. Let me share again. Can you see my screen now?
Yes. Yeah.
Good.
All right.
Okay.
Is that a right presentation?
Can you see my screen?
Yes, we can.
Right. So the title of my talk is Leveraging Artificial Intelligence for Infectious Disease Surveillance using COVID-19 and Malaria Case Studies. That is slightly different from what's in the presentation outline. And as like I mentioned earlier, I needed to incorporate some of the other work that we are currently doing in our lab. And I appreciate WACCBIP for this opportunity. So we know that it's important to carry out infectious disease surveillance for so many reasons, to prevent and control disease outbreaks, and to ensure efficient use of healthcare resources, and to also inform decision making and policy development, and also prevent the spread of infections across borders. But there are different methods, approaches for this. The traditional methods involving lab surveillance, case reports, sentinel surveillance, but then there are now modern approaches which includes the use of electronic health records, mobile applications, machine learning, artificial intelligence, and genomics. This particularly is quick, rapid, fast, and also offers predictions for future outbreaks. So it involves the use of big data analytics, and of course it involves a bigger scope. You know, it's extensive and more global. So with that in mind, we in our lab are interested in a coordinated artificial intelligence framework for infectious disease surveillance using some of these approaches. For instance, we are interested in working on predictive modeling, logistic regression modeling, using supervised and non-supervised machine learning algorithms, and then deep learning approaches. So as far as this talk is concerned, I'll share with you some of the things we're doing regarding deep learning approaches for infectious disease surveillance, as well as using logistic regression models and machine learning, particularly supervised and non-supervised learning approaches for disease surveillance. And this leads me to one of the things the projects we're currently working on, which is to carry out retrospective analysis of effectiveness of non-pharmaceutical interventions against COVID-19. in Nigeria. So this is important because, yes, COVID-19 may not be a topical issue anymore, but of course we need to learn some lessons from the pandemic. And one of the things we set out to do was to look at the effectiveness of non-pharmaceutical interventions against COVID-19 at the early stages of the outbreak. Vaccines were not available, therapeutic options were not available. So most strategies involved the use of non-pharmaceutical interventions, including the use of face masks, school and workplace closures, contact tracing, public event cancellation, transportation cancellation, and the rest. So using available data in the public database, we wanted to find out the effectiveness of these MPIs, right? In case we'll need to use them in the future to attack or manage, mitigate the spread of pathogen X in the future. So we used correlation metrics and unsupervised, type of error here, correlation metrics, and then of course unsupervised machine learning approach, including k-means clustering, and then of course we use regression model to look at the effectiveness of some of these MPIs. So first, we need to understand the trend, all right, the trend analysis and transmission peaks during the pandemic. And then, of course, from these figures, we could tell that the first non-reported case was, in March of 2020 in Nigeria. And of course, we looked at the number of daily new cases and of course the number of daily new deaths using this smoothed values, meaning that we considered the possibility of fluctuations, corrective fluctuations. So we used smoothed values for our analysis. So in the early stages, there were, short, troughs, multiple peaks in short periods, duration or within short periods of time, suggesting that there were, a lot of challenges in the early days managing the spread of the virus. So it showed us some of the trends and analysis. So basically, what we also looked at was to look at how the MPIs, the effect of those MPIs on new cases and deaths. So we carried out regression analysis, used the regression model to predict compare the predicted cases versus the actual cases. And of course, we observed the moderate effect of MPIs for the mitigation of COVID-19 or reduction in new deaths associated with COVID-19. Both values were just moderate. So then we wanted to find out, okay, which of these MPIs were really crucial to the mitigation of COVID-19 cases and deaths, and of course the new positive rates. And then of course this plot shows reduction in new cases versus the MPIs used, reduction in new deaths versus MPIs used, and of course the reduction in positivity rates following testing versus MPIs used. So here you can tell that facial coverings and school closures seemed to have been correlated with the reduction in new cases, at least looking at it, correlated the most with the reduction in new cases, and of course cancellation of public events and school closures again seemed to have correlated with most with reduction in new deaths, and of course school closures correlated the most with reduction in positivity rates. So then we also wanted to know the patterns of the effectiveness of the MPIs, right? The characteristics, were they related in a way, the pattern of effectiveness, which of the MPIs could be grouped into a certain cluster. So here we used the K-means clustering to determine the similarity between MPIs and potential patterns of effectiveness. We were able to estimate our cluster number using the select scores. So this, each individual, like in this figure on the left, right, you could tell that the select score with the highest number showed the optimal number of clusters that would expect our principal component analysis to, and the groups of the MPIs fall into. So here, we use that number to estimate our principal component analysis. And you can tell here that contact tracing seems to be distinct from the rest of the group, which possibly suggests that the effectiveness of contact tracing, or the pattern of effectiveness of contact tracing, was quite, different from the rest of the cluster, right? And so going further, we make some inferences. I mean, of course, we had some more data to share, but of course, given the amount of time we have to discuss this, and of course, I have to move on to some other thing. But basically, the inferences here in the first project case in the country was in March of 2020, and then, of course, there were short intervals between peaks of new cases using deaths in the early months, which suggested that rapid transmission and potential, there were early challenges during the outbreak, of course, which is apparent. And then there was public transportation closure, which was part of the API's use, and it appeared to be the most impactful in reducing new cases. And then still restrictions showed significant association with the reduction in new deaths. So this actually, in summary, I highlighted the importance of multifaceted MPI approach to mitigate pathogen outbreak. So, and then of course there's another interesting project we're carrying on in the lab, and this was born, it was based on the fact that more than 90% of malaria tests in Nigerian public institutions use RDTs, right? So RDT is mostly in use. We have other, tools, microscopy, PCR, which, are not very significant compared to RDT. But in any case, if RDT is mostly in use and we have that threat of HRP2, HRP3 deletion in malaria, then of course we should be looking at other diagnostic tools. So I, gave my PhD student the research topic to work on developing a mobile or a web-based, web app-based malaria diagnostic tool using convolutional neural network. So that brought about the idea of developing a convolutional neural network-based algorithm for detecting malaria parasites in blood smear images, and we named it Plasmo3Net. So basically, what's this about? So first, we have a set of images obtained by microscopy, the microscope slide images that we conventionally use, and then of course we process the images to be recognized by the computer, and of course develop a model architecture which is convolutional neural network based, and then of course train and validate our model, okay, and then of course compare our model, the performance of our model with other established model, models related to, used for the same purpose. So basically that's what, the whole project is about. I'm not going to go into the computational analysis or the computational details of coding and analysis. So So we had about 14,980 images which were generated following augmentation. Okay, 7,419 uninfected from uninfected parasites, blood smears, and then of course 7,490 infected blood smears. Here we are focusing on Plasmodium falciparum. Okay, and then of course
please kindly try and wrap up for us.
Oh, okay, so the training and validation data sets were about split into 8 and 20. So I didn't know I was already out of time. But in any case, let me just go to this, okay? So what the whole model is about is to be able to detect malaria parasite. This is a detection for in the first instance. We're not working on speciation yet, and we're now also looking at different stages of the parasite, just detection. And from what we could, carried out, we were able to detect, these are samples from our model. We were able to detect parasitized blood smears, and of course also detect parasite blood smears, and the performance levels where the accuracy was 99.3, and the precision level was 99.1, and recall was 99.6 compared to other existing pipelines. So I'd like to thank WACCBIP for the opportunity. Of course, also thank the funders for NIH, Bill and Melinda Gates Foundation, RF, EDCP, and African Academy of Sciences. And I'm also mention, acknowledge my team members that are working with me in the lab. So thank you very much for this opportunity. I thought I was going to be within time, but thank you anyways.
Okay, thank you, Ayebola, for your presentation. We give floor for 2 questions. Okay, thank you. Let's go for the next presenter, who is a postdoc alumnus at WACCBIP and currently an APTI fellow in Kenya. Dr. Daniel Kiboi, welcome.
So hello. Okay. So thank you for this opportunity. So at the start, maybe I need to thank WACCBIP for the invitation, and also thank you everyone for coming for presentation. So it has been an honor to be part of the WACCBIP family, and also an honor to be part of this celebration. So congratulations, WACCBIP. So my name is Daniel Kiboi. I'm from Kenya. I'm an alumni of the WACCBIP, currently an APTI fellow. So just finished my first 2 years at the NIH. So the work that I present would be based on what I did at the NIH under Tom Williams Lamb. So so you hear about me mentioning about persisters, or tolerant, or let's see, dormant parasites. So in bacteria and let's see, cancers. So those words are used interchangeably. So so the title of today's is Isolation and Characterization of Plasmodium falciparum, blood stage persisters by improved selection method. So, and I don't want to bore you with a lot of introduction in terms of the malaria burden. But I just want to take you back to history. And in terms of history, so it's just to say that we received a gift from Chinese medicine in terms of the artemisinin. And right from the discovery of these drugs, you agree with me that there was, they have changed the landscape of treatment in malaria, for malaria, that is globally. But within this change of landscape, you realize that from the inception of the use, then the treatment of malaria using an artemisinin drug, all its analog, as a monotherapy, resulted to more than 40% of recrudescence in patients. So, and even the 7 days course of monotherapy of artemisinin also resulted to recrudescence. And this was before, and this was long before the emergence of the artemisinin-resistant phenotype. So, what caused this, recrudescence? So, one of the main reasons is what we call artemisinin-based or induced dormancy. So, what is this dormancy? So, this dormancy is basically a parasite temporarily arresting its growth and for days or weeks, and then resuming its normal proliferation and to cause another infection or to cause new infections. So, sorry. So this slide shows the life cycle of malaria, but the most important thing I want to highlight is the ring stages that the dormancy occurs at the ring stages. So this is the ring stage that is normally you see on the slide when you make a slide. This is normal active ring stage, but when this ring stage is exposed to drug, or let's say DHA, dihydroartemisinin, all and any stress, so the assumes a dormant morphology, and this dormant morphology, you take note, remember this, that assumes a dormant morphology of a condensed chromatin and a small cytoplasm. So, and this can remain for days or weeks before resuming the normal proliferation when the stress is relieved, or maybe adaptation to stress. So, and then the next question that you ask is, so is induction of dormancy only in artemisinin? And the answer is no. So you realize even in other drugs that have come before, like pyramidine-induced dormancy, this was shown to cause dormant parasites, and also recrudescence happened after pyramidine treatment. So it doesn't matter whether you increase the dose or reduce the dose. And again, you also in sorbitol, and sorbitol we use it often in the labs to do synchronization. So, and you see even sorbitol does not have any activity against parasites. So you see that recrudescence happening. And this as we have also done in the lab, and we see in the dormant parasite in terms of the morphology that I've explained. So, and recently there was also shown another paper on the chloroquine-induced dormancy. So, and you can see from here, so these are the days the dormant parasite were observed, and then on day 11, day 13, you see now emerging with the new, the normal active parasite. So this all in vitro. So does it happen in vivo? So, and the answer again is yes. So, sorry. Yeah, so this is a study that was done in Australia, and they showed using the control human malaria infection, and they showed after infection, and then you treat with a testinate, then a dormant parasite was observed on the 3rd day. So, and these were isolated, and it doesn't matter whether the parasite carries a K13 mutation that is associated with slow clearing phenotype. So now I've mentioned about the dormancy aspect, and whether it's in vitro and in vivo. So, but then there's need to understand this mechanism of these dormant parasites. And there are a number of ways that people have done it. And and the most popular method is this, where you have a mixed parasite stages, and then you do synchronization using sorbitol, 5% sorbitol, and then on 0 hours you do DHA exposure for 6 hours, do the wash, and because not all parasites are killed, then there are 2 methods that you can eliminate the parasites that have evaded have not been killed. One is magnetic column, which is expensive, and then requires manipulation of the parasite outside the normal culture conditions. So then the other approach is you can either use sorbitol to remove all the other parasites that have not been killed or induced into dormancy by DHA. So, but these methods, like I mentioned, they have So these methods are all confounding, these steps are all confounding in terms of isolation of dormant parasites. So how do we improve this protocol? So and we can take advantage of the activity of the DHA itself. So and this slide shows that despite the slow clearing parasites, so this is a slow clearing parasite having a mutation in K13 and also fast clearing parasites. You see at the ring stages, so there's a difference in susceptibility, but at the 9 to 12 to 18 hours, there's no difference, there's no significant difference between the two parasites. So therefore, you can use DHA to use as a method of inducing dormancy without having to use max column or using sorbitol, because they introduce confounding steps in those steps. And that is the way I did, and I used So I used two parasite line. One is from Cambodia, has a phenotype of C580Y, and the other one is GB4, coincidentally is from Ghana, and has doesn't have the mutation, so this is wild type for C580. So then I used mixed parasite stages, approximately more than 70% ring stages, 2% parastemia was the starting parastemia, then hematocrit of 5%. Then I used to monitor dormant parasite because he is very rare and also would go to low parastemia. Then I used two stains, cyber green the stains, or the infected red red cells, plus the mitotracker that stains the parasite that it has intact mitochondria, meaning that parasite is live. So I used, but also confirmed, use GIMS, normal GIMS stains for the morphology. So, and this is a busy slide, but the aspect is we optimize what we call the 4-drug pause experiment, and we did at every 24 hours a treatment, and after 6 hours we did a wash, and this is for 4 days. And then we monitored for the until 2%, which was the starting parastemia, which was the definition of our recrudescence. Then we did the same with our vehicle, which is DMSO, the same. And at every 48 hours, because of the parasite growth, we diluted and then recultured until 14 days. So we replenished red blood cells, 10% of it, like 100 microliters of it at every 7 days. So what did we get from this? So this is how we monitored. So I stained with cybergreen and mitotracker, and this is what we quantified as the dormant parasite, and this is what we used to draw the recrudescence curve. So we also interested in, once we do that, we was also interested in sorting and separating of these dormant parasites. So we still use the cybergreen and mitotracker, and these are the pycnotic ones that I'm labeling here. These are the dead ones. These are the ones that we say that mitochondria intermediate, meaning they have an intact mitochondria, and then these are the high ones, just in case you have either trovozoites or schizones. So we use this to assay for some genes, and also we use for cytospin. Cytospin is just once you collect that 50,000 cells, you concentrate them, use centrifugation, and then you you splash them on a slide, and then you watch you observe them on a GIMS-sustained slide. So this is just a recrudescence curve of what you observed, and we use different concentrations, 200, 400, and 700. So, and you can see this just shows that the black one shows that the DMSO grid as expected, with dilution at every 48 hours, and 14 days we stopped because, the parasites were growing as expected. But you can see there's no big difference between the 200 nanomolars and 700 nanomolar in terms of recrudescence. So there was no significant difference when you analyze. And this was the same for the GB4 line that does not have the C580Y mutation. So now the question also arises is, so can we measure, so what was the morphological, or the morphology of this dormant parasite that we selected? So we observed this is what we did with cytospin. So we collected like 50,000 cells and then spun them on, and then stained them on a slide. So, and you can see as the one that is zoomed, this is on day 2, which have received 2 drug pauses, and you can see the dormant morphology, assuming the dormant morphology. So, and then we monitored throughout all the different days, that is day 4, day 7, or day 11, 14, and 16. And you can see from day 11, they start assuming resuming the normal ring stages. And this is day 14, where you have now expansion of ring stages now. And then from day 16 or the day 21, now you have a recrudescence in terms of parasite resuming normal growth. So the question that we now asked is, can we now do a molecular analysis of some of the markers that are associated with, one, dormancy, and two, the ring persistence. And we look at two genes that have been associated with dormancy, and one of them is acetyl-CoA carboxylase. So we measure we use this one because it has been used by Pitt et al. And they showed that despite the parasite having the KTAT mutation, there was upregulation of these genes after 3 days, that is after induction of the dormancy. So then we also used another one, which is skeleton binding proteins, which is basically measures or detects or measures the ring stages and is only expressed at the ring stages, so we can monitor the persistence of ring stages. So what we did was to follow up those genes, and this is the data on that. So we first determined at the baseline what was the expression at this baseline, and then we followed throughout the recrudescence curve. And you can see the red dots are the ones that are significant. You see a spike on day 7, and for the ACC one, and then you have a downregulation on day 14, which we associated with maybe like waking up of the parasites after dormancy. So you see the same. You have on day 2 you have a spike, but it was not significant for skeleton binding proteins. But this also measures that increased expression of the ring stages in the dormancy state. But then after waking up at day 14, then you have a downregulation on day 14, which seems to be the same as ACC. But then again, we associate that dormancy is a temporary phenotype, and we also see that it could be temporary mechanism, molecular mechanism, and you see returning back to the normal baseline levels after recrudescence. So in conclusion, we have developed what we call a 4-day drug pause and FAC separation procedure for efficient purification of population of dormant parasites. And this requires no sorbitol treatment or magnetic columns, and therefore we can use it as one way of purifying DHA dormant parasites, DHA-induced dormant parasite that we can study for molecular analysis. So again, we also see that the return of acetyl-CoA carboxylate and skeleton binding protein to the normal baseline levels suggests temporary nature of the molecular mechanisms of associated with the dormant phenotype. So with that, I want to acknowledge many people. So the first is Tom Willems, where I did my mentor, where I did my first 2 years. So Juliana, Akshay, and all the others, Noah, Alex, Kiara, and also Anna. So when we had a person who helped me, flow cytometer is Caroline, and then we have LMVL administration. And then we have AOT, we have Sharon, Gail, and then we have Fogarty, which is Peter, Stacy, and Jackie. And of course, Mu, who helped me with the genomic core at the LMVL. Thank you.
Thank you. Thank you, Dr. Kiboy. We give the floor for 2 questions, please. Okay.
Hi, thank you very much, Dr. Kiboy, for your wonderful presentation. Please, I have a question, and I would like to find out if you try the pulse exposure beyond 6 hours to find out if that could have probably had an effect on the dormancy in terms of whether there was delayed development of dormancy or timely development of dormancy in the ring stages.
So you see that without what? Repeat the first statement, please.
So did you try pulse exposure beyond 6 hours to determine if longer pulse exposure had any effect on the dormancy stages?
Yes, so we have, okay, this I didn't show that, like longer exposure, like 24 hours of DHA exposure.
Yeah, so instead of the 6 hours, this time around, you make it 12 hours or 24 hours.
Yeah, so we didn't determine that, but it's something that we thought we would need to give the parasites like time to recover. and you know, just pressure them for the second day. But that's a good idea that we can push them for like 24 hours and then remove the drug. Yeah.
Thank you.
Yeah, thank you so much for the presentation. So I'll like you to clarify. So with this dormant morphology we are seeing, can you hear me? Yeah.
No, I can get. Go ahead.
Yeah.
Go
can you
ahead.
hear me
Go
now?
ahead.
Yes. So with the dormant morphology you're observing, how sure are we that this is actually something the parasite is, this is a form the parasite is taking to escape drug, and rather not part of the morphologies the parasite takes when it's actually dying? In a sense, also following up with what she asked, if you continue to put the drug pressure for 12 hours, 24 hours, how sure are we that the parasite is not going to die?
So yeah, so I'll answer the second question first. So like I showed before from the first slide, like people have tried even this in vivo, like even 7 days course with that medicine, and the parasites still come back. So it doesn't really matter how far, I mean, the number of pulses that you give, especially for DHE, they still come back. So the second question you mentioned about the morphology is whether they assume it, and then return it, or I mean return to the normal. So it's a morphology that it's a way of mechanisms. That's what you asked the question.
Okay,
a way of drug resistance.
Let me take it again. So when you apply drug pressure, there are some morphologies the parasite takes towards death, right?
Yeah.
How are we sure that this is not what's happening, rather than the parasite actually actively taking this morphology to escape drug pressure?
So there are two ways. So I'll answer this way. So the parasites, so when we assume that, in my thinking, we assume that these dormant parasites already possibly exist. But we also assume that other drugs can induce dormant parasites. So the reason I say that is because if you use sorbitol, which we tried, that has no activity against parasites, so you still have those dormant parasites. It has no activity, just stress. So, but then if you use DHA, then you have more dormant parasites. So I agree with you that this is one of the mechanisms of parasites assuming, you know, resisting drugs or escaping drug action. I wouldn't say resisting drug. I would say maybe escaping drug action.
All right. Thank you.
Last question, then we can end this session.
Daniel, thank you for the wonderful talk. Do you think at all, I think you and I have discussed this already, but do you think at all the immune system may play a role in inducing dormancy in any way? Since you think it's a phenotype that is there, but maybe drug stress increases the probability or the possibility of developing dormancy.
Yeah, so digging deep into the clinical aspect, I think parasite respond to stress, my thinking would be to respond to stress, including like immune pressure. But I wouldn't say that they are responding to, you know, in a way of dormant, assuming that morphology. So that one I will not say because I've not done the in vivo aspect. But in terms of drug, we have, you know, there's a lot of observed studies in terms of dormant parasite responding to that, you know, assuming that morphology in response to drugs. In terms of immunology, I may not say that.
Very much. We can. Yeah. Peter, our job is done. Thank you.
Thanks, Jerry and Crystal, for keeping us, getting us back on track almost. Thank you. Before we go for lunch, I know lunch is the next item, but we have an alumnus who is going to give a short talk about a study that he's, so you can come up. Just like 2 minutes.
I think they're recruiting healthy volunteers.
Hello, good morning. Sorry, good afternoon. So I'm actually Michael Berma Kumi, and I'm here with the alumni Jot Ni, and we're actually from Azidos, Ghana, and we are here to talk about our organization. So Azidos Laboratory Limited is a bioequivalence bioavailability center, and we're actually the first in West Africa, as well as the first in Ghana. We have our mother company in India, and they've been in existence for about 20 years now. As well as we have branches in the USA, we have branch in Brazil and other countries. In the USA and internationally, we are recognized by the US FDA, as well as World Health Organization and other health regulatory bodies. In Ghana, we are being approved by the Ghana FDA and other health regulatory bodies. So what we actually do is bioequivalence, bioavailability, and what we do is we compare two medicine, and we believe that every medicine which is being sold is actually in two forms, which is the innovator form and the generic form. The innovator form, we say the first company that discovered the medicine and actually the first company that produced that medicine. The generic is like a photocopy. We're actually producing a brand of the innovator. So what we do is we compare these medicines and we are looking at their bioavailability. So comparing the two, that's the bioequivalence, where we are looking at their concentration in the blood. So basically that's what we do. And with our study, we use healthy participants. We use healthy participants for our study. The reason why we use healthy participants is we want you to come to our facility healthy as well as live healthy. So that's why we use healthy participants for our study. We can actually monitor you. So as well as our study, we check in our volunteers are being checked in at our facility, meaning our volunteers will actually have to stay at our facility during the process. That's amazing. So with that study, when a person, before our study begins, we'll actually screen you. We know you are healthy and you tell us you are healthy because we'll be asking certain information, but still we'll actually screen you where we'll be doing a biochemistry test, we'll be doing hematology test, serology to make sure you are fit. So that's one benefit of volunteers, that we actually screen you and you know that you are healthy. Now when you pass our screening test and you are fit for our study, we actually call you for the study where we give you the time for our study. And we check you in a day before our study. And we check in a day before our study so that you don't take any medication or any food that might interfere with our study. Because we're actually going to do medicine, we are going to do drugs, medicine. And probably might take something which might interfere with our study. So we check you in a day before, and the day of the study, we give you just a dose of the medication. So we give it a dose where we'll be taking your blood. We have our time points where we'll be taking your blood, and that will be for the first med. And depending on the study, you are being made to stay there for about 36 hours. Where we release you, you go and come back. We'll let you go and come back for the second medicine because we are comparing two medications. So it depends on the study, we'll let you go a week or two. With some studies too, after taking the first dose and your blood samples are being taken, we still make you stay there and give you the second dose. And when we house you, we take care of your feeding, everything. We take care of your bedding and everything that will make you comfortable. Yeah. So we actually don't just tell volunteers after the study to just go. Yeah, we are very thoughtful, and we also we are very grateful that our volunteers really spent off their busy schedules to be with us. So we volunteer and we compensate our volunteers. We talk to you. We'll actually be around for some time. We'll want to actually collaborate with other scientists around and also see how best we could help the science community as well as medicine is involved. Thank you very much for this opportunity. We are very grateful.
All right, so that's the end of this session, and without much ado, let's go for lunch. Hopefully it's here.
Have the beads. We have the traditional ones and the ones for casual. And we have bags. Yeah, we have bags. We have brooches too. Yeah. And we have the material cloth, the fabrics, and these are the African prints we have here.
Surprise. We have made in Ghana, batik, handmade. We have KJ glove. We have the GTP that also made in Ghana. And we have sewn the material as well. We have the ladies tops and ladies dresses. And we have something we call it the different designs. We have a lot of them here. We have an office wear as wrap dresses. We have the casual wear and rich auntie as well.
We have kids dress. We have necklaces, fans, and materials.
My name is Francisca. I deal with all kinds of African wear and African crafts in handmade. Example like this. This is a talking drum. It's made with animal skin with a symbol at the front and the back. And I have the clock made with African print. This is African print. And there's a Ghana map inside. And we have magic bag. This is called magic bag. This is how it is. And when you finish shopping, you just fold everything back this way. Here is even a champion, and here we have so many African prints. We have the kente, we have the fans, we have the purses, and we have the beads. We have the traditional ones and the ones for casual. And we have bags. Yeah, we have bags. We have brooches too. Yeah, and we have the material cloth. The fabrics, and these are the African prints we have here.
Enterprise. We have made in Ghana, batik, handmade. We have kente cloth. We have the GTP, that also made in Ghana. And we have sewn the material as well. We have the ladies' tops and ladies' dresses. And we have something we call it the different designs. We have a lot of them here. We have office wear as wrap dresses. We have the casual wear and rich auntie as well.
We have kids dress. We have necklaces, fans, and materials.
My name is Francisca. I deal with all kinds of African wear and African crafts in handmade. Example like this. This is a talking drum. It's made with animal skin with a symbol at the front and the back. And I have the clock made with African print. This is African print. And there's a Ghana map inside. We have magic bag. This is called magic bag. This is how it is. And when you finish shopping, you just fold everything.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space. And also, that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department. And the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the thing. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Teresa, Osborne, Abidad. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Ayite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than 50 million US dollars for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development.
All right. I hope we didn't eat too much, and we'll be live and active for the second half of today, which is the last half of the conference. And it's my pleasure to introduce our next keynote speaker. I think almost 4 years ago when we got the Geyser grant from Bill and Melinda Gates, we had this pseudovirus assay going, and they're like, yeah, we want you guys to talk with Penny Moore and figure out. Long story short, we had the same assay. But Janelle was leading a lot of that work, and we've met over the years. And incidentally, she was supposed to speak on Tuesday. So I'm not going to go into her profile because it's right here. But she's had excellent training from Wits University? Yeah, from Wits University, has gone to the US, worked in the same department as Ian Wilson, because I forgot who your supervisor was. But she's done excellent work and has been leading a lot of the HIV work at NICD. That's the National Influenza Control, and NICD in South Africa. And also did a lot of work in SARS-CoV-2, detecting some of the variants of concern, though they've been overshadowed by certain people. Anyway, and we've met over the years along with Chinedu in the Geyser program. She co-leads Geyser South Africa, and they support a lot of the Geyser sites. And 2 of our postdocs just got back from doing rat-pig training, spending 5 weeks in their lab. And she's a fantastic speaker, and I look forward to hearing about what they're doing currently in South Africa. Oh, and incidentally, the way the schedule worked out, she was supposed to speak on Tuesday. She's speaking now, and I'll leave her to tell you about how she's related to the person who speaks after her.
Thanks, Peter. And thank you to WACCBIP and the conference organizers for inviting me to speak with you today. It's been a really fantastic week of science, and I've loved every bit of it, even though most of it I'm not an expert in. And congratulations, WACCBIP, on your 10-year anniversary. And he's waiting outside the door. But happy birthday, Peter. Okay, so I'm going to present today 3 studies that we've done looking at SARS-CoV-2 neutralizing antibodies, mostly in the context of infection-induced immunity. This way. Am I pressing the right button? Okay. All right, so I always like to start with putting the situation into context. So this is the SARS-CoV-2 epidemic from 2020 until 2023 in South Africa. And so on the vertical axis is the number of sequences, genomes, and then on the horizontal axis are the epiweeks. And so you can see, as with most other places in the world, there were numerous epidemiological waves, and each of these waves was characterized by a different SARS-CoV-2 variant. What am I doing? There we go. And you probably all know this, but each variant that emerged with SARS-CoV-2 had accumulated more and more mutations until when we reached Omicron, it had 30 mutations or more away from the original ancestral SARS-CoV-2. And now, of course, we have Omicron 2.0 with the BA 2.86 lineages and sublineages that now have an additional 30 mutations on top of that. So in the context of South Africa, you can imagine the situation happening with very low levels of vaccination across the country, and also most of the immunity that you'll see is infection-induced immunity that has resulted from multiple infections. Okay. The other big epidemic in South Africa is the HIV epidemic, and so I can't go through a whole talk without speaking about that. So in South Africa, we have about 7.5 million people living with HIV, and more than 2 million of those people are not virologically suppressed. And if you look at the little schematic on the side, there's a nice little summary in a review that one of my postdocs just did, which talks about the impact of co-infection of SARS-CoV-2 and HIV. And for the most part, we did not see an increased susceptibility to SARS-CoV-2 in people living with HIV, but we did see increased hospitalization and mortality. And most especially in ART-naive people living with HIV, we saw evidence of prolonged viral infection and prolonged shedding thereafter. So that brings me to what we were really interested in. We were interested here in how does this prolonged SARS-CoV-2 infection actually affect viral diversity in people living with HIV. And so we worked with a group from the NIH, from the Vaccine Research Center, led by Eli Boritz, to look at sequencing of people who had prolonged SARS-CoV-2 infection, whether they were HIV positive or negative. I just want to, yeah, okay. So if you look at this phylogenetic tree, I'm actually just going to block off one part of this pie chart, this phylogenetic tree. And so if you look at these individuals here in the light blue, these are all individuals that had people living with HIV that had a CD4 count less than 200. So they're immunocompromised. And then if you look at the people here, these are people living with HIV that have good CD4 counts, that are well controlled. And then in the black here are people without HIV. And the stark thing that you see when you look at this figure is this insane amount of SARS-CoV-2 viral diversity in people with HIV who are not virologically suppressed. And this is going to come into importance later on in the talk. But a lot of these individuals actually developed beta-like mutations. So they were infected with the D614G or the ancestral variant, but they started making mutations that looked very similar to the beta variant. So it really supports this theory that these variants of concern are emerging from individuals who have prolonged infection. So we were really interested to know, what is this increased viral diversity in people living with HIV? What effect is it having on their antibody response? And how does it compare to people who don't have HIV, but who also have prolonged infection? And so we selected some individuals, or we managed to enroll some individuals in a hospitalized cohort during the first wave of infection. So all these people had sequence-confirmed infection with the D614G or the ancestral variant. Again, they had severe COVID, and all of them had at least 6 weeks of shedding post-hospitalization. And so we tested longitudinal serum samples from these individuals at each week of hospitalization for binding and neutralization against 3 of the SARS-CoV-2 variants listed here. And then we also looked at their effect against SARS-CoV-1. So if you look at this graph, which shows here spike binding antibody titers on the vertical axis and then days post-symptom onset on the horizontal axis, you'll see that we didn't have consistent sampling. And for some reason, the confidence intervals did not come up. But you'll have to trust me then. So because we didn't have consistent sampling with the same number of samples from every single individual, we used polynomial regression analysis to be able to assess the differences between the HIV, people living with HIV in red, and the people without HIV in gray. So if you just focus now on these trend lines, you can see that consistently between these time points, not really early in infection and not really late in infection, but somewhere in the middle, you can see a consistent difference between these two groups, with people who don't have HIV having significantly higher antibody titers than the people living with HIV. And we saw this trend matched also in binding antibody titers against beta, BA1, and even SARS-CoV-1. But I think this here is what is really striking. Regardless of whether you had HIV or not, you had these cross-reactive responses against the beta variant, the BA1 variant, which was significantly different to what they were infected with, and SARS-CoV-1, which is only 70% similar to SARS-CoV-2. So really striking and really interesting. So we then also looked at the neutralizing antibody responses in these people. And I haven't done the polynomial regression analysis yet because, as you can see, we haven't completed testing for these samples yet. So what we're showing here is area under the curve as a measure for that longitudinal neutralization that we see for each individual. And for both the D614G and beta variants, you can see that the people with HIV have significantly lower antibody titers than people without HIV. This didn't hold true for the BA1 and the beta variants here. The BA1 and the SARS-CoV-1 variants over here. We're not really sure why that is. It might be real, or it could change when we complete the testing. And you'll see actually there's a lot fewer individuals that we've tested here compared to D614G and beta. But still very interesting that all these individuals develop not only cross-neutralizing binding antibody responses, but cross-neutralizing antibody responses as well. So what we then did is we looked at the median titers for each group. So these are now neutralizing antibody titers for the people living with HIV and without. And what you'll notice here is that in the people living with HIV, their titers against D614G are 32-fold lower when you compare the titers that the people without HIV have. That's a huge, huge difference in titer. But as you move along this spectrum of variation, you'll see that that fold difference actually becomes negligible to the point where, against SARS-CoV-1, the people living with HIV actually have higher titers than the people without. And so this suggested to us that these people living with HIV, even though they have a much lower antibody titer, it seems that they have a better quality of response in the sense that their response is much more cross-reactive. And that's basically what I said there. But the conclusion then from this study is that regardless of HIV infection, people with severe COVID and prolonged SARS-CoV-2 infection develop these really amazing cross-reactive antibody responses. They're infected with the D614G variant, but they can even neutralize SARS-CoV-1. And then, despite the fact that there was no difference in cross-reactivity of responses, we think that how each of these groups get to that differs. So the people who don't have HIV, they elicit this enormously high antibody response. And so just by chance, we think that they also then elicit these low-frequency cross-reactive antibodies. In contrast, because of what we know about this increased viral diversity in people living with HIV, we think that even though they elicit a very suboptimal antibody response, that response begins to mature to become cross-reactive because it's exposed to all these circulating variants. So, if you recall, I said that some of these individuals here developed beta-like mutations over the course of their prolonged infection. And so we wanted to know, does this cross-reactivity that we've seen in these HIV-infected individuals, does it translate on a population scale in people who were actually exposed to the beta variant? And so we leveraged the National COVID Antibody Survey, which was conducted between November 20 and February 2021. It was set up ready to establish infection rates of SARS-CoV-2. And the timing suited us very well. They didn't obviously anticipate that this would happen, but the collection period happened right at the time when the beta variant emerged. And so while they enrolled quite a number of individuals, about 14,000 individuals, we actually were interested only in the people who were nuclear capsid positive, who had evidence of SARS-CoV-2 infection. And then from those, we tested them for spike binding antibody titers, and only the ones that were positive were then screened for neutralization of both the D614G and the beta variants. And so here's just a breakdown of the 4 profiles of neutralization that we saw. There were some individuals who had absolutely no neutralization, even though they had binding antibodies. Others that were only able to neutralize the D614G variant, some that were only neutralizing the beta variant, and then these very rare, very interesting people who could neutralize both variants. Well, they're not rare, but very interesting. And so this is just a map of South Africa. And so at the top here, the shading represents the frequency of D614G variants that were present in the different provinces, and the darker the color, the more of that genome was present. And at the bottom here is the frequency of beta variants at the same time point in the same month. So in November, there was quite a bit of variation, with some provinces having high levels, some provinces having low levels of one or the other of these variants. But by February 2021, 4 months later, the beta variant had completely replaced D614G and was present at more than 90% frequency in all provinces. So what we wanted to do was investigate how the antibody response at a population level is changing as this variant is emerging. And so this is just a really tiny little plot showing you the raw neutralization values. So we've got antibody titer here on the vertical axis for each of the different provinces. The blue neutralization is of D614G, the purple is of the beta variant. And I don't want you to pay too much attention to this graph, only really to look at the fact that the median titers against beta were low compared to the D614G variant, and this really persisted across all 4 months. But what I really want you to look at is those people that were double neutralizers, those people that neutralized both D614G and beta, because those are the interesting ones. And so what we did here was we looked at a fold change in titer. So if you have dots, which are representative of people, above this dotted line, which is the line at position 1, those individuals are able to neutralize the beta variant better. The ones at the bottom are people who can neutralize the D614G variant better, and then everybody here in black, they can neutralize both viruses equivalently. And what you see is that as you get this replacement of the beta variant, well, the dominance of the beta variant, you also see a nice shift in population immunity. So in November, when there was still quite a lot of variability in genome frequency, you can see that most of the individuals had better neut against D614G. But as time goes by, that D614G slice decreases, and the better neutralization against the beta variant or equivalent neutralization against both variants increases. And so we also wanted to, that's just frequency, that's just numbers, we also wanted to quantify the quality of those responses. And so we looked at median titers of each of the either the beta neutralizers, double neutralizers, or D614G double neutralizers over the same period, so over this period when the beta variant came to dominate. And you can see here, as the frequency of the beta variant increases, so too do the GMTs against the beta variant. And we have a concomitant decrease of the D614G median titers, which was really nice, and it shows that yes, at a population level, you can have these massive sweeps in humoral immunity. And so to summarize that little part of the talk, different variants trigger obviously different specificities, which is what we've seen consistently over the course of this pandemic and now epidemic. But intriguingly for Peter, it looks like the beta variant elicited more cross-reactive responses even at a population level. So even if we're not looking at a hospitalized cohort, most of these people probably had asymptomatic infection. And so then for the next part of the talk, how does infection-induced immunity then translate into protection from infection and protection from reinfection? So this is the last part of this talk. And it's really just going to be speaking about correlates of protection and antibodies as correlates of protection. So if you recall, in the early days of the pandemic, there were many randomized controlled trials. Everybody was looking at correlates of protection at a peak time point post-vaccination. And obviously, antibodies came out tops as a winner. But very little since then has been done looking at what are the correlates of protection after infection-induced immunity has been elicited, and more especially, what does it look like against immune escape variants like Omicron? And then the kind of other lacking thing in the field is what is the impact of antibody waning? I mean, we know that the antibodies wane. We know that we're still getting infection, but how do we quantify that impact? And so what we did is we worked together with Cheryl Cohen and Cecile Verbeau and Sankey Kayun San on a household transmission study. So this was like a really insane feat. We set this up at the peak of the COVID-19 response, as if we didn't have enough to get along with in terms of our public health response. And so what we did is we enrolled individuals from a rural site here in Agincourt and then an urban site in Klerksdorp. And we enrolled about 100 households from each of these two sites, total of about 1,200 individuals. And we wanted to follow these people up. We wanted to swab them as often as possible. We wanted to collect blood and find out everything we could about SARS-CoV-2 and what it was doing in our communities. And this paper was just published last week Friday, so I will skip over some of the detail, but it's available if you're interested to find out more. So this is just going to help me orient you in terms of the cohort and how we set it up and how we test it. So like I said, the cohort was set up at the peak, June of 2020, and we followed these individuals all the way up. Here on this plot is up until May 2022. We had a very intense follow-up period that ended here in August 2021, so actually just after the Delta wave. And these individuals were swabbed twice weekly. Those swabs were tested with real-time PCR for flu, SARS-CoV-2, and RSV. And then here as well, you can see we collected blood at 2 monthly intervals. And so this is the rural site, this is the urban site. You can see in the urban site, we just missed, we came in a little bit too late. We came in once the first wave had already been pretty well established. But nevertheless, we used serology, so anti-nucleocapsid antibodies as an indicator of infection in those individuals. Okay, so actually before I come here, so we've highlighted here in blue blood draw 5 and then in red blood draw 8, because these are the two blood draws that preceded either the Delta wave or the Omicron wave. And we use these two blood draws as our markers for assessing correlates of protection. So either correlates of protection against Delta or correlates of protection against Omicron. Okay, so firstly, we wanted to quantify the neutralizing capacity of these participants prior to the Delta wave. And so what we did is we selected a subset of these individuals, about 800 of them. Some of them had, well, firstly, none of them had prior infection. Some of them had been infected, and some of them had never been infected. The ones that were infected only had one prior infection. And remember, this is now determined by intensive PCR sampling, and we didn't actually bias according to symptoms. There was an attack rate of 34%, and here's the breakdown for the naive and for the infected individuals. This is obviously in the Delta wave. All right, so what were the titers? We wanted to know what were the titers here just prior to the Delta wave. And then we also wanted to know if these individuals were infected, at any of these points here, where was the peak and what was the waning from the peak time point until blood draw 5, and how did that impact protection from infection? Okay. Ooh, the resolution is not great on this. But here's the data. Okay, so here we've got neutralizing antibody titer on the vertical axis, and then we're showing the titer at the peak response for each individual and at blood draw 5. And you can see consistently there is, for most individuals, significant waning. So at the peak response, we had a GMT of 125, but at blood draw 5, we had a 1.4-fold decrease in titer to a GMT of 85. And interestingly, 45% of the people who had previous infection had no detectable neutralizing antibodies. So kind of sad, but interesting. And you'll see how that plays into working out correlates of protection. All right, so here now what we've done is we've broken them up. So here in this gray shading are all the people who didn't have any detectable serum neutralizing antibodies. And then here we've just showed you the neutralizing titers and their distribution in the people who did have a detectable response. And so if you compare those who didn't get infected versus those who did get infected, you see a significant difference in the GMTs, with, thankfully, the people who didn't get infected having higher antibody titers. And we saw the same thing at blood draw 5. Sorry, this was at peak. And we saw the same thing at blood draw 5, although because of waning, the overall titers were much lower. So here, because of waning, the titers in the not-infected group were 97, and in the infected group, 44. So still significantly higher, but much lower than at the peak of the response. All right, so how did we then use this information to calculate a correlate of protection? So we did a mediation analysis. And what a mediation analysis is it's simply looking at, if you have an exposure, how is the outcome affected by a potential mediator? And in this case, the mediator is the neutralizing antibody. We used a causal inference framework, which just simply put is, if you see an association and you run it through this mathematical model, you'll be able to know that that association is the actual cause. It won't just be, oh, yes, there's an association. Don't ask me specifics about the maths because I'm not a mathematician. You'll have to trust me or read the paper. So in this causal inference framework, we wanted to calculate SARS-CoV-2 transmission probabilities. And the probabilities were basically the risk of acquiring SARS-CoV-2 either from the general community or from within the household, because remember, this was a household transmission study. These probabilities were then dependent by a couple of different co-founders, which were obviously the individual's prior infection history, the mediator, which was pre-existing NAB conditions in this case, and then age, sex, and comorbidities. Those were the things that we factored into this model. And so using this model, well, Sankey did all the fancy maths. We found that firstly, immunity conferred by prior infection mediated 61% protection from reinfection, which is actually really nice, especially, well, against the Delta variant. But remember, the antibodies at peak showed a 1.4-fold drop to blood draw 5, which was just prior to the Delta wave. And so because of this, we calculated that antibodies only mediated 37% protection against reinfection. And so there's other effects, potentially, that are mediating the rest of that protection from infection that we've seen. Kind of sad, but maybe there's some reasons for why we've seen that. Then I'm not going to go too much into the explanation for Omicron just because of time, but we did the same analysis for Omicron. And instead of looking at peak versus blood draw 5, we looked at neutralizing antibodies against D614G and neutralizing antibodies against Omicron itself. And we calculated the difference between those two to estimate the effect of immune escape of Omicron. And so using that, likewise, we also saw a 61% protection from reinfection conferred by prior infection for the Omicron wave. But there was a much more significant drop in titer, and this is because of immune escape. So we had a fourfold drop in titer against Omicron. It means that any antibodies that were elicited because of prior infections, there was not that much protection because of the immune escape. And because of this, so there was 37% protection from infection mediated by serum neutralizing antibodies against Delta. But because of this, in Omicron, we only saw 11% mediated by serum neutralizing antibodies. And then the rest, of course, mediated by other effects. And so in summary, on this part of, the last part of this talk, the relationship between antibodies, neutralizing antibodies, and protection obviously depends on the degree of immune escape of the variant that you're looking at, where with Delta, which was a relatively similar strain to the infecting variants or the beta variants that these people were exposed to, there was a much higher level of protection than with Omicron. And then just to quantify it, there was only 37% total protection accounted for by serum neutralizing antibodies against the Delta variant, and even less, only 11% against Omicron. And yeah, it's sad, but the majority of the immune protection from prior infection that we saw could not be accounted for by serum neutralizing antibodies. And it is sad, but I think there's things we can do to figure this out. I think mucosal immunity is an important one. So if you're having a reinfection of a mucosal virus, your antibodies might hone more quickly to the site of infection rather than going into the blood, which is a waste of effort. Obviously, there might be other things playing a role, like T cells. There might be non-neutralizing antibodies playing a role in mediating protection. And those are things that we can actually investigate. And we're currently doing a mucosal antibody investigation, correlates of protection investigation with the same cohort. So hopefully that shows something interesting. And that is at least a partial explanation for why we didn't see as high levels of protection from antibodies as we would have expected. And then I just summarized some key lessons, one key lesson from each of the studies that maybe could be applicable to other viruses and other pathogens and parasites. Firstly, especially in sub-Saharan Africa, I think keeping in mind the impact of HIV in all the things we look at is kind of key. And what we saw in our study in any case was that these people may actually display greater intra-host diversification when they're co-infected with other pathogens, which could be a bad thing, but it also, in this study in any case, looks like it actually was a good thing. It made those people have a better quality of response, and in part maybe it even contributed to why we didn't see as drastic an effect on people living with HIV as we expected there to be. And then from the second part of the talk, exposure to antigenically different versions of the same pathogen can create these massive immunity gaps that then continue to drive the evolution of whatever parasite it might be. And then from the last part of this talk, I know we put a lot of weight into serum and studies with serum, but they might not be representative of what's actually happening if the site of infection differs. And so with that, thank you for your attention. And I'd like to thank our colleagues here at the NIH, specifically Cecile, Sankey, and Eli, our colleagues at the Human Sciences Research Council, Sizulu and Lakeness, and then our colleagues at the Center for Respiratory Diseases and our director, Adrian Purin, and all the individuals who were involved in the first C household transmission study, from the field teams to the clinicians to the lab staff, and of course the many, many funders. And finally, I'd like to thank everyone in my lab, the Antibody Immunity Research Group over here, specifically Donald, Hajira, Mpo, Tanusha, Sharon, and Kaniso, who contributed to lots of the data that I showed you, and my boss, Penny Moore, who has really set up an amazing laboratory for growth and raising of the next generation. And thank you to Peter.
All right, thanks, Janelle. So, I'm actually, I'll kick off the Q&A section. I was, your beta data, you know how the reason Irene showed some data where we're not neutralizing, but we have some beta sort of cross-reactivity that we are seeing here, even though beta didn't really show up in Ghana. And the only reason we did that work was because we were having challenges isolating ETA, and basically she used the viruses that we were able to isolate. And so my question is, because alpha, for instance, never picked up in South Africa, it was beta. Did you ever do those cross-neutralizing assays, and can you comment on that?
We didn't do it, obviously, in this population study because it's just way too many samples, but we did do it in the hospitalized cohort. And there's cross-neutralization of alpha and gamma, which were all similar and emerging at the same time. But I think from Irene's talk, what she ended off saying was that a beta immunogen might actually elicit a more cross-reactive response, which I think this shows very conclusively. And we actually did a study with Sanofi where they used beta as a boost. And the beta-boosted individuals developed more cross-reactive responses than the D614G beta-boosted individuals.
I think I had another question, but I'll hold off. I see that Dominic has a brief question.
Yeah, that was a very great talk. I'm not a virologist, but I kind of liked the SARS-CoV work. I know that HIV normally moves with TB. I don't know the prevalence in South Africa. And I also know that the immune system, there are some that are generalized. Could you comment on probably, did you check if some of them had TB, and what could be the contribution? Because I realize in your, is it the third distance, it's not conclusive as to where some of the responses are coming from. Yeah.
So pretty much everyone who's immunocompromised in South Africa has TB. So all those individuals who had CD4 count less than 200 likely had TB, but we don't actually have that clinical data.
Okay. Chinedu.
Thanks, Janelle. I have two questions. One, do you think you were doing the blood draw for that at day 5, but the Omicron you did at day 8? So do you think the length of time may have affected the protection, the reduction in the protection for Omicron compared to Delta, rather than just the virus structure? Then secondly, do you also consider immune imprinting from the white D614
yes.
to either Delta or Omicron? So the biases to do, which is why you are having the reduction in fold.
Yeah, so probably if I explained the Omicron part better, this would have made sense. But for both, so how we did it with Delta, we selected individuals who had only one prior infection. And we did the same thing for Omicron, only one prior infection. For both groups, that prior infection was either D614G or beta. We didn't choose Delta individuals for the Omicron. The difference between blood draw 5 and blood draw 8 definitely would have had an impact, but they're actually not they're months apart. But how we try to compensate for that is we try to look at the Omicron-specific antibodies. So we tested for blood draw 8, we tested against D614G and Omicron. We said, okay, what are the titers against D614G minus the titers against Omicron? And we got this Delta NAB, which is basically all the antibodies that are useless against Omicron. That's how we try to compensate for that immune escape. So for imprinting as well, you can't 100% cover for it because we had mixed first infections. We didn't only just have D614G or beta, but that was kind of as best as we could do.
Any other questions? All right. Thanks, Janelle. And if you think of any other questions, you see her after. Oh, Janelle didn't tell you how she's related to this next speaker, so I'll let her come and tell you.
It gives me great pleasure to introduce my husband, who will be the next speaker, but Peter can properly introduce.
So she won the fight to come to Ghana. Anyway, I'm not going to introduce Kurt. Dr. Dominic Amuzo and Dr. Abdurrahman Kamara, who will be the chairs for the next session, will do that.
Good afternoon once again. It's always a pleasure to be in WACCBIP. It's my second chairing at a WACCBIP conference, so I'm excited as an alumni. It shows that I'm growing. For this afternoon's session, our next talk will be given by a virologist. We are fortunate to have a virologist from South Africa in the person of Dr. Konstantinos Kurt Webma.
Can I go ahead and share screen, I guess, then?
Yes. Can you go ahead?
All right. Do we have that visual there? Can people see slides?
Yeah, go ahead. We can see your slides.
Great, thank you. So I guess I used to be a virologist. I'm more of a structural biologist, really. Biology was my early introduction when I was a grad student. But yeah, thanks very much for letting me talk at this meeting. It's a real pity that I couldn't be there, but as Peter said, Jin won the fight. So that was it. And ideal is ideal. And really, I guess the focus of this talk is going to be the main focus of our lab, which is developing next generation antivenoms for snakebite therapy. So I go by the name Kurt from Johannesburg, South Africa, and this will be the talk. So often when I tell people that I work on snakebite, the first question I get from most of them is why snakebite? And the answer that I give to most of them is because snakebite is the biggest public health crisis that they've never heard of. So it's really astounding. More than 150,000 people die from snakebite every year. 4 or 5 times as many people lose a limb, like a hand or a foot or an eye. And those are insane numbers. Those equate to 1 person dying from snakebite every 5 minutes. So it's really a massive problem. And unfortunately, it's a problem that affects family and community providers. So when 1 person is bitten, it affects an entire community when they're no longer able to produce. It's also a neglected tropical disease, possibly the most neglected of the WHO's NTDs. And that means that it's a disease that infects impoverished and rural communities, but doesn't really receive a lot of research funding or research in general. But that's thankfully changing in the last few years. So just to say that current antivenoms are incredibly effective. The antivenoms that we give today will save your life, but they're based on a really ancient technology, more than 100 years old. Essentially what we're doing is we're giving snakebite victims animal blood or plasma from animal blood, from hyperimmunized horses usually. So this is obviously a very impure medicine. Only 10 or 20% of the antibodies in these antivenom vials are actually on target for snake toxins. There's massive batch-to-batch variability. It's a very unsafe medicine because it's mostly animal protein, so you have to give it in a hospital because people go into massive allergic reactions and can die from the antivenom. It's a temperature labile product, and it's very expensive. So people often have to sell their land, their animals, their houses, just to be able to pay for some antivenom medicine. This is obviously unacceptable, and we believe that the solution is in making next-generation antivenom monoclonal antibodies. So these are things we can make in the lab. They're 100% pure, they're 100% on target, they're reproducible, they're safe, we can humanize them, and we can make them more potent, more broad, and more stable with structural biology insights after we've discovered these antibodies. And if we do this right, they're also going to be a hell of a lot cheaper than current antivenoms. That all being said, venom is an incredibly complex drug target. So when a snake bites you, it injects more than 50 different toxins in their isoforms. So that's quite a concoction. But we can generally group these snake toxins into 4 major toxin classes, really. And so the idea is to get a broad and potent monoclonal antibody, maybe 1 or 2, against each of these 4 major toxin classes, and then combine them into various tri-specifics, and then from that make a cocktail of monoclonals that would be our next generation antivenom. And so for the purposes of this talk, I'm going to focus on some neutralizing monoclonals that we've developed, really broad and potent antibodies against one of these major venom classes, and that's the 3-finger neurotoxins, which are in fact the deadliest. These are the kind of toxins that kill you within an hour or 2. And so the first thing we did was set up a rapid 14-day monoclonal antibody isolation pipeline from horses. So we just start with the horse blood, fluorescently labeled proteins. We pass these through the single-cell sorter, get our cDNA, amplify up heavy and light chain genes for the antibodies. And then we have a high-throughput gel extraction, cloning, protein expression workflow that culminates in ELISA data that tells us whether the monoclonal antibodies we isolated bind to the sorting antigens or not. And this is our first sort. So we started with a long-chain neurotoxin from black mamba and a calcium channel inhibitor from the same venom. And we used it to sort, it's about 32 antibodies, and 8 of these that we've circled in purple, if you're familiar with sort plots, these were antibodies that were actually toxin-specific, so they bound to the snake toxin. And these are the ELISA data showing that, so again, strong signal correlating with strong MAB concentration. And 5 of the 8 antibodies bound to the long-chain neurotoxin, and the other 3 bound to the calcium channel inhibitor. So in this particular instance, there was no cross-reactivity between the two, but we at least knew that we had antibodies that were reactive for snake toxins, so that was the first hurdle achieved. And now the next question was, okay, we have an antibody, it binds one toxin, how broad is it? Does it broadly bind some certain subclasses of toxins? And the first thing we wanted to assess were these major long-chain alpha-neurotoxins. And so we did a panel of alpha-neurotoxins in binding to our antibodies. These toxins come from mambas, non-spitting African cobras, spitting African cobras, and some cobras and crates from other places in the world. And this is kind of a heat map summarizing those ELISA data. And as you can see, with all 8 of the antibodies that we isolated, we actually got complete coverage in binding of all of these long-chain neurotoxins. For comparison, here there's a 2554 antibody that was isolated by another lab somewhere else in the world. And you can see our antibodies are actually really, really broad, like really broad binding for 1H, but also exceptional binding from some of these other antibodies. And that really got us excited because we're like, okay, now we have something that's really broadly binding to long-chain neurotoxins. And we thought, hell, why not go and test it against short-chain alpha-neurotoxins, even though we didn't have those kind of toxins in the sort strategy. And these data is when our mind really started to get blown, because this was incredible. As you can see, we've got broad binding with this antibody down here, number 4E, but really, really exceptional broad binding by these 2 antibodies, antibodies 3A and 3D, across both long-chain alpha-neurotoxins and short-chain alpha-neurotoxins, which are these 2 major subclasses of 3-finger neurotoxins. And in fact, there's just 29% sequence identity between these 2 different subclasses. So really incredible breadth of binding across diverse toxins. but binding isn't the end of the world. We need to kind of show that these antibodies are also neutralizing. And so we sent them to collaborators up in Liverpool who developed this in vitro neutralization assay for testing neutralization of neurotoxic snake toxins. So it's a cell culture assay. There are these fluorescent calcium ions that go through the N-acetylcholine receptor into the cell, and you get a fluorescent signal. If the receptors are blocked by snake toxins, obviously there's no signal. And then if the snake toxins are in fact neutralized by, say, an antivenom or monoclonal, then you get restoration of the signal, the fluorescent signal. And these are our data. So these curves you can see really high restoration of signal, really potent neutralization of both long and short chain neurotoxins, really confirming that we'd isolated a pair of both really broad and also really potent neutralizing monoclonal antivenoms. So being a structural biology lab, we wanted to know how this was possible. How can these antibodies be so super broad and super potent? And so we solved the crystal structure of antibody 3A bound to the short chain alpha neurotoxin from black mamba, and antibody 3D bound to a consensus short chain alpha neurotoxin. And so these two antibodies are somatically related. So as you would expect, they come in with the exact same angle of attack. They come in and hit those toxins between fingers 2 and fingers 3 over here, and this cleft over here and over here. But there were some differences in how they did this. And so if we zoom in on the interaction, now showing the CDRs of these antibodies as these kind of cartoon spaghetti loops, and then we have the toxin in surface view in the back, and I've highlighted finger 3 in green. And if you look at CDRH2, it comes in with this long extension over here that reaches around and really grips onto finger 3 of the snake toxin. And that's how 3A does it. But 3D doesn't have that insertion. And instead, it has an insertion in CDRH1 that kind of reaches in and touches in the same kind of way. So two different mechanisms that these two related antibodies acquired over somatic mutation to gain increased interaction with snake toxin and increased affinities. We then wanted to understand how the toxins are so super potent. And so we looked at cryo-EM structures of how short chain alpha neurotoxins bind to the N-acetylcholine receptor. So this is a cryo-EM. The N-acetylcholine receptor is a pentamer, which I'm showing in various shades of blue. And in fact, maybe if we rotate that around 90 degrees, you can see the toxin comes in and binds between a dark blue and a cyan, so between two different protomers. And then if we superimpose our antibodies onto these structures, you can see that the antibody completely sterically occludes the ability of this toxin to bind the N-acetylcholine receptor. So there's just no way that the antibody and the receptor can bind at the same time. And complete steric occlusion is a really strong way of getting potent neutralization. So that kind of explained that. If we focused in on the interaction between the receptor and the toxin, you can see that from this cyan protein over here, there's a contribution of what's called loop C that comes in and binds between finger 1 and finger 2 into this teal part of the epitope or binding site. And two antibodies recently isolated by other groups in the US are really broad and potent against only long chain neurotoxins. Ours is the first that has this breadth of activity between long and short. And both of those antibodies bind between finger 1 and finger 2 and in fact mimic this loop C interaction. On the other side of the toxin, from this dark blue protein, there's another interaction that comes from loop F that binds in between finger 2 and finger 3. And as I mentioned, that almost 100% overlaps with the epitopes for our antibodies. It's almost completely the same amino acids in the same way that are recognized, which really kind of explains the breadth between both long and short chain neurotoxins from our antibodies. And so in summary for this part of the talk, just to say we isolated a related pair of really broad, really potent, really super awesome antibodies that can neutralize both long and short chain cleaving neurotoxins from snake venom. And we identified a novel epitope for binding and neutralizing short chain neurotoxins. Going forward, structural insights to see if we can improve breadth and potency, specifically exploiting this loop C site. And of course, we're isolating more and more antibodies to some of the other major toxin classes. And that would be the end of it. But since this is my first WACCBIP talk, I kind of wanted to just harp on the fact that we are primarily a structural biology lab. And so while 80% of the work that we do is antivenom work, the other 20% is really kind of collaborations and working with other groups to provide some kind of structural support. So these are data I generated when I was at postdoc at Scripps, kind of showing how we did structures for strain-specific neutralizing antibodies bound to V1, V2 from HIV-1, and then there were structures of broad neutralizing antibodies, and we were able to use angles of approach to kind of define a strain-specific neutralizing face and a broadly neutralizing face, which is important for future efforts for vaccine design, in this case of HIV-1. We've also more recently been collaborating with ARU, where Jin works, and the Antanasievich lab in Switzerland to implement cryo-EM workflows in our lab. So these are cryo-EM data, antibodies isolated by Jin's team and Penny's team at ARU that have really broad cross-reactivity against SARS-CoV viruses. And we show with cryo-EM that they come in at this kind of very vertical angle of approach at RBD, bind to a very conserved epitope, and really warp the whole spike to get in there and link the proteins together. So kind of a cool explanation for their breadth and potency. We're really passionate about structural biology, so this is the current workflow that we're setting up. We've just acquired a mosquito. We have an imager, we're getting a hotel and a scorpion flying in very soon. So robotic automated workflows for setting up and developing crystals, and then we send them to a particle accelerator, get the data, it comes back, and we solve the structures. We're also in the process of implementing a cryo-EM workflow, so getting the money for that and trying to set that up. And as I said, really passionate about trying to work on African problems on continent and develop a community of structural biologists. So if you're somebody who's interested in structural biology or want to work with a structural biologist, please don't hesitate to reach out. And I'm going quickly to make it in 15 minutes. So here are my acknowledgements. Poppy Mashilo was an amazing workhorse back when I just started the lab. She did a lot of the work in getting the toxin expression set up and helping to get the MAB pipeline set up. That MAB pipeline was funded by the Morris Animal Foundation. Then currently in the lab, so this is my lab over here, PhD student Francis Ayres and MSc student Carla Hutton have both done an incredible amount of work on the ELISAs and some of the structural work for the 383D paper. Our collaborators in Liverpool for the in vitro neutralization assay, Diamond Light Source, where we collect structural data, ARU and Tanasievich and Wilson Labs, because I presented some of that SARS-CoV-2 and HIV data. I was a Flair Fellow at the beginning of this work, so thanks to these guys, Royal Society, AAS, to the NIH that funded some of the structural work, and now to WellCome, who've agreed to fund the antivenom discovery project for the next 8 years. And that's a picture of me with a very beautiful Cape Cobra. And since I still have about a minute on my time from when I actually started timing myself, because I was waiting for my slides, happy birthday, and thank you very much.
That was a nice talk, and he actually gained a lot of time for questions. So at this point, we welcome questions from anyone. I think I will start. So first of all, that is a very interesting work you're doing. Part of the training in WACCBIP was to develop a comprehensive proposal. And I remember I tried to get peptides against, I think it was hepatitis B virus. And the goal was to look for toxins or peptides in venoms that were available in scorpion. So I just wanted to know, now that you are working on snake venom, how similar are the peptides in the snake venom to that of the scorpion in relation to what you are working on?
So they're very different. The targets are very similar. I think there's kind of this convergent evolution towards the same kind of targets, like calcium channels and neural targets and stuff, developing and destroying muscle tissues and things. But when you look between like invertebrates and reptiles, the way that they get there is very different. It's interesting, if you look at snakes, you may find that the same kind of toxin, even something that's related to like a pancreatic enzyme, evolved multiple times. And so in the same, two different, completely different snake lineages, you'll have the same protein structure come up doing the same thing. But when it comes to comparing scorpions and snakes, unfortunately, that's not the case. They've sourced their toxins from very different host genes to turn them into venom toxins.
Thank you. Next, Chinedu, Dr. Chinedu.
Yeah, excuse me, sorry, there is an announcement. For those who have their cars parked in front of the building, can you please move them? And yes. to set up for dinner, and also for just to remind all the faculty to keep scoring the talks. Yeah, thank you.
Thanks, Scott, for the wonderful presentation. I always admire what you do, anyway. Just that Gina always win all the time. The vaccination in horse. So 2 questions. Is it only horse? Why horse to get the antibody? And is it only horse that you can use to do your vaccination? And did you give, except if I mix it, did you give the horse antivenom, or you gave them an immunogen? Did you give the horse a venom and you gave them an immunogen that you then isolated the antibody?
Yeah, so sorry, I had to speak really quickly to make sure I got to that final happy birthday slide in time. But just to say that we're using horses from the existing antivenom production unit in South Africa. So it's the only place that makes antivenom. And so they use horses. Traditionally, horses are most commonly used because they're big animals. So you can give them a fair amount of venom and then get a fair amount of blood out of them. Sheep are also used in some of these antivenom programs, essentially hyperimmunize with whole venom from snakes, and the animals develop a polyclonal antibody response that you can then pull from the plasma. There are groups that are looking at novel immunization models now. So there are some people looking at llamas to try and make nanobodies. There's some people that are looking at humanized cows, again, because it's a big mammal. And those are the kinds of studies where we can start to look at giving engineered toxins as opposed to just raw whole venom. But in the case of isolating these antibodies, these horses have been hyperimmunized for years. And somatic hypermutation has been taking over, making these antibodies into super antibodies. And so that's really why that's where we focused our interests.
Okay, from Dr. Amwa. Yeah.
Yeah, very nice and interesting talk. So my question is, the venoms, I'm wondering, and also the antivenoms, are these mainly targeting the neurotransmitters? Are they targeting the postsynaptic receptors, presynaptic terminal? And what is the severity in any of these if there are differences? Thank you.
So specifically the venom toxins, right? We're talking about here from different snakes. So the antibodies that we've isolated are specific to toxins that are neurotoxins that bind to N-acetylcholine receptors at that synaptic junction. There are other types of toxins in snake venom that do other things. So there are phospholipases that just rip membranes apart. There are 3-finger toxins that inhibit other channels, but that also contribute to cytotoxicity. There are proteases that activate or deactivate the coagulation cascade. So those are the kind of things that fit into those 4 major toxin classes that I mentioned. And so 3-finger toxins that are neurotoxins, specifically that target the N-acetylcholine receptor, fall into one of those 4 major classes and make up a major component where there are neurotoxins or cytotoxins from those 3-finger toxins. So very complex venom is, and that is the one focal point that our antibodies are able to bind and neutralize to. But obviously we have to get more monoclonals still that can bind and neutralize the other major toxin fractions before we have something that can completely replace current antivenoms.
Okay, we take another question from Dr. Kwashi.
No, mine is just a comment to say great talk and thanks for the birthday wishes. It's so unfortunate that she won and you lost, but
it's not that unfortunate, though. Right?
I mean, hopefully, the Epsilon goes through, and we'll see you. So next time you bring AB with you, and we'll have a big jolly party.
Yeah, it'd be fantastic. Thank you very much for the opportunity. I really appreciate it.
Bye bye.
Shall we give him a round of applause? Okay. It's not often that you have a husband give a talk right after. Yeah, so on the birthday of a deputy director. So I think we are really honored to be here. Yeah.
Okay, that was interesting. So please let's move on to our fellow session. So our first speaker is Olatu Comfort-Tosin. So he's presenting on antibacterial potential and phytochemical constituent of C. diffulosa rest. Olatu, please. Is it in person? Is it online? So I don't know. Is it online, please? Okay.
In the meantime, I'll reiterate the announcement. If you park your car at the forecourt of the department, you have to move it away so they can prepare for the closing and awards night.
So we have a late break-in presentation that was not on the schedule, and that's DJ Perkins is going to talk to us about some of the new data they have.
Thank you very much. It's a pleasure to be here today. I've been coming to the conference for the last 8 years, except for a few during COVID when we couldn't travel. And I got here and I had the dates incorrect, my fault, extended the trip, and then I was talking to my friend Peter yesterday, and there was an individual that wasn't going to be able to give the presentation, so I put this together this morning. And that's not an excuse. I think it'll go away. But just really quickly, I want to thank Gordon. We are always praising Gordon. He's done a wonderful job, and I think without that N=1, this wouldn't have started. But I also think that it's really important to acknowledge that he has an amazing team of individuals. One of the challenges we have in Kenya, I've been there for 25 years, is we don't have this epicenter. We don't have this mass of faculty that are intelligent. There are smart people, but they don't have this mass that we can build upon. And so I think it's an amazing testament to all of you. We watch the conference grow each year. We see more money come in, which is very important. And we see high quality work come out. And the science that you all do here is world class. So I applaud you for that, and give yourselves a round of applause. So I've been working on severe malarial anemia for about 27 or 28 years now. And I can tell you in a sincere way, you could write on the head of a match what I know about malarial anemia, okay? Malaria in general is very complex. Malarial anemia is no more complex. But the point is, these diseases that we see that we call something monosyllabic like malaria are highly variable and different around the globe. So let me just jump into some of the stuff that we've done in Kenya. This is, I think, and this presentation that I'm giving today, I hope that the senior members and the faculty enjoy it, but I really want to give this as an example of how to use some omics approaches to try to solve problems. So we'll talk about malarial anemia, but this really applies to anything. And so there is the targeted approach you can use, and that's when you think you know what's going on, or you can just go fishing with really big nets and see what you pull out. So when we think about severe malaria, this is the most important slide in there that none of us normally pay attention to. If you look at the severe disease sequelae there, okay, and then we work backwards and we say, what are the genes and gene pathways that actually condition these clinical phenotypes? There aren't any, which is sincere, because those phenotypes are so different. So one of the challenges that has based malaria is the genes and gene pathways that condition the development of cerebral malaria are going to be very different than severe malarial anemia. So in some ways we're lucky. The region that I live in, we don't have cerebral malaria. It's a really just one phenotype, which is malarial anemia. So this really illustrates the point. I don't need to go through all the details, but I think it's important to realize that where one is born, how frequently you get hit with that thing called malaria, and how often you get hit, literally conditions the clinical phenotype that you see. So in a high transmission region, you're going to get severe malarial anemia. It's going to be at a very young age. When you go into intermediate areas, you're going to have a little bit of an older distribution, an admixture between cerebral malaria, sometimes severe malarial anemia. And then if you go into those really immune naive populations, and I think some parts of Southeast Asia represent that. So when we go in, someone's doing logging, not been exposed, you really don't see very much anemia at all, rather you see cerebral malaria. So when we first get hit, it's related to some of the epigenetics, certainly that we had this beautiful talk about this morning. No one knows what causes that, but there's clearly something that's conditioning that's going on that really shapes our immune responses and then shapes them across time. So we primarily work on innate immunity, and the reason why is because in a high transmission region, it's these children that are getting sick. They've not yet developed naturally acquired immunity, and so we've really been focused on what makes individuals really sick, and can we parse that out and find networks that are protective versus pathogenic, and then try to find therapeutic targets to make an impact on that. So severe malarial anemia, for those of you that don't work in those regions or don't know much about it, about a third of the individuals, right, who show up at hospital have a really severe problem. And the reason that is, and it's really funny, it's human dynamics. So when you see some overt clinical sequelae, mom takes the child to the hospital. When the child gets febrile, listless, stops feeding, mom waits, caregivers wait, and so by the time these kids show up at the hospital, they're pretty far advanced in disease. And so the one thing I'll just say, I'm sorry, my pointer's not working, which is no big deal at all. I don't think so. There it goes. When we think of infectious diseases, we always think about the fact that it's the bioburden, the pathogen burden that's conditioning this. Severe malarial anemia is quite different. So the children with the really low levels of parasitemia, it's there and you can see it, but it's nowhere near as high as it is in children that have milder forms of disease. And so the reason for that is most likely related to when we first see this insult. Like all infectious diseases, it's our immune system and how we respond to that insult that really conditions and influences our pathogenesis. And so this may accidentally be animated. Let's see. No, it's not animated. Well, I guess it is. It's missing one. The animation didn't work. So these are the platforms we use. So you can look at DNA variation. There's a lot of ways to do that. The immunochip is up there. GWAS is now sequencing. You can look at transcriptomics. There's all kinds of different methodologies that you can look at transcriptomics. What I'm going to talk about today is transcriptomics using a 3' ligation approach. So what you end up getting out of this kind of whole blood transcriptome are expressed RNAs or mRNA, okay? And then we've mixed in a proteomic platform across time. Normally that's done with mass spec. There's a really cool technology that's pretty new. It's an aptamer-based technology. I'm not putting a plug in for the company, but it's really cool. The price is good, and you get around some of the challenges of mass spec. It's actually DNA aptamer-based. And so a magnitude of scale for those of you that are interested in proteomics, the company is called SomaLogic. And you can get currently 7,000 different proteins in plasma or serum where you could create a cell lysate for about $700. And it's a really, really good, robust technology with good bioinformatics support. So I'll stop. But those of you that are interested in proteomics, it's an interesting platform. You should check it out. And then you can follow up, of course, and do all kinds of more functional assays, either with multiplexes or some specific in vitro experiments related to finding your particular mechanism. So let's step back out of the box and say that we really don't know anything and stratify kids into severe versus non-severe disease. So when you have large groups of children that you enroll, this, what I'm going to talk about today is cross-sectional. We have some longitudinal stuff, but there's not time to go into it. The point being, you recruit a lot of children, and then you get the ability, because you have these things biobanked, so then you can go in and say, I want to match on age, I want to match on X, Y, or Z. And so you have a better matching strategy when you have a bigger catchment. So what I'll talk about today is children with non-severe malarial anemia, 41 of those individuals, and then individuals with severe malarial anemia, which is 25. And so don't stress about whether you know this or not, whether it's simple or complex. Just think about the strategy. So it's really a case comparison group between those two. And so if you look in the upper left there, the only reason I show that silly Venn diagram is because, as you can see, when you're a non-SMA versus SMA, we're not going to drill into this in great detail today, but say you wanted to find a therapeutic target. Are there common things that are expressed, and are they a different level, or are there unique things that are expressed? So you can identify those genes in those clusters, the 602, the 493, and then you can go and make networks for those. And those are oftentimes really robust for drug discovery when you want to find a target. And then the Venn diagram, just to orient everyone, you've seen very many of those. I only put that up there. This is from a recent paper that we just had in Nature Communications. There's actually no magic. We get kind of sucked into these mathematical games and importance of fold change and whether or not it's significant. And so I'm not saying it's not important. Rather, what I'm saying is that when you start to find signals, you set a really stringent threshold. And so p-adjusted values and more than 20,000 genes. And then as you can see there were 279 that were downregulated. And then 1,403 that were upregulated. So the only reason I say that is that's a starting point. You could focus on that and make pathways and networks from that, but you can definitely broaden the catchment to get additional information. And the one thing I want to say to people, these numbers change very much when you include things that occur in a natural setting. So for example, you have severe malarial anemia, and then all of you are familiar with sickle cell anemia or sickle cell disease. This analysis excludes those children, okay, to try to get at, if I don't have this other issue with a hemoglobinopathy that's driving my gene expression or suppressing it, those kids are removed. We have analyses with it. I just want to make that clear to everyone of what the comparison is. And so I mentioned this yesterday or the day before. It's been a long week, so thanks for everyone's attention. There's a deconvolution analysis you can get. So when you do your sequencing, there's something called CyberSort, okay, and it has it's a 22 matrix file, and it's essentially leukocytic profiles. And we talked very briefly about that. I just wanted to put that in here to show the crowd that this is a technology. So when you get, in this context, your entire express transcriptome for zero money, you can go in and do CyberSort. And so there's 20 different cellular phenotypes, and those cellular phenotypes are defined by a lot of bioinformatic work that's been done by people that says, if I'm a memory B cell, I have this repertoire of genes. If I'm this other particular type of immune cell, for which there's 22 that have been characterized with this robust panel, I then have this expression versus that expression in case versus control. And so now that you all are somewhat on the same page, this is just the heat map looking at non-SMA versus SMA. It's a little off on the side there, I apologize, but as you can see, the asterisks indicate significant differences between SMA and non-SMA. And so if you look over here, in the SMA group, and I won't go into all of this, we don't have time, it's a 10 or 15 minute talk. The point is, B cells naive, okay? Those are higher in individuals that are getting severe disease. So this is nothing more than not having to spend the money to do the flow cytometry, just getting a molecular look at some particular phenotypes that exist. And then neutrophils and NK activation is a big one. That's over on the far right that you can't see there. So then what one can do, be it malarial anemia or any disease you're looking at in a case control fashion, and remember almost all of these bioinformatic platforms that exist are group A versus group B. There's different types of analyses for longitudinal measures across time. And so essentially what you see here in the upper panel is if we take those 1,600 genes and run it through enrichment networks. This is done with a program called Metacore. KEGG, no disrespect, use it, it's free, but the annotation of that is not as robust as some of these other platforms that you actually spend the money, about $10,000 a year for. But it's actually worth it, as silly as it sounds. So what it does is it goes through this enrichment analysis, and as you can see there, it's parsed into different types of immunological or cellular processes, okay? So the left panels there are nothing more than the top 10 ranked. The panel on the top left there versus the panel on the bottom right, I just threw that in there for the crowd, for the students. Oftentimes you want to validate your work, and it doesn't necessarily, there's not another cohort. There was a cohort in, actually in Uganda, some work by Chandy John et al., and then as you all know, one publishes this, and you got to put it into an international archive so people have access. So we got access to that. And so the reason one would do that is to look at generalizability. So what pathways are important in this population with a particular genetic background, what's important in the other, is their generalizability. And the generalizability that really converged here was something on hypoxia-inducible factor 1, or HIF1, and how we respond to oxidative stress. And that's what's shown over here. And of course, we won't go through this complex network, but the point is, if you look at the thermometers are on there, okay? Red is upregulated, blue is downregulated. So all that does is you get this pictogram and you say, here are different genes within the network that are up and down. What's the convergence across different populations? And then you can begin to comb through those for therapeutic targets. So generalized theme, response to oxidative stress is important. We need to appropriately manage these erythropoietic insults. And so that's why the HIF pathway is listed there. Sorry. And so I'll wrap up here pretty quick, but if you look in that upper left quadrant there, right, that's essentially saying if I take the proteome and I take the transcriptome, do they agree? And I just want to throw something out there for you guys. Everyone is going to tell you they need to have a high level of concordance. It's not even common sense, okay? When you concomitantly measure a transcript and a protein based on the timing and the dynamics of expression versus turning in with post-translational, post-transcriptional modifications, they're going to be different. So the point really is, if you want to do it properly, you have to dissociate in time, but you can do it concomitantly and look at common signals. So if you were looking to find a particular drug that targeted a gene/protein target, what you would do is you'd look for similar directionality between the two concomitantly if you didn't have the ability to do longitudinal studies or longitudinal samples. So when you get agreement, be it up or down, that's one thing you want to do. So what's shown up here right now is, again, that HIF1 target pathway, which was top emergent, and it'll show you, if you look at the arrows, they're the same color and the same magnitude, that says that's a potential target within that pathway. Then what you do is you do some informatics, you use drug bank, different things, there's some platforms in here, and you say gene X at the transcriptional as well as the protein level is down. So then you could look for something that augments it and go through the pathway and find compounds. As you all know, it's very difficult to find compounds and make those compounds based on rationale. So one of the strategies that can be utilized in things like malaria is essentially finding FDA-approved drugs in pediatrics with a common mechanism of action, be it an agonist or an antagonist, and you can shortcut a lot of the other efforts that's done for the 15-year drug development pipeline. Why do that? Yeah, you can find drug targets. Another thing you can do is find biomarkers. This is a machine learning algorithm that we used, and so really what it does is it compares gene expression, protein abundance or protein expression, and it also incorporates into that parasite gene expression, i.e. falciparum, okay? So you can create these machine learning models to try to parse things into categories, and if you actually look at the number of molecules and were to count it there, you find that some platforms work better. Not surprisingly, proteomics, because it's the mature product that's bypassing some of the other modifications that can take place. Your human immune response is better at predicting your outcome than is gene expression in the parasite. And the other thing that's really interesting is the number to create that area under the curve for robustness is really low. What does that mean? That means instead of having to measure in some type of platform 100 molecules that would then composite into a biomarker, it converges on a relatively small number. So if you look at the panel there and you actually count them, what ends up happening is not so many molecules define very strongly with an area under the curve what the clinical phenotype is going to be. And the reason that's important is because those things can serve as biomarkers. And so we had very little time to talk about it. There's a lot of landscape there that's complex, the level of transmission. Co-infections, as you all know, are really prominent. This population that I showed you today were tested for HIV, bacteremia, a partridge in a pear tree, and everything you can in kids. And those kids that were found to have any other infections were excluded from the analysis. And so I think in this short period of time, you can see if you use some powerful tools and look at everything, You can make a checklist. It doesn't mean the checklist is correct, but it's sending you in the right direction. And the reason that that's important is this is a starting point. Then you can drill down and do molecular-based studies on specific pathways. And so this is more of a strategy than go work for the rest of your life on HIF because it's the most important thing in malaria. It just gets the highest priority rank, but there's many, many important pathways. Oops, I'm sorry. I'm left-handed. I'm trying to do this backwards here. This is the group. I just want to thank them. I thank you for all your attention, letting me get up here and pinch it for whomever it was. Wonderful to see everyone. Acknowledgements to all these awesome people. Our strongest level of acknowledgement goes out to the funding agencies, of course. And then I'd like to thank, of course, all the children across the last 25 years and their parents that have participated in the studies. Thank you.
Great. So you've noticed there is some change in the program. Do you have any question, please?
Yeah, thanks. Wonderful presentation as usual, DJ. So I'm wondering, I'm hoping to get into the infectious disease space, but you said something that early exposure and frequent exposure to malaria may correlate with severity. So I'm also wondering whether, for example, let's say placental malaria, let's say prenatal malaria exposure, for example, in the fetus, could that also contribute a bit? And you also alluded to the epigenome, for example, but I'm also wondering with the transcriptomics, is there also any interest in, let's say, the non-coding RNA space, for example, microRNAs and the like, which may contribute some of the discordance from transcriptome to the proteome? Thanks.
Yeah, there were a couple questions and I couldn't hear them all. One appeared to be related somehow to endemicity and malaria in pregnancy. Does mom getting malaria during pregnancy influence somehow the development in a child? Was that the question? Yes, and the severity. It depends on who you ask is the truth. And as with many things in malaria, all things are true. It depends on whether or not mom is healthy during pregnancy. And just because she gets malaria doesn't necessarily mean she's unhealthy. It depends on whether or not she has HIV. But clearly what happens, more so than, I believe, departure of immunity transplacentally, I think it has to do not only with malaria, HIV, anything. It's how healthy is mom when the child is in the in utero environment. And, I was talking to some folks earlier, and anyone that works on this, please don't be offended. We read in textbooks, mom's immunity is important. Of course it's important. It's highly important. Mom has transplacental antibodies that protect the child until about 6 months, and then some hemoglobin F wears off. I believe that occurs, but in a high transmission region, we see kids all the time, 1, 2, 3, 4 months old, profoundly ill with malaria and dying. So that's an answer to your first question. I think it depends on endemicity. It's never good when mom is sick and is carrying around a child. You always normally have untoward effects. And then the other question related to epigenetics, I think the answer to that question is it important? Yeah, certainly. It depends on the platform that you're using to measure, okay? And so there's all kinds of different things we can do. I believe that you first have to say which pathways does one want to focus on. And for 20 years, us and others have focused on this is important because this is what that cytokine does. If you just use really big bait and go catch as much fish as you can, things that you would not have expected will emerge. And I think that's a starting rather than an ending point, like I said. And then you can go into different molecular mechanisms or why, HIF emerges as the top. Well, number 2 on the list is reactive oxy-species generation. So it's all important. So deciphering which is most important, I don't think is straightforward because I think it's a model of convergence in which is the summation of all of those events that occurs.
Yeah.
Any other questions?
Yes, DJ.
Oh, hi, Alfred.
Yes, hi, my good friend. Yeah, thanks. Sorry, I missed part of your talk because I was having an interview.
There's no excuse for that. The quiz will be. We'll have the quiz over some Johnny Walker. Okay.
We'll talk about.
Don't stress.
We'll talk about it over whiskey. That said, I was just wondering, in a place like Gambia, the prevalence of malaria has reduced significantly. There's been a significant reduction in malaria. I think we're around, like in some places, less than 5%, less than 10 over 1,000 in a whole year in terms of incidence. So what we've seen in the last couple of years are more adults, not really young adults. We're talking about 20, 25, 30 with severe malaria. Actually, 2 years ago, the mortality was a little bit shocking to me because we had 53 cases in our cohort, recorded for us for recruitment in our work, and 27 of those died. And most of them died of AKI. Right. So I'm trying to correlate the issue of pre-exposure and the correlation with severity later on in life. I don't know how it seems as if it's going the opposite way, because most of these individuals were probably not exposed due to the lot of control that had been in the Gambia. I can't tell because they are 25 years, they're 20 years ago. Yeah. Well, the mechanisms that you've seen now and these pathways, are they still holding in these adults? Because most of what we know is severe malaria in children in high endemic areas.
No, I'm so glad you brought that up, Alfred. We didn't plan that, I promise. What I could hear, and something's buzzing in my ear here, what I understood is there a changing dynamics, and when you shift that dynamics and you sometimes see more or less mortality, are the molecular mechanisms the same? Was that the question? And so what we have witnessed in Western Kenya that's holo-endemic, we fortunately lose a lot less children. When we first went to the hospital 23 years ago, all-cause mortality, any child that walked through the door, the mortality was right at 22%. Any child that walked in, they mainly had malaria. Non-arrogantly, but with a great, robust team and the ability to intervene and have laboratory diagnosis, we reduced that to about 2.9% in terms of mortality. Now, disease, when we follow kids in the community, we have noticed a dramatic decrease in severe malarial anemia. Now remember, it's a moving target. There's lower numbers of cases, which makes our job more difficult. But we haven't seen the emergence of cerebral malaria. But I totally agree with you that as the dynamics change, as the pathophysiology will change. So if I formerly was in a high transmission area and I still live there and the transmission changes, it's going to take a while for my genetic repertoire to catch up with the historical changes that have taken place. And so, yeah, I think what we will see, I'm not sure I'll live long enough to see it. We'll see. But in Western Kenya, that was high transmission. When you get bed nets in, when you reduce it, the other big thing, Alfred and crowd, is polyphenic disease. So kids with severe malaria oftentimes have bacteremia, HIV positivity, HIV exposure. As the population gets healthier, your signal shows up in malaria, and you get less severe malaria because these really sick kids that die are polygenically infected. And so changing landscape, see what happens. I think we'll start seeing cerebral malaria in Western Kenya where we didn't see it previously. 100% agreement. You can buy the drinks. Thank you, sir.
Sorry. Thank you. We are running a bit late, so I think we have to move on, please. Let's apply for Professor. Thank you very much. So our next speaker is Madina, and the talk is about mRNA vaccine design for Epstein-Barr virus and immunoinformatic approach. So it's going to be online. So Madina, please go ahead and share your slides. Can you hear me? Madina, can you hear me, please? It seems that you can hear me, but we can't hear you. Madina, can you please check your...
Madina, could you please unmute yourself? Could you please unmute? Okay, sorry, unmute. Yes, okay, now please.
So, Madina, excuse me, can you hear me?
Yes, I can.
Okay, now we can hear you. So please go ahead and share your slides. The floor is yours. You have 12 minutes, please.
Good afternoon, everyone. I'm Akimboade Madinat Warala, and I'm going to be presenting on mRNA vaccine design for Epstein-Barr virus and immunoinformatic approach. Epstein-Barr virus is a double-stranded DNA virus.
Excuse me, can you please share your slide? We can see your slides, please. Okay, all right, please go ahead. Okay.
Epstein-Barr virus is a double-stranded DNA virus. It's an envelope virus and belongs to the human herpes family. Epstein-Barr virus is a very deadly virus that causes multisystemic disorders, one of which include infectious mononucleosis, cancer, and also autoimmune diseases. Some types of cancer that EBV cause include blood cancer, nasopharyngeal cancers, as well as some types of lymphatic cancer. Primarily, EBV is transmitted into the human body through body fluids, but the most common way through which it is transmitted is through saliva, the human saliva, and probably sharing of food items, and also through kissing. And sometimes it is the infectious mononucleosis caused by EBV is sometimes called kissing disease. So this, the EBV enters the host through its glycoprotein. So it has, since it is an envelope protein, so it has a glycoprotein at the outer surface of its envelope. Then it enters by penetrating through the epithelial cells of the human cells, and enters into the human cells, then replicates itself. EBV primarily targets immune cells, and commonly the B cells as well as the T cells. So it then enters into the latency stage, where it remains in the body for several years, and after the immune system has been compromised, it then reactivates itself and then proceed with its infection, and which causes persistent infection. So some of the symptoms that are associated with EBV infections include sore throat, as well as enlarged throat, skin rashes, enlarged lymph node, fever, and sometimes fatigue. So the aim of our work is to design an effective universal mRNA vaccine using bioinformatics approach, presently, there are really no approved medicines to treat EBV completely. So most medicines that are available for EBV are mostly targeted towards the symptoms. They help relieve symptoms, but not clear the organism away from the body. So our aim is to be able to design a vaccine in order to prevent the infection of EBV. So this is a walkthrough of the methodology that was used in this project. We carried out the project starting with a genetic analysis, where we got different strains of EBV. Like we worked with 75 strains of EBV genes, which are gotten from different databases such as NCBI and VBRC. So we selected and retrieved some gene sequences that codes for protein. So after that, we then tested, we selected 19 genes from EBV. After that, we screened them for their antigenicity, their allergenicity, and toxicity, after which we were left with 16 16 sequences, 16 proteins. So from the 16 proteins, we try to screen them for the ability to induce cytotoxic T cells activity. We also predicted the HEPA T cells activity, and we also predicted the B cell epitope activity. So this will enable us to channel design of our vaccine towards the eliciting immune response. So after that, we further screen the HTL epitopes towards the ability to stimulate some cytokines, and the cytokines that were considered for this project include interferon gamma, interleukin 4, as well as interleukin 10. Then afterwards, we try to look for the coverage, like what's the percentage by which our vaccine is going to work throughout the continent. Then we proceeded to trying to design vaccine constructs, and then trying to also check the properties of our vaccine constructs. So all these are carried out using computational biology approaches and So from our results, we were able to work with 16 antigenic genes from Epstein-Barr virus, which include the nuclear antigen, as well as some latent membrane proteins, and so we were able to work with them. Then we have 30 epitopes that were screened out from these 16 antigenic genes, which include B cells, 10 B cells epitope and 10 CTL epitopes, as well as the HTL epitopes. And this is an image that shows the schematic representation of our vaccine constructs, which include a 5' cap and a poly-A tail to give it an mRNA inducing ability. Then we have the COSAC sequence adjuvant, and we have some linkers that are used to link both the adjuvant and also the epitopes together. So from our studies, we were able to see that the antigenic score of our designed vaccine constructs, as predicted by Vaccigene 2.0. So we also are able to get to know that our vaccine construct is not toxic to the human body and also is non-allergenic, is non-allergenic. So these are other properties of our vaccine construct, which our vaccine has a molecular weight of 117 kilodalton, and also has about 1,997 amino acid sequences. So it has a total number of negatively charged residues of 112 and positively charged residues of 115. Basically, this chart also shows us that our vaccine is stable. So it's stable with a stability index of 35.4 from 0.4. So after that, we proceeded to try to model the structure of our protein. So we started with trying to model the structure of this, the secondary structure of the vaccine construct, where we have a percentage of 47.54% of alpha helix, and also 34.85% of random coils, which shows a very good percentage that our vaccine constructs will likely be able to form globular conformations and also induce more flexibility. So after that, we went ahead to design or to model the tertiary structure of our protein, and this is a three-dimensional structure of the vaccine constructs, as well as the Ramachandra plot, which shows that most of the amino acids of the vaccine construct lies within the acceptable regions. So like 65% of them lies within the acceptable regions of protein. So this is a chart showing that. So to show that our vaccine construct will be able to bind or to interact with some proteins that are found on the membrane of the cells. So we targeted the toll-like receptor. So for this, the toll-like receptor 5 was actually was docked against a vaccine construct using a protein peptide interface, the HDoc server. So this is a protein, and this is how the interactions look like, and this is what the interaction looks like. So after that, we were able to do a statistical analysis of the population coverage, since we work with 79 isolates from different continents. So this is the population coverage. So the total population coverage shows a percentage of 89.1%, which shows that our vaccine construct can actually be used worldwide, and with Europe having a percentage of 95.65%, and that is because most of the sequences were gotten from the European country and North American country, with lesser one from the African region. So in order to further substantiate the activity of our vaccine construct to be able to elicit immune response, so we want to check the immunogenicity of our vaccine construct. We further did an immune simulation. So the immune simulations, with our immune simulation, we're able to get some information regarding how a vaccine might behave upon administration. So we have our graph shows an elevation of immunoglobulin M and immunoglobulin G, as well as induction of different immune cells, such as the B cells, and also such as other cells, the dendritic cells, and other important immune cells that are very important for to elicit immune responses in the human body and against Epstein-Barr virus. So, conclusion, this comprehensive study is going to lay a groundwork for development of a promising mRNA vaccine against Epstein-Barr virus, and also we hope that this will also help in developing and also for testing EBV vaccine, and also provide a solid foundation for further research and clinical development in this area. So acknowledgements. So my appreciations goes to Associate Professor Elijah Kolawole and other participants of this project, which are listed here, and also for the opportunity to present this work at the WACCBIP Research Conference. Also say a big thank you to the staff and interns of Leeds Biogen Institute, Nigeria, for their technical support and sponsorship of this project. I'm also grateful for the coordinators of the WACCBIP Research Conference for providing a platform to present the research to the world. Thank you all for listening. These are my selected references. Thank you for listening.
Thank you very much, Modena. That was perfectly in time. So we have time for a couple of questions. Yes, Peter.
Hi, thank you for the presentation. I have one question relating to you showed a protein-protein docking interaction. I was just curious what, maybe you mentioned it before, how you did that docking, and basically, like, did it make sense in terms of buried surface area and the strength of the interaction?
Okay, thank you for the question. We did use a bioinformatic tool that allows a protein-protein docking protocol to occur. So after that, the result generated several dock complex, which were ranked according to their strength. So the one we actually picked was the one with the highest finding. Strength. Okay. So our result was ranked, it generated a lot of results, and we further ranked those results according to their binding affinities and this was the one we selected, the best model of them.
I'm just curious on what the tool was.
Okay, the tool was HDoc.
All right, thanks.
Is there any other question? So there is one question over there.
Yeah, thank you for the wonderful presentation. I'm just curious, in your vaccine construct, you have 2 adjuvants in there. Can you explain how they are supposed to work? Is it because they are very immunogenic? And if that's the case, don't you think you are going to have immune responses directed towards your adjuvants, which are definitely going to be expressed?
Okay, thank you for the question. True, we did use 2 adjuvants, but those adjuvants are actually the same. We make use of flagellin adjuvants, just because we want to increase the immunogenicity of our vaccine constructs. Actually, during the development stage, this can still actually be adjusted. But for now, like, we are still in the designing stage. We wanted our vaccine construct to actually be of a very good to be on a very good immunogenic point. So that was why we opted for 2 adjuvants.
Okay, I think that's cool because you may have protection to other pathogens, but maybe you might want to consider the fact that probably your immune response shouldn't be polarized towards those regions. Thank you.
Okay, thank you for that. We'll take that into consideration. Thank you for the comments.
Okay, thank you very much. We have time for one more question. Okay, so if there is no question, thank you very much, Modena. Thank you, please.
Thank you, everyone.
So I think we can stretch our legs a little bit. They have to do a little bit of configuration for the final sort of subsection, because it's still part of this section of this year's conference, which will be an alumni panel discussion. What we haven't really highlighted enough, we mentioned it, is that we wanted to really showcase our alumni. And actually, myself, Yabedia Akoya Aniwe, Lindsay, Lily Pemka, we are all alumni because we all came here with, even though we as faculty, but also with postdoc fellowships. I came with the CAN, Yao came with CAN, Aniwe came with Deltas and later had CAN also. So we are also all part of alumni. So I thought it was only appropriate as we look forward to showcase our alumni, and Dominic will remain as the moderator to keep us on track, ironically. And so I'd like to invite several members of our alumni who are at various stages of their career and were part of various cohorts. Professor Modibo Sangare. Is he here? I thought I just saw him. Dominic. He was sitting right here. Can someone call him? Oh, I meant stretch your legs here. Okay, so are we on time? No. Okay, so the next person would be Dr. Chris Abana. Okay, and then Dr. Nana Adade Egle. So as soon as Professor Sangari comes in, then we will get it going. And then after that, everything will be done outside at the closing ceremony and dinner. Just give a few minutes. Let me see if I can find him. So he's coming. So those outside, you might want to come inside. While we wait, I think I'd like to once again thank all the faculty who have chaired the various sessions. All our Jerry's been here every day. You know, he's the secretary of our faculty union. So he's a very busy man, but he's been here every day. All the faculty from Noguchi, from medical school, who have been with us, and of course all our international faculty who are always with us and always supporting. And I'd like to say, if you haven't gone on the Google Doc to score either the poster sessions or the orals, especially the orals, please do so if you received a link, because we will be collating those very soon, and the orals, as those who come every year know, will have prizes for the best presenters. So it's very important that we get enough scores for those. All right, I'll hand it over to Dominic for what I hope is a very stimulating and session. Thank you.
Thank you, Dr. Kwashi. So as it has already been introduced, this section is for alumni to discuss the impact of WACCBIP and the way forward, the experiences that we've had. So I would like to start by, first of all, thanking WACCBIP for this opportunity to have this panel discussion. And then secondly, I would like our panelists to please briefly introduce themselves, the year cohort they were in, your graduation year, if you've graduated, your current position, and how WACCBIP has influenced you to get to that position. Yeah. And I'd like to start with Nana Adade.
Thank you very much, Dominic. Good evening, everybody. My name is Nana Adade, and I'm part of cohort 5. And I joined WACCBIP, and that will be in 2018. Cohort 5 was 2018. And I have been a staff of the Ministry of Health with Kolebu Teaching Hospital for several years, and currently I work with the Orum Institute. Orum Institute is an NGO where we implement several grants in the country. I am a microbiologist, and I'm with the AMR team at Orum Institute, where I serve as a technical advisor, the lead microbiologist for the institute. Thank you.
Yeah, so thank you so much for this. Thank you so much for this opportunity. Well, my name is Chris Abana, and I came to WACCBIP in 2019. I'm in the cohort 6 batch. Prior to coming to WACCBIP, I was working at Noguchi. I still work at Noguchi before coming here. And currently, I still work at Noguchi. Yeah, I'm a chief research scientist in the virology department and working on viruses, but my interest is on HIV. So that's what I did for my PhD. I'll tell you more.
Hello. I am Dr. Modibo Sangare, Associate Professor at the University of Science, Technique and Technology of Bamako. I joined WACCBIP in 2016. I was an assistant professor, and now I am associate professor. Thank you.
All right, thank you. I am also Dr. Dominic XY Amuzu. I was with PhD cohort 3. I was the class president and currently the alumni rep. So it's a joy to chair this session, moderate it, and make sure we use this opportunity to bring the alumni together. Before I came to WACCBIP, I just finished master's. The plan was to run away to France and never come back because I wanted to do science and I wasn't getting the opportunities. Luckily, I heard about WACCBIP. I came for the research conference at that time. No, it wasn't the research, it was a workshop. I came, I saw what was going on, I was confused, but then I knew this is where I should be. And my master supervisor encouraged me to apply because I felt I was never going to be picked because they know who they will usually pick. And when I put in my application, he said, I know the kind of stuff WACCBIP wants to do, and they are looking for people like you. It was 50-50 until I got an email from Prof. Wandari, and at the bottom of it, he said, congratulations in advance. You know, for someone who wants to run away, that is encouraging to hear that from an African professor. And I think that is the kind of stories we want to hear from everyone, and this is what the discussion is about. So I'd like to start with Chris. How do you find, how did you get into WACCBIP? I just explained how I heard about it, and yeah.
Yeah, so I'm on this university campus, ahead of WACCBIP in 2014. And that there's a new center in the university called WACCBIP. And around that time, I didn't even know how to pronounce, I didn't know the full meaning. But I think I was driving around and I saw that, I saw the signboard and explaining West Africa Center for Cell Biology of Infectious Pathogens. And that was 2014. And the striking thing I heard about WACCBIP around that time was they're accepting students for postgraduate studies, being masters or PhD, and that it is highly competitive. You cannot just walk in like that. So around that time, colleagues and I, my other colleagues and I were all planning to write GRE to run away from the country. I mean, go and do a master's or PhD outside Ghana. And I remember one of my colleagues, Henrietta, who is now Dr. What's the full name again? Yeah, Henrietta Mensah Brown. I mean, she came here and she wrote the exams. Helena Chimbo of blessed memory, they were all here. They wrote the exams and they were picked. And they came to tell me how competitive it was. Like, if you don't study, you don't know your basic biology and biochemistry, you can't get in. So people thought that it was something that you can easily walk through. But the GR that we're even planning to write, I mean, people also give us a very competitive process to even get it. So I got in 2019 writing the GRE of WACCBIP. And it wasn't easy. I was told that you have to do first of all, written exam, then you do an oral exam. So the written exam, I mean, give you different aspects of biology, from biology, physics, biochemistry. That's general for everybody to go through. After passing that, then you go through the oral examination. And at the oral examination, you have to come in with a proposal that you want to pursue when you come in. And the kind of questions they were even asking you at the interview, I was like, wow. It wasn't easy, but like I said, I have been a researcher like in Uguchi, so I have a bit of experience. So I was able to answer most of the questions. And then I was there one day and I saw an email, congratulations, you've been admitted. And I was so excited. So even before I got into WACCBIP, I knew some of the faculty members who have been here, like who have just come in outside the country, they trained outside, but they were here. And what attracted me was the people were kind of, the news that kind of went to the university that if you don't know, you cannot even come to WACCBIP. People were afraid to even apply because they were like, one person told me, oh, I've applied for like 3 times, I was bounced. And I'm like, hey, you want to go? I'm like, yes. So for me, one thing that attracted me that if people are afraid to try something, that's what I want to do. I always want to try it. Let me also try it so that I'll be bounced. If I'm bounced, fine. But it doesn't even matter the number of times. If you don't try it, then you're almost like a failure. So I tried once and I got it. He said he did it 3 times. He didn't get it, then go back and learn your basic biology, biochemistry and stuff, and you try again, and then hopefully you should get it. So that's how come I got to WACCBIP. And when I got into the kind of lecturers, the teachers that were here, it was so exciting. I mean, it's very encouraging that these are the kind of people that are going to teach you throughout your time. Yeah, thank you.
That's a good one. Since we have our admission process is almost similar, I think we would like to hear from our postdoc on the panel. We'd like to hear from our postdoc on the panel about his experience of how he got into WACCBIP.
Okay, thank you. As I said during the opening day, when I finished with my PhD at the NIH, my boss was convinced I could stay at the NIH and become a principal investigator at the NIH. I said, okay, but I would like to go home. He insisted, and I said, I am done, I want to go home. You know what? If you turn 18 and you decide to leave your parents, you go out. Sometimes if it becomes so difficult, you will think of reconsidering your decision. When I came back home, I had to teach 15 hours of English course to scientists to showcase my teaching skill, because I took, to be on the safe side, I know neuroscience doesn't mean anything in Mali because I had to pioneer it. And that's too difficult. So I showcased my teaching skill in English because I realized they don't speak English. And for some reason, after 4 months, some scientists in the lab start speaking English, and their PIs were so impressed. They said, we did everything. You brought people from US to teach English. We gave them private English teacher, English book, but they could not even read English. What is your magic? And then I got the chance, the rector invited me to ask, would you like to have a position at the university to make our faculty bilingual? I said yes. I was hired as English teacher. It was good because I had the opportunity to be at the university and I can still do research. I will teach because teaching is my main hobby. When I teach, the pleasure I have, and you can feel this is just teaching is different. So I had no idea, no mentor, and I was counting on those big professors in the US, Professor Gashwin and Professor Sarah Tishkov at the UPenn. My first K43 application, they did not even look at my specific aim. I submitted, I miserably failed. It was even not reviewed, so I could not get any comment to improve. The next year, Professor Gashwin commented on my specific aim. It was better. It was reviewed positively, but not funded. If it was not Professor Jakite, I would not know about WACCBIP. And even if he emailed me, I went online to see what WACCBIP does. He said, I am doing autism. This is an infectious disease. Do I have any chance to get a shot at it? But I try. I recycle my K43, I submit it. When I received the email to say that I have an interview on that day, we have to come to Ghana to present, I start praying. I will tell you what, I said, if they give me the chance to listen to me, even if they take only infectious disease, I know they will give me a chance. Because when I was at George Washington, the first time I gave the lecture in my lab, my English, I could write, read, but sometimes I had to write on paper to show to people so that they can understand what I was saying. The first time I present, I bored my lab mates so much. People could not eat their lunch because we present at lunchtime. But I practice. When I defend my PhD dissertation, the vice rector called all the graduate students at GW. If you want to see someone presenting as typical in the textbook, do not miss the thesis defense of Dr. Sangare. So, and I start praying so that when I come at WACCBIP, people could listen me with their heart, not their ear. And God accepted. When we were going to the dinner, I asked Professor Teresa told me, congratulations, you did a nice presentation. I said, yes. And I asked, what do you remember from my presentation? She said, yes, the little girl you talk about in your rationale. I said, okay, if based on my rationale you were positive about my research, that is good. Give me the chance, I will prove that we need autism research. To make the long story short, I was so in need of funding and I had no hope. You know, you can be resilient, you can be courageous, but at some point, if it is impossible, giving up is an option. I was about to throw the white towel. You know that expression in boxing. But WACCBIP saved me. Thank you.
I think the experience is very similar to both PhDs. I don't know about the MPhils, but my experience is similar because, to be honest, I never knew WACCBIP was going to pick me because I didn't really know anyone there. And the time I realized I had to be in WACCBIP was at the interview. You know, normally you go to an interview to prove you want to be part. But it was the other way around. The kind of questions they were asking me, as he rightly said, made me realize they were actually listening. What have you done in the past? What do you want to do now? What does it mean? And all of that. And the kind of questions that were asked made me feel at home. In that interview, I told myself, I think this is my home. There is no need to run away out, especially for the last born in a family where everybody says go to US, you even have visa lottery, you've won it, but you still want to be in Africa. You can see how challenging it is, where people call you are stupid, you are not serious, you have visa, why will you want to be in Africa? So I think these are the kind of stories that WACCBIP has kind of changed for us, and I've heard of other stories that I will not want to dwell into. And moving on, I think I have to ask Nana, now that we've entered WACCBIP, how do you find the course model, the way it's structured, and comparing it to other, I would say other departments or other models where PhD is not taught, you just do a research, you have to pay fees, you don't have reagent. How do you compare that with the WACCBIP experience?
All right, thank you very much, Dominic. I'm not sure if I will be able to compare with other departments. And all I know is I don't have the same experience as you because I don't think I ever wanted to live abroad. I've always wanted to be in Africa. So I remember finishing my my master's, my MPhil at Legon, Kolebu Teaching Hospital, Legon campus. And then I had a colleague at Kolebu. His name was Seth Ajiman. At the time he was here. I think he was his first year. I think he was your mate. So he said he was at WACCBIP, and then I had finished my master's. He was asking me, what do I want to do next? I'm like, I don't know. I think I want to rest for 1 year before thinking of the next thing I wanted to do. So I was like, oh, he will encourage me to apply to WACCBIP. Before then, he had introduced me to Dr. Adodu. Dr. Adodu was my PhD supervisor. He introduced me. We were doing some work together on AMR, and then we knew each other. He was like, oh, talk to Dr. Adodu about that. You want to come to WACCBIP. So I didn't talk to him, but when the forms came out, I applied, and then we came for the examinations. I told him that this is what I wanted to do. We sat together and tried to come up with a project, and then we got into WACCBIP. WACCBIP has been, for me, a life-changing experience. I've had very good experiences with WACCBIP. The course modules for me were, how will I say it? Will I say all-encompassing? In the beginning, I think because of my background was pure microbiology. So I think in the beginning, I struggled with the molecular aspect and the bioinformatics. But with the, when I say perseverance of the lecturers and the teachers, I was able to scale through. First year was really daunting for me, but then because of the open door policy, like Dominic, you've mentioned already, every lecturer here has an open door policy. So you can easily walk into any office, ask your questions, and then when we have conferences, I'm sure at a point, most of those coming from outside, I'm sure they were tired of me because I was always going to all of them. I remember always troubling Kek. Kek is sitting down there in the audience right now. I'm sure he doesn't remember me, but my first year, if you came from outside and you presented something that I felt was in my area, I will chase you down during lunch. during dinner and ask a series of questions. I asked a lot of questions. Sometimes you get tired, but they were patient with me. And then Dominic also mentioned something about being inspired. When you look at the crop of people here, you look at them, you know that, okay, I want to be like this person. I want to be like this person. There are different people here that you can look up to and say, I want to be like. So for me, WACCBIP has been a very great experience, from the rigorous and competitive admission processes, to the lectures, to the practical sessions, and even to being able to walk into someone's office to ask for suggestion. I remember when I was doing my proposal for my, what's it called, the first proposal that we do for our comprehensive, when you have to come up with a new proposal altogether. I'm sure everybody in this department, they heard of what I wanted to do and contributed in one way or the other to shape my comprehensive proposal. So if you are here, you're a WACCBIP student or postdoc or wherever you are, just know that if you're a shy person like me, you can walk into anybody's office. You don't need to be shy. Everybody has, it will be accepted. No matter how stupid you think your ideas are, just tell it to somebody, and then they will help you to shape your ideas. So for me, WACCBIP has been a very great experience for me and has really shaped me greatly. Thank you.
So, Chris, I think maybe you can do us the honors to share your experience. What do you think about the seminars that we normally have? Some departments have only probably one seminar the whole year or academic year, but in WACCBIP we have 2 seminars a week, and on average you have 4 presenters presenting on different topics. Do you think this probably has also contributed to the quality of product or the alumni that WACCBIP is churning out?
Yes, Dominic. So for me, I would say that WACCBIP has been like that goldsmith or a blacksmith who is sitting by the fire, making sure that you're going through that fire, you're being refined. Because the training here is rigorous. If you don't want to go to it, please don't even come near. It is rigorous. And you come out as a refined gold. Because gold cannot be refined, gold cannot be pure, cannot be nice if it doesn't go to the fire. And anything in life that you see the thing in its quality, you see, WACCBIP is attractive. The brand is good. Why is the brand good? Because its quality is going through the fire. The head itself is a fireman. So you see that he will call you at any time. Like there's no. His slogan is what? Who remember his slogan?
No excuses.
Just get it done. So just like Nike, the same thing. Just get it done. Just do it. For me, just do it. And that WACCBIP training, the seminars like he's talking about, every week there are seminars here. You stand and present, and someone will just, faculty member at the back will make sure that he punches holes into your work. You get down from the stage and like you are light. You like. So I remember sometimes the ladies cry. They do cry because they come out in my class. Some of the ladies will come in. I was the class rep. Charlie, this one, it was hot. But like I'm saying, like the blacksmith, you are going through the fire, going through that refinement to make sure that, you be a fine researcher or whichever way you want to go. So for me, all the various experiences that I've had from WACCBIP has been so impactful in my life, especially the seminars, the PhD year 1 seminar that we've been having, inviting top-notch researchers from outside Ghana. I remember Jack Santa and his colleagues, they come here to teach us. And that 2 weeks was very rigorous, teaching us all the various aspects of learning, experiment, the kind of methods you want to use for experiments. It will explain to you and you understand it. And then also the experiential learning process in year 2, connecting you to top-notch research institutions in Ghana and outside Ghana to make sure that your chosen area, through your comprehensive work that you want to do and the kind of research that you want to carry out, you'll be taught more. Most importantly, we also had mentors. Every WACCBIP fellow, being PhD or master's level, has a mentor. Apart from your PhD advisor or supervisor, you have a mentor. And I'm glad Reverend is here. He used to coordinate all that, making sure that everybody is mapped to a particular mentor. And it also helps you. Maybe if your supervisor cannot see you, the mentor is always there to support you. And one thing also, I've been in this university campus for a long period, and I've seen things that have happened around. But WACCBIP gives you the opportunity. You can be a first author in a publication. It's not like other departments that the student will lead the work, and then the boss will say that, okay, I'm the first author. Then you, the student, you'll be swimming somewhere within, you don't even know. Or if your name doesn't even appear. But WACCBIP gives you the opportunity. I've seen most of my classmates leading, they've been first cohort, leading like first authors in very good journals. And I think that's a very good thing that WACCBIP does. And the training here is also so impactful. So for me, like I said, WACCBIP is that goldsmith. Even the Bible talks about it. Malachi 3:3 says that he sits as the refiner of gold. Reverend is here. He can attest. But if I'm not quoting the right verse, and WACCBIP is that goldsmith, that to make sure that if you don't want to go through that heat, please don't even come here. So yes, don't even apply at all. But if you want to. So anyway, thank you so much.
To add to what Chris just mentioned, one of the main reasons why I joined WACCBIP was in that interview where I was asked why will I want to be part of the team. And I remember clearly my response was, WACCBIP, I was in cohort 3, and so that means I'm the 3rd batch of PhD students. And I had a lot of friends who were on the program already. All of them kept saying the same thing: you need to come to WACCBIP. And so during the interview in that room, I told them point blank, WACCBIP has been doing a good job, and it has reached a stage where they want the Deltas forces, because the program was on the Welcome Deltas PhD program. So I told them, I just want to enlist as a soldier for the Delta forces, because I know they already have the Navy SEALs. So I was already psychologically prepared to go through the fire, as Chris has just said. And I was also encouraged by Professor James Abugri that it's going to be tough, but after 1 year, whether you make it or not, you will still be fine. And that's one good thing about the WACCBIP program. If you don't do well, I think there are 3 rivers to cross. If you fail the exam twice, you are not really laid off, but like the university says, you are asked politely. You might not, if this is 1 year and you can't survive it, you probably might not make it in 4 years. And so it's a very rigorous program. And once you set your mind to it, by the end of, I think, the first semester, I felt at home. Again, I think we've reached a stage where I'd like us to clap for all the international faculties that have been coming to support. The workshops really helped. I was new to malaria. I was interested in personalized medicine and pharmacogenetics. And so everywhere I pass on the corridor, I kept talking about cytochrome P450, cytochrome P450s. And then I sit in a lecture and Prof. Lasvir is talking, and I'm like, oh my God, you don't know this one. He's talking about VAR2. In fact, it was the first time I ever heard the gene called VAR2-CSA. And so you can imagine what I was going through. But I am someone who loves challenges. If you don't go through challenges, you don't really grow. It's a season. So for me, the workshop was a springboard. How to write travel grant. Professor Kegduch is here. I remember clearly he told you my CV was too long, but you know, when you are young, you want to add more stuff, want to add more chaff so that it looks heavy. And he was telling me, no, it doesn't work like that. Just stick to the point. It was also the first time we started reviewing papers. In the past, you take a scientific paper, you read a title, and you're like, nah, let me go watch Chelsea match and come back. But through WACCBIP and through the workshop, and Kirk was one of the mentors or the PI during our group sessions. And I remember clearly he took us through how to break the paper down into pieces, how to be able to know this paper is rubbish, and you don't need to waste your time on it, how to select it online, how to, the legend, the figures, and I think that is the component of the WACCBIP program that makes the students stand out, because my first author paper was from WACCBIP, and when it went out, it came back without reviewers' comment. And I remember Prof. Wandari was looking for me around 6, and I was hiding because I know sometimes I'm troublesome, so I'm wondering what have I done again. So I was hiding till around 6 when everyone was gone. I said, okay, now I can go face the music. And he told me, Dominic, the paper has been accepted. You know, it's my first time, so I don't know what he meant by the paper has been accepted. So I was like, okay, that is good. And he was like, you don't understand. Your paper has been accepted. And I'm like, yeah, is it not a good thing? He said, you don't get it. Usually, your paper will go through a lot of question reviews. They will ask you about p-values, all kinds of things. And I'm like, but Prof, I thought you trained me to write like this. And I said, Dom, you don't understand what you've done. I am a professor. When I tell you you've done a good job, you've done a good job. He stood by his car for about, he was holding his bag. I remember clearly. He stood there for some time and looked at me and said, you're going to go far if you continue like this. Most of you agree with me, in Africa, you are not likely to get this kind of encouragement. Some people might see you as a threat. You are going to be rising too fast. But here is a professor, who is my supervisor, telling me, you've done a good job. I still didn't even know it. So I started asking people, and I know that's not possible. Papers don't go out, you are lying. And then I showed them that it's here, it's been accepted. So I think it is the kind of training we had, the kind of workshops we had, how to read those papers, that is what has contributed to be able to write a good paper of that caliber. And I think I would like to now go back to our postdoc. He is from Mali. Most of us are Ghanaians. I don't want to be, discriminating that much, even though he's our postdoc on his own. Yes, Prof. Yes, Prof. Yes, Prof. So I would like to, can you share your key standout moment, the learning experience, on how WACCBIP has impacted your career?
Okay, I will just mention 2. The first one is when Professor Awanda presented he presented a table about how many grants he wrote before he got the first one, and some were colored in red, some in yellow. And I remember the one who was colored in green was 9th or 10th. Then I said, just after 2 grant applications, I was disappointed and frustrated. I know now how it works. You have to write 10 to get 1. From that point, I kept applying. Thank you for inspiring me on that. And also, when I attended the career development grant writing workshop, and we went through mock peer review, I said, okay, I understand now why and how my first grant was not a review at all. And this was a stepping stone for me for grant writing. I started putting into practice what I have learned here. That is why when I went for the first workshop as participant to the IRF Essential Grant Writing, right away the facilitator hired me as a co-facilitator, and since then I have a contract with the IRF. Thank you, WACCBIP.
Nana, how do you perceive the evolution of WACCBIP And what significant positive changes have you witnessed?
For me, WACCBIP has been okay. From I remember the first conference we had, or the first conference I attended here in WACCBIP, and I remember coming for that conference. I think that was my first time making an oral presentation at a conference. So WACCBIP gave me the opportunity to make my first oral presentation at a conference. But then I remember sitting through most of the talks, and majority of the talks were on malaria. Malaria wasn't my forte. I am a microbiologist, so sometimes I just see them like, why are we always talking about malaria? Every time malaria. But I think in subsequent conferences, I've seen that WACCBIP has progressed a bit to include other infectious diseases. At least for this conference, for instance, we see a lot of people that presented in bacteria and other organisms. So for me, and that has been a great progression from what I used to know, because in my head, or maybe even outside there, people think WACCBIP, because Prof. Wandare is malaria, in quote, malaria-centric, it looks like WACCBIP is all about malaria. But I think that as time has evolved over time, we see that other infectious diseases are now coming on board. We have different departments. Although SARS-CoV-2 is there now, we have bacteriology department, we have molecular biology, and every other department. So WACCBIP is opening up, and that's, for me, is a positive evolution sign for me. Thank you. Over.
To Chris, you've been the WACCBIP student rep. Should I say the past? The past student rep. What role do you think alumni can play in creating an environment that fosters interaction between students, alumni, faculty, and also for the growth of the community, the research or scientific community?
Okay. Yes, so that is one thing that WACCBIP does so good is it's an all-inclusive environment. Everybody is involved. So like you said, I was a student rep, representing the students on the management committee or the management council, contributing our quota students into the management of WACCBIP. Prof. Wandare made sure that everybody was involved. I mean, any problems that the students have, we discuss it at that level, and the faculty members make sure that questions about students are resolved. And I think it's a good thing, because there are other institutions that will not give you the opportunity. Because if you are running an institute like this, you need an inclusive mind from students to also help you. However, other departments will not do that. WACCBIP does that and gives you the opportunity to contribute what you have as a student. And as an alumni of WACCBIP, we can contribute our quota in so many ways. Number one, like you, Dominic, right now you are you are the president of the alumni group, trying to make sure that you organize all of us together. And the training from WACCBIP, let me borrow the words of my good friend, Dr. Aniwe. It's like you come in with a blunt cutlass, and at the end of your training, you see that your cutlass is well sharpened, that it can cut through every place, every edge. Whatever you want to do with that cutlass in life, that cutlass can help you. If you want to build a career in academia, career in technology, or whatever you want to do, you're able to do it, because that training helps you to sharpen all your skills, the kind of skills you have, research intensive, and so on. And at the end of the day, you can give back. How do you give back? You can become a faculty member too one day by supporting research or collaborating with other alumni and faculty members as well, to help teach the next generation of WACCBIP students coming up so that we continue to keep the fire in this institute burning. And I think that's one good way that we can contribute our quota as well. Thank you.
To the only post, I will not say the only postdoc, the only professor on the panel, as an academic, how can your journey and the transformative experience at WACCBIP inspire and empower the next generation of African scientists?
Thank you. So, again, when I came at WACCBIP, right away I was inspired. I said, okay, I have an opportunity to do the same thing in Mali for mental health in general, or neurodevelopmental disorder in particular. And that is very relevant. I'm telling Prof. Gordon, whatever the WACCBIP fellows have been doing for vaccine development, to improve treatment for infectious diseases, one can boldly predict that within the next 1 or 2 decades, when more children will survive from infectious diseases, we will see that neurodevelopmental disorder will be more prevalent. If you have no plan to do WACCBIP plus NDD, please help me establish the Center for Autism Research and Training in Mali. I am preparing for it. Professor Jakite is here, he can witness. I supervise 10 to 14 undergraduate students a year. I am piloting my center. If I can do this by myself, when I have other people, and from my experience in training people in mentoring, supervising, coaching, I'm pretty sure we can get it done. And at WACCBIP, I was inspired because this is a unique opportunity to change the narrative for mentoring in Africa. Peer mentoring. Those who are trained in PhD, master, and postdoc, start training them to learn to co-supervise, to mentor, so that the deficit of good mentor we have on the continent, we can fill that up. And I know, being at WACCBIP, it made people at my institution aware that I am a physician and a researcher. When Jakite was promoted vice rector, I replaced him as the secretary principal of the medical school. And most faculty thought I was an admin. I am not. And worse, most people think I was an English teacher, meaning 3 years after high school. I am a medical doctor with 6 years of neurology and 6 years of neurogenetics. So people were aware of my research capacity at my institution and nationwide. Most importantly, I can tell you, in early 2018, Professor Gashwin called me and he requested a letter of support. I said, Professor, what is this letter for? He said, the US Congress has funded my research for 5 years, and I'm applying for a renewal. To show that I have connection in Africa will be a strong argument to get funded. I said, okay. From being an English teacher on admin at my institution, now they will be talking about me at the US Congress. Thank you.
So, yeah, before we move to hear from other alumni, just a brief one. If WACCBIP tries to put up a legacy project, will you be willing to contribute a research facility? All panelists.
Okay, of course I will contribute. The reason I did not want to stay in the US because my country invested in my research up to being a medical doctor. And for being trained at George Washington, I still have my email, and each year I contribute $50. George Washington has that policy. They email you, it is voluntary. And if you look at the number of alumni and you multiply this, the minimum contribution is 50. And you can see how much it can cost. And this can serve. Some people will come here, they have no fellowship, they want to study, but for some reason before the end, they cannot afford. Those money can be used. I will be able to donate, and I will be grateful to offer my little expertise for training people in mentoring, supervising, and grant writing at the PhD fellowship level. Thank you.
Nana, briefly, so that we can move. Yeah.
Yes. So of course, it's always for me. It's always a flex to be associated with WACCBIP. So of course, if there is anything that I can contribute to make WACCBIP go higher and do better things in the world, why not? I'm in 100%. Thank you.
Yes, I'm also in 100% support in any way. So we discussed this together the last time about how alumni can contribute. Like he said, they contribute at $50. We even decided to, should I mention the amount? Yeah. We wanted to do 100 to 200 CDs a year, for the start, so that along the line we can attract more people to this legacy project, what we want to do. And like he's saying, Professor said, some students will come in and they might not get funding, and these alumni funds can be accessed to train more people to lead in their areas of research. So I think I'll be fully supported in a way. Yeah, thank you.
I have my full support already. In fact, before I even graduated, I was thinking of establishing a scholarship scheme for my supervisors. So I think it's in the right direction that we all contribute and give back. On this note, I would now like to take comments, contributions from our other fellow alumni, and maybe they can react to what do you think about WACCBIP. Is it evolving in the right direction? If yes, you give a reason. If no, you also give a reason. Thank you. Any of our alumni? I know you were here. Maybe I'll start mentioning names as Prof usually does. I think I'll start with Felix. You have been an undergrad. That's Felix's image there, if you can't see him. He was an undergrad. So he was one of the perfect alumni for WACCBIP. I always say this. He started as an undergrad, came to the lab, did a master's, accelerated into PhD, finished the PhD, is doing a postdoc. If you have a clone line, I think you'll all like, this is a perfect clone line for WACCBIP. So what is your take? Are we moving in the right direction from undergrad all the way to postdoc level? What do you think?
Yeah. So I think WACCBIP is in the right direction. That's it. Okay, so like you mentioned, I did my undergraduate at biochemistry, master's, and also PhD here. Right? And I've had several opportunities. So I've had several opportunities to travel to a conference. And after my PhD, I've also had the opportunity to join another institution to do, let's say, some research. And when I compare myself to some of my colleagues, I feel we are good. We are not bad, right? So sometimes they ask me what I do. When I tell them I did my PhD here in Ghana, they don't believe. Yeah, so they always think maybe I studied in the US or maybe in the Europe. So far, for me, I think the training I've received, even during my PhD, when I visited some of the institutions, they thought that's where I am. When I speak to some of my colleagues or some friends, they think I'm schooling there. But so I think WACCBIP is in the right direction, at least for now.
Thank you.
And I believe it's going to be better and stronger. Thank you. Anyway, I wasn't expecting you to call me, so.
I think the next question will be, as you know, WACCBIP is planning to build a new research facility. What do you think should be included? How can you contribute as an alumni to this project? And will you be willing to donate to this legacy project? WACCBIP is an international center. I always tell people this. And we've been hearing a lot from the Ghanaians. So I would like to call on Ilani Philip. You've been here for some time. And I know you are an international student as a Nigerian. Are you willing to contribute to build a research facility in Ghana? Knowing Ghana and Nigeria, we always fight over jollof.
Okay, thank you very much. So one thing I feel WACCBIP could do.
It will be good to introduce your cohort here so that others will know you properly.
Okay.
Thank you.
My name is Philip Ilani. I joined WACCBIP in 2017 as an MPhil student, finished in 2019, and then came back for PhD, 2022 cohort. I'm in my third year PhD. So one thing I feel WACCBIP would do in the new institute, the new building we're talking about is having core research facilities. We have different PIs working on different projects, but there are equipment that cuts across, that people use across board for whatever you are doing, cell sorting, microscopy, and other equipment. So I feel it will be a very good component of the new project if we could have core facilities that every lab could use with very little grants that you can use for consumables. You know that once you get into the building, you have access to these core facilities and you could do experiments that you would want to travel to London or other places to go and do. And yes, I'll be willing to contribute to such projects. I could make a pledge now worth $3,000.
I hope it's being recorded, you know.
And yes, that might not come in cash, but I've got equipment that is worth that amount, and I'm I'm willing to leave that behind if it will be of any help. I got my pipettes worth $3,000. So that's something. That's something to start with. But yeah, it's a good idea, and I'm willing to support. Thank you.
Well, looks like we've been roaming around in West Africa. So, Dr. Collins Misita Moranga, my partner in research crimes. Could you also react to that? Are you willing to leave Kenya and come and put some of your money into a research facility here in Ghana?
Yeah, thank you, Dominic. And I think it's a great opportunity to say that as WACCBIP celebrates its 10th anniversary, it's going in a very good direction. So for the new building, I think currently as WACCBIP is set up, I think we have almost everything that we need, probably except maybe the animal research unit. So probably in the next building, that will be a priority, as well as a unit for, you know, highly infectious pathogens, containment units, and those kind of things. So thank you very much. And I'll be willing to support as much as I can.
Thank you, Dr. Moranga. Since Collins mentioned animal modeling stuff, we have an expert in the room. She's in the person of Constance. Are you willing to donate your skills, time, teaching, to help WACCBIP grow in that direction? Yeah. So if you're an alumni, get ready. I know everyone.
Okay, thank you. Yes, I'm willing to donate my intellect, my skills. I have skills in animal experimentation, like Collins mentioned just now. So I could help WACCBIP students who are doing any research with laboratory animals. I will help in that direction. And I also have skills in wildlife sampling. So all those who will be interested in pathogens of avian origin and would want to sample wild birds, that is my passion. And so the other thing I would want to add is that in the new building, if WACCBIP would consider probably putting together those research areas that are not mainly within the scope of the facility, which are falling outside of human malaria, HIV, and all these. Some of us are working in the areas of biodiversity, wildlife, disease ecology, fisheries, and the likes, so that they can also put those of us doing all those nice things together. Then we can evolve in that line and train the next generation of molecular epidemiologists under the One Health concept. Thank you.
I think it is fair to move to Dr. Irene. Yeah. I know you are a great teacher, both in the lab and out of the lab. And so are you willing, this is just to find out for our alumni, if they are willing to volunteer to not just mentor, but to teach, guide, because we had a lot of internationals come down to help us. Yeah.
Okay, thank you, Dominic. Yes, I may not be able to contribute in cash, but yes, I'll be happy to contribute my expertise in training and then giving, of course, producing antibodies for sale. Maybe we could produce and sell to generate funds internally. And one thing I would also want to say about the new facility is that it would be nice to, when the plan or design is ready, we have most of our alumni who have worked outside facilities in WACCBIP, and so we know different and more convenient facility designs. It would be nice to publish the design that would go into the building and give a time limit for fellows and alumni to contribute or to comment if necessary, just to make it a more encompassing and easy to use facility. Thank you.
So just last two, because we are running out of time. So I'd like to call on Sultan. I think we've not heard from any intern, and it is just fair we hear from the youngest of the youngest, yeah. Sultan, yeah, he's at the back. Yeah. Just a quick one, yeah.
Thank you. So I'm glad you at least mentioned an MPhil
because the MPhil students were whispering in my ear that they felt a bit left out. So we're gathering a rebellion. So I'm glad that at least that ends it. So I think, are you asking specifically about our contributions to the building, or that's okay? So like I said, we are the youngest, so cash-wise, maybe not yet, but I think it would definitely give an opportunity to train more students, and we are still early in our career, trying to find our way. So definitely more opportunities would come from that. And whatever ways you think we can contribute, I'm very interested in seeing how the alumni can come together and form a very well-founded and well-grounded organization. I think avenues for how we can contribute will grow from there. If I can give a contribution to how the plan would be like, at least a bigger conference room would be nice, and a bigger washrooms as well. Thank you.
Thank you, Sultan. And just to finish it up, I think we all agree a lot of people are willing to contribute. So maybe as an alumni, we plan on setting up alumni research fund, but I think we also have to consider a GoFundMe so that we can solicit for more funding, not just for what we want to do for our research fund, but also for the WACCBIP project. And on this note, I would like to call on the alumni of the Alumnus, Professor James Abugri, to give us his final thoughts, because he has been in WACCBIP, I think the first WACCBIP PhD or something. Yeah.
It has been a pleasure being here. I will want to quote I want to give a quote from Alexander Pope's poem. It says, "Drink deep or taste not the Pyrenean spring. There shallow draughts intoxicate the brain, and drinking largely sobers us again." WACCBIP has provided a conducive environment, has brought together critical minds that would allow the development of the great minds for the future. It is only a bigger opportunity, a nice place. Once you are here, as the quote says, you drink deep or you taste not the Pyrenean spring, because the shallow draughts would intoxicate the brain. But when you drink largely, it sobers us again. So once you're here, immerse yourself totally in the opportunities that are provided. I am so happy to have been associated with the inchoate sages of WACCBIP when it was asked, what would excellence be like? I can say this is a personification of excellence, with a lot of the products doing greater things. And I hope, I've been trying to link up with most of you. I think that together we can do bigger things together. WACCBIP is here to stay. WACCBIP will go from strength to strength, and we wish WACCBIP all the best in all facets. Thank you.
At this point, we'll take our final comment from our panelists, starting with Nana.
Thank you, Dominic. So we've all said nice things about WACCBIP, all the beautiful research everybody is doing, how WACCBIP has impacted our lives. But I think that if the new faculty that is being set up, if there's a way for an advocacy and policy department, you may do all the nice, from our experience, from the work I'm doing currently, people do nice research and everything, and then it doesn't influence any form of policy. So what we do is we try to take the research to the door of those that matter. So, but then if we all sit in our little corners doing all the research and it is not influencing policy, our world will not be a better place, or it will not have the impact that we desire. So I think in the new, going forward, if there's a way where we can incorporate some form of policy and advocacy training to maybe like a department or some form of training in the courses, that would really help. And finally, I want to leave you with something I learned from Prof. Wandari. The first class he taught us, that was in 2018 or 2019, when he came to the class, he stood and looked at us and said that you must stay focused. And there's something he said that has stayed with me all these years. He said, some of us, if you want to do research, you must be focused, because some of us, sometimes someone hurts us, or someone has done, maybe you are in the bathroom, for instance, or you are still, the only thing you are thinking of what Constance did to you yesterday, you're thinking of what Angela did to you yesterday. If you continue thinking of what everybody says about you, the negative things that people think about you, you will never be focused to do research. And that has stayed with me all these years. So if you want to do research, you don't have time for things that are not research oriented. So focus on what you want to do and forget about what doesn't contribute to your research. So I'm leaving you with that. And thank you, Papa Awandari, for teaching me that. Thank you. Over.
I just want to say that, like I said previously, if you want to be like gold, then please come to WACCBIP, where you'll be refined, and it will help you in every part of your life after graduating from here. So my simple and final words is that please come to WACCBIP, those listening to us online and all over the world. This is the best place to be. They don't run away to US or Germany or anywhere. Come to this place, because the best of the best are here. I mean, most of the faculty members have been trained from US, from UK, and all the other places, and they're all here. So please come to us, you'll be trained well, and you come out as pure gold. Thank you.
Okay, for my final word, thank you, WACCBIP. Thank you, Professor Jakite, Professor Gordon. As I said, I am really inspired. Whatever WACCBIP is doing, we need the same thing for mental health. If Prof. Gordon has a plan for WACCBIP plus neurodevelopmental disorder, I will be available. But I want to replicate what is going on here in my country. And most importantly, there is a need. We have to be focused, but there is a need to look into the connection between infectious and non-communicable disorder. There are many grants for that, and it will bring more people, give opportunity to more people to come from abroad to do research on the continent. Thank you.
At this point, I would like to use this opportunity to thank all those who have contributed to organizing this conference as WACCBIP celebrates its 10th anniversary. I also like to thank WACCBIP for making this place what I call a research home to many of us. And also would like to thank the ICT support. Without them, a lot wouldn't have been going smoothly. Over the years, we have improved. From time to time, the pointer and all that was on point. And also would like to thank the faculty, both the international faculty, for taking time out of their summer holidays when they could have been in Barbados and the rest chilling, but they are here with us, supporting us. So thank you so much. And our local faculty also. On behalf of the alumni, we say thank you. To our alumni community, this is the time for us to be well organized, to showcase what we've been made to become. And who are we? We are great scientists. I believe we can do this. We showcased it during the COVID where we were students, but we were leading the fort. And so I would like to end with this quote. Alone, we can do so little, but together, we are infinite. And as Professor Wandari says it, let's just do it. Thank you all.
All right. Thank you very much to our alumni panel. So let's give them a last round of applause. I hear the beer is ready, and if we let the beer get warm, Lars would not be happy because he has a flight to catch this evening. So we will not allow the beer to warm up. So we're going to go very soon. So I think we've had a very entertaining, enriching, and enjoyable 1 week together, right? I hope everybody has found the conference to be worth their time and effort. I want to thank all of you for taking the time to join us, for contributing to the discussion, for being good guests and good hosts, all those who are also our students. They are also co-hosting with us. We want to appreciate especially our collaborators and international faculty who joined us here today. So let's acknowledge all our usually we do an evaluation, an oral evaluation, in addition to the written evaluation. I hope they've sent an evaluation for you. Have they? Oh, not yet. Okay. They'll send an evaluation by email, which you can do. So please look out for that. But normally we take quick comments from those who have traveled to come, especially those who are here for the first time. If they have any impressions, good or bad, we're happy to listen to both. So any comments from our friends who are here for the first time? Yeah? Okay. Wellington has some comments.
Well, thank you very much. The feedback I have to give, really, is a very positive one. Very, very positive. I may not have had the opportunity of comparing the years, but I mean, looking at the depths of intellectual discussions, especially within the growing academics, I'm talking about those at the graduate level, is globally competitive. And I really saw a resonating of very strong leadership, research leadership pathway amongst the participants in the center, which of course is cross-country. So we're seeing people from East Africa, we're West Africa, and then of course the willingness of the international faculty to take these guys on. And that machinery, which I've had you talked about most times, I've seen within the people machinery, and I've also seen within the equipment machinery, because I went through the labs last night. And the equipment machinery, I think, is huge. It's huge, and I could imagine the hard work that is coming into getting all of these things working. So the only thing I can say that we are proud, I am personally proud of these achievements, celebrating 10 years. And because again, we've been to different places too. And seeing what we have here is a great pride to Africa. And a salute to our colleagues from the other parts of the continent, expressly showing that friendliness, that liberty of knowledge and tech transfer. So I'm delighted. And I will say congratulations.
Thank you. Okay, Dennis, yeah.
So it's my first time at WACCBIP. I mean, it has been an amazing experience, an amazing week, I have to say. So I think I'll repeat one thing that was just said, that is, what is very impressive is this community of countries. I mean, this is absolutely impressive, so, and very rarely seen, I have to say. So that's absolutely impressive, that it's a community of countries coming together at WACCBIP. So that's truly impressive. And what is also impressive is that I know that it's not going to just be at this level that is already excellent. It's going to grow and grow more. And so never stop, never stop growing, because it's just it's like a rocket. So congratulations.
Thank you, thank you, thank
you. Thank you very much. It's my first time in Ghana, so I've eaten a lot of jollof rice. And, and I think
I'm surprised that you are the first to mention the food. I thought everybody would have mentioned.
Now I have my own personal verdict, which is better between Ghana and Nigeria jollof. But on a serious note, I think this has been a very great week for me personally, because in my career, I've never been to a place where I've learned this much, I've interacted this much, and it hasn't cost me less than $500. So, and to be able to do all of this for free, I think it's great. And congratulations to WACCBIP for all the organization was excellent. So when I was talking to the camera people outside, I said it was very malaria-centric, malaria-intensive, but I heard somebody said over the years, so they have diversified to other infectious diseases. So maybe I take back my comment earlier, but I think...
Yeah, you can move here and bring more TB.
Well done and congratulations on your 10th anniversary, and please keep doing the good work. Like yesterday or the past few days, when I walk into the poster room, the young ones who are presenting, they are running and pulling you to their posters, just being excited, talking about their work and what they are doing. I think is a good sign that the knowledge that they are getting and the training that they are getting is very international and can take them very far. But thanks a lot for it.
All right, thank you. All right, so I think it's time to wrap up. Three fun things to talk about. First is, let me commend all of you for the energy throughout this conference. There's been a lot of energy. So let's... And the reason why I realized that there was a lot of energy was around the WACCBIP cloth. Yeah? This cloth, we got it on Friday, just a week today. And it was like a challenge. Who can get their tailors or seamstresses to get something sewn before Monday? And on Monday, we had so many people already wearing it. In fact, our comms team also did a fantastic job of getting, I can call it a factory, to produce 8 T-shirts in 48 hours. And, since Monday, people are, buying the shirts and sewing, buying the cloth and sewing, and there's just been so much energy around the cloth, and I want to appreciate that level of support. Thank you so much. It may look like a trivial thing, but it's sort of a rallying point for us to be able to all identify with what we're doing here through this fabric. So let's keep the fabric and wash it and wear as many times as we can, especially anytime you are coming to WACCBIP. Make sure you have your WACCBIP uniform. So thank you for that. Second thing we want to do, I want to recognize one of our students. Mami, please stand. How many of you are on Twitter? How many of you follow her. If you are not following her, you are lost. She's just amazing, the kind of talent that she has. She sits here and every talk, she turns it into a poster. She draws it up with all the messages and everything. It means that she's focused. She's listening to everything that's being said, you know, and she's talented enough to transform that into a visual within minutes. I mean, this type of talent, I've never seen some before. So, yes, in fact, the reason why I've called her up is that we are up for business. If you, I mean, this is science communication at its peak, right? So if you need a science communicator who can do cartoons for you in minutes, you just tell her the message, she will do cartoons for you. See me, don't see her. I'm her manager, so we'll arrange a contract. So please follow her if you are not. And we are going to compile all her drawings and put it together and publish it online for all of you to have, you know. It's part of the legacy of this meeting, and we really are really grateful. Thank you so much. All right. Finally, we had some people celebrate their birthdays. I'm sure many people did, but two of us, our colleagues, celebrated their birthdays. Dennis celebrated his birthday. Was it yesterday or 2 days ago? Dennis, when was your birthday? Yesterday or the day for yesterday? Okay, that's 2 days ago, right? So let's wish him a happy birthday. And today is our own Peter Kwashi's birthday. Where's Peter? Peter is missing. He's busy arranging the beer, so we'll join him there to celebrate. So it's good that, you know, we've had these very important events in their life whilst we were here together. So we would like to add that to the toast for the dinner this evening. So once again, thank you all. I want to thank my team, the machine, right? The WACCBIP machine. Yeah, let's acknowledge the WACCBIP machine. The reason why we are successful is because of the machine. We just have to think about ideas and then they go and do it. Okay? We can just think overnight and say, tomorrow let's do this, and they'll get it done. So they're really amazing, and we're really lucky to have such an efficient and committed team. So thank you to all of them. I want to thank all our students and our fellows who made it here, especially the alumni who traveled specifically to come and join us celebrate. We're grateful. You're part of the family. I'm glad you are organizing yourselves. Let's see how you can help us put up a new building to add more space so your siblings can come. And then finally, we want to thank the funders. Everybody has noticed that you don't have to pay to come to this conference. That's made possible because of our funders. Most of the grants that we have are capacity building grants. And even those that are not capacity building grants, we are able to convince our funders to put some budget line to support our conference every year. And that's how we're able to, you know, pay for everything so you can be here without having to register. It may get to a point where we have to ask people to register and pay a registration fee so we can keep going. But for now, we are very grateful for our funders, and we want to appreciate all of them. Obviously, their support was complemented by the sponsors. So we had quite a number of sponsors, especially those who are suppliers of reagents. So they contributed a significant amount of money to help, you know, us defray the cost of organizing the conference. So we are grateful to them for their partnership. So let's appreciate them as well. Then our speakers, all the speakers, we were very grateful for taking the time to prepare presentations to speak here, especially the keynote speakers. These are all, you know, outstanding individuals who are exceptionally busy, but they took time to come here and be with us and give talks. we are very grateful to all of them. And we hope that they will be back soon and often. So thank you very much to all the speakers. All right, so we're done. WACCBIP at 10 is over. See you.